Sorry to interrupt your conversations. Could you please take a seat? Good morning and welcome to the final day of Science at the Shrine Dome 2024. I would like to begin by acknowledging and paying my respects to the Ngunnawal people, the traditional owners of the lands on which the Academy office is located. I also would like to acknowledge and pay respects to the traditional owners and elders past, present, and emerging of all the lands on which the Academy operates and its fellows and staff live and work. They hold the memories, traditions, cultures, and hopes of Aboriginal and Torres Strait Islander peoples of Australia. I'd like to ask guests to place their phones on silent, but we encourage you to share this event on social media using the hashtag ShrineDome24. Thank you all for joining us today to celebrate our honorific award winners. For those who have been with us during this week, I hope that you have enjoyed Science at the Shrine Dome so far. And those who are joining us for the first time, welcome. In our pursuit of advancing science in Australia and abroad, celebrating outstanding scientific contributions is at our core. Earlier this morning, I was honored to meet winners of the Academy's Aboriginal and Torres Strait Islander Scientist Awards and Max Day Environmental Science Fellowship Awardees. It was a privilege to meet so many talented scientists and recognize their hard work and commitment, which serves as an inspiration to us all. It was also an opportunity to thank many of the donors to the Academy who make our awards and our work possible. We continue our celebrations today with the awarding of honorific and career awards. Honorific awards recognize contributions at the early and mid-career level, while the career awards recognize lifelong achievement. This year, we'll be celebrating award winners from 2023 and 2024. The magnitude of achievements that we are celebrating today is truly staggering, and it is something we would not be able to do without the support of our generous donors and sponsors. Before we hear from our awardees, please join me in acknowledging our event partners. The list is long, but please indulge me as I mention them by name. The Department of Defense, CSIRO, Department of Industry, Science and Resources, the University of Sydney, the University of Queensland, the University of Adelaide, Western Sydney University, Deakin University, the University of Melbourne, Australian Institute of Marine Sciences, Queensland University of Technology, Proto-Axiom, ANSTO, Australian Research Data Commons, the University of Western Australia, Swinburne University of Technology, Science and Industry Endowment Fund, Macquarie University, University of New South Wales, Sydney, NCI Australia, the Granted Group, and Victor Chang Cardiac Research Institute. We are very fortunate to have had the generous support of our event partners to bring Science at the Shrine Dome to life this year. In particular, I would like to acknowledge CSIRO, Australia's National Science Agency. CSIRO are one of the platinum partners and have been supporting this event and the Academy more broadly for many years. Unfortunately, Academy Fellow and CSIRO's Chief Executive, Dr. Doug Hilton, is no longer able to join with us. Please enjoy this CSIRO video.
Every challenge contains a seed of opportunity. Opportunities to improve life for all of us. We know this because we've been trusted to solve challenges for more than 100 years. We are proudly and uniquely placed to solve these great challenges. through innovative science and technology to unlock a better future, better for our community, our economy and our planet. CSIRO, Australia's national science agency.
Now, to begin today's awards program, I would like to invite Professor Malcolm Sambridge, our former Vice President and Secretary for Physical Sciences, to introduce our first award recipient.
Thank you, Jagadish. Good morning, everyone. The morning after the night before, I believe. So I'm honoured to be here today to introduce the 2023 Matthew Flinders Lecture. The Matthew Flinders Medal and Lecture is a career award that recognises scientific research of the highest standing in the physical sciences and honours the contributions of Australia's early scientific researchers. Along with Macfarlane Burnet and Ruby Payne-Scott medals, it is one of the most prestigious career awards of the Academy. The 2023 winner of the Matthew Flinders Medal and Lecture is Professor Lidia Morawska. Professor Lidia Morawska's 30 years of innovative work brings us closer to breathing safely. The fundamental science that she pioneered and advanced in the multifaceted field of air pollution is critical for humanity to understand pollution and its impacts, and to build bridges translating science into the public health applications. This work laid the foundation of the 2021 World Health Organization Global Air Quality Guidelines, which included recommendations on ultrafine particles from combustion processes for the first time, providing authorities around the globe with the basis to develop regulations to control this pollutant to improve health and save lives. Professor Morawska's seminal work on particles for human respiratory activities became critical during the COVID-19 pandemic. In recognition of the importance of aerosol transmission and convincing the WHO and national regulatory bodies to review public health policies and practices from school places to workplaces, making these environments safer for more people around the world. Lidia received her medal last night at the gala dinner, and today we will hear her lecture. Please welcome Professor Lidia Morawska.
Thank you very much, Malcolm. Good morning, colleagues. I hope that there's still a little bit of space left in everyone's brain to listen and absorb a bit more. But after this wonderful 3 days, the fourth day is becoming overloaded. Well, I'm extremely proud and, as I said yesterday, lacking words for having received this award. And I'm excited to share with you my career. This is a career award. So share with you my career, which means share with you a little bit about the science of the air. But I will start with art. About 3 years ago, the Queensland Gallery of Modern Art, GOMA, was in the process of planning an exhibition, Tight Air. Well, they reached to me asking to help a bit with some, of course, science, not art, of this. And eventually I was also asked to write an essay for the book of this exhibition. And then I realized how difficult it is to talk about something which is invisible. Now, art, visual, it's visible. And I'm talking about presenting something in the art which is not visible. So I started my essay by inviting readers to imagine that they are standing at the top of Mount Coosa in Brisbane, looking at a distance, clear, crisp morning, Northern Ireland, well visible. And I ask a question, okay, is there anything between you and the island? No, nothing. Well, what about this 30 quintillions molecules in every centimeter cube of the air? Is it nothing? Well, it's even more difficult when you talk about indoor air. Is there anything here we should care about, should be interested? Is there any science in this? On one occasion, when giving a presentation to colleagues from a different field, I presented these photos and I said, well, at QUT, we have a program on designing future buildings on Mars such that they will provide good indoor air. Oh, this attracted attention. Mars, wow. Well, they were very disappointed to learn that these are indeed photos from QUT, but this is ventilation system from one of our already sophisticated building at QUT. So, and it provides to that building good indoor air quality. Is it interesting? Well, what I'll do in this presentation, I'll try to present a big picture of science of air. So look at the slides as pictures. I'm not going to be talking about scientific details of them, but just to give you perspective on the breadth of this air, of this whole field. Well, as I said yesterday, I have a degree in nuclear physics, and somehow during my master and then PhD, I got attracted to environmental radioactivity. So I got all my degrees from the Jagiellonian University in Kraków in Poland. And then I continued this work at the University of Toronto, also at McMaster for a shorter period at McMaster University in Hamilton. So my focus during my PhD and then continued at the University of Toronto was radiation of the air, radon, and its progeny coming from radium in the soil. One of my first papers on this topic was influence of sealants on radon emanation from building materials. Now, there were some other aspects of this. At first glance, this may not relevant at all. That was what I was doing at McMaster uranium series dating. So it's still physics of radiation. I didn't do much later in this area, but this came very useful understanding of these processes. And one big step forward when working on atmospheric nucleation processes, which are some of the most complex to understand. One pathway is ion-induced nucleation. Those ions come from all kinds of sources, including combustion vehicle emissions, but one very interesting pathway is radon coming through the roots and then trees and then enter into the atmosphere, and this way we've got additional ions in the atmosphere. So this is my earlier period. This brought me this understanding of interaction between radon and radon progeny and particles in the air, which is crucial whether they stay in the air or not, brought me to the science of air, and that's what I've been doing at QUT. So I came there with a firm view of, or idea of establish a unit researching this, which initially was called Environmental Aerosol Laboratory, then International Laboratory for Air Quality and Health. And recent addition is Thrive program, specifically focused on indoor air. So over the period of over 30 years, I came to QUT in 1991, we've basically researched any aspect of air in any environment from any perspective, from using any kind of instrumentation. There are just some images. But the focus and our main area of expertise are particles in the air. Most of them are very small, ultrafine, smaller than 0.1 micrometer, or even less, few nanometers. Combustion is the sources of these particles, particularly in urban environment, but not only. This nucleation and these banana curves, which we can see here, banana, that red thing is banana, when particles form and then they grow, and then that's how it looks like. So again, we've looked in every aspect of these particles and interaction with everything else. We investigated these particles and mechanism of formation and trends with colleagues around the world. This is one of the papers of one of my PhD students. We've also looked at exposure and what it does, with a specific focus of children exposure. This was a project where we looked at children in 25 schools in Greater Metropolitan Brisbane area, and we looked at in health surface area dose, relative intensity in different microenvironments. And this allowed us eventually to conclude what it does to human health. And interestingly, we found out that these particles have mainly the systemic effects, not respiratory, what we would affect, but systemic. But this is because they get deeply into the respiratory tract. Well, but there have been all kinds of other things, like this discovery by a chance, in a way, about what happens when we fracture concrete. Well, you can imagine clouds of dust, but that was not that interesting. The most interesting was that we found even more particles which are formed by nucleation and high temperature emission of some volatile products. Or this experiment, which we were able to do thanks to this massive dust storm, which cleaned the air, in fact, of ultrafine particles, well, because they attach to larger particles. And I had to stop our media saying that the dust storm cleaned the air. Well, cleaned the air of ultrafine particles. This also allowed us to look at the very broad picture of what's happening in terms of air pollution. And here with colleagues, again from around the world, we looked at the frequency, intensity, and duration of such episodes, noting that, well, discovering that the situation in Australia is deteriorating, and this is because of bushfires. All this led to our contribution to global burden of disease studies. I didn't lead these studies, but that was the first one which placed outdoor ambient air pollution and risk number 9. Since then, I contributed to several tens of these studies where our understanding of particles is an important contribution. In all of this, instrumentation was critical. Instrumentation very sophisticated, but now we are in a different era where we need also instrumentation for scale up measurements and mass. This is a low-cost monitor, Koala. I came up with the name, but one of our master students with that abbreviation where it came from. We first used it at the Commonwealth Games where the network operated. So, okay, you can buy low-cost sensors. There's one sitting there on my desk, commercially available. But the point is, when we are talking about low-cost sensors for particle measurements, it's a whole complex science because they are optical sensors. They don't measure gravimetric mass. So translating this into exposure response curve, and which then eventually led to the WHO guidelines, is quite complex. Now this Koala eventually populated Australia, and beyond we had networks of this Koala operating in many different places. And eventually, that was mainly outdoors, but eventually that was this paper when we start talking about indoor air koalas. I didn't realize then how important this was going to be. That indoor, again, indoor has been part of our work all the time, since the beginning. Since, 4 years after I came to Australia, I organized this conference, Indoor Air Integrated Approach. We only now start talking about integrated approach. And we've assessed sources, emissions, pathways of exposure in basically any kind of environment. Discovering basically every time something interesting, like in these schools, these massive concentrations. Where did they come from? Eventually, we found out that was nucleation of particles, secondary particles from paints and other arts products which the kids have in the hospital. In surgical theaters, this very suspicious situation was then related to outdoor pollution and how it penetrated to the surgical theaters. We always looked at this integrated indoor environment, which means it's air, pollutions, particles, mechanism, what does it do to the whole system and understanding the system, including energy. And not only in Australia, but beyond, and not only working with colleagues in other developed countries, but investigating exposures in countries like this, in Bhutan and Laos, PDR, where the situation in terms of indoor air pollution and outdoor air pollution is quite significant. Some of these papers here are in Laos with my colleague, close colleague Kerrie Mengersen, who will be speaking a little bit later. Now, but there was also a very different aspect of this physics of respiratory infection transmission. What does it have to do with all what I've discussed so far? Everything which we emit during all our respiratory activities are particles. So before anything else happens, these are the particles. SARS-1 brought our attention to this field, and this paper was the one which was considered later and now fundamental to, as an argument against the WHO. We summarized our work in this field in the mechanisms, sites and mechanisms of respiratory particle generation recently in this Nature Physics Review paper. So you can see how many different processes happen there. And then there were printer emissions. What this has to do with anything. We were doing a study in one of the government office buildings in Brisbane when we encountered this huge concentration of something. Well, everybody and everything was a suspect. Somebody smoking, what was it? Eventually, we linked this to printers, but there were the questions, what were the mechanisms of this? To answer this, we had to line up a whole system, including the tunnel which we designed for studying human respiratory activities. And instead of human head, there was a printer sitting there. We also then needed to do some chamber experiments. The problem is that any particles, as we suspected that there were secondary particles, they are liquid, you cannot collect them and put under a microscope. You have to somehow investigate them differently. So this volatilization, humidification, then the mobility analyzer, which we designed, served for this purpose. And it eventually led us to the answer that these are secondary organic aerosols and what those particles, and we narrowed down on the particles. This was the huge deal. This was in August, summer holiday in Northern Hemisphere, and printer manufacturers who were on holidays, as we were told, were brought back to deal with this issue. So from science to practice, influencing change to help us thrive, starting with the printers. Printer emissions 10 years later, we did measurements of a large body of printers, and we were pleased to find out that the emissions quite significantly decreased. Well, we knew then that printer manufacturers took it seriously, what we found out. The work with the WHO and ultrafine particles. In the 2005 edition of the guidelines, there was only that one statement about ultrafine particles. There's toxicology, but not epidemiological evidence. Now, that situation continued. There was no epidemiologic exposure response curve, but we knew so much. So I set up this team, which we called ourselves Thinking Outside the Box team. We generated this white paper, and this white paper became now a chapter and de facto recommendation in the recent edition of the WHO Air Quality Guidelines in 2021. It helped that I was a co-chair of this activity, but it went through a very stringent process. Now, then came COVID, and when I've heard WHO and public health authorities, including ours in Australia, saying that the virus is not airborne, I said, what a nonsense, what a nonsense. We know that it is. Well, these were the most, perhaps, difficult 3 months in my career, I thought then, to convince the world about this. With a group of 36, there was the group which I established, and with support of a larger body of scientists. So the next day after this paper published, the WHO acknowledged that the virus is airborne, and the wheel started turning the other way. So with now bigger focus on indoor air building designs And this pointing out that we need better buildings to provide better air, and we have the designs. So this was that Science Policy Forum. And the recent edition is the blueprint, a blueprint basically how to do it. This is not an opinion, it is a blueprint exactly how to do it. If countries lead by example, standard maybe increasingly became normalized. Now, we talked about this topic a few weeks ago with our Prime Minister. One common factor of this is scientific consensus of large interdisciplinary and international group of experts. So that was my work, bringing people together to do this. So we started with art, we finished with policy, and this is towards clean indoor air and outdoor air for all, based on science. So I thank you for listening, and add to the thank you note which I said yesterday to the Australian Academy of Science, Queensland University of Technology, particularly that I was allowed to do what I wanted to do, and I had a vision to do these things. And to colleagues from around the world, colleagues in this room, if I wanted to put photos of these groups and talk about the groups, this would be the whole presentation. Thank you very much.
Thank you, Lidia. A sweeping talk there. Difficult to convince people of something you can't see. That came across for me. So we're going to open the floor to questions and also online. If anyone wants to submit one. Yes, we've got one here.
Thank you for a marvellous presentation of absolutely world-changing research put into practice. Thank you so much. Has anyone from the space station asked you to comment, how are we going to cope when we're living outside and recirculating air, presumably for years, possibly?
No, not yet. I've been contacted by many different industries, including aviation, but that thought probably hasn't occurred to anyone yet.
Francis?
You work, and I'm sure the guidelines include details of how you place the instruments that you measure the air quality, and I was wondering how much it would vary depending on where you place the measuring instrument in the room.
Well, of course, it varies because of the flows of where the air is delivered, how much air is delivered. But this is not the biggest issue in all of this. One of the biggest issues was in this document, we suggest just to monitor three pollutants, PM2.5 particle, which is particles CO and CO2. Now, anyone hearing about this, but there are so many more pollutants of importance on our health. So why don't we do this all? The reality is that we cannot put an air monitoring station in every room, and in every room we would need to have monitors like we have smoke alarms. So we need to have a very different approach to doing this. As I mentioned before, low-cost sensors and scientific approach to how those millions of sensors calibrate themselves and how they interpret the data, what's happening. So this is really the problem, not that much the location of an instrument in a room.
A great talk and congratulations. I'm just curious your thoughts on the main knowledge gaps or issues regarding bushfire smoke and air quality and human health?
Well, as I mentioned when I talked about that paper, where the bushfire smoke are clearly decreasing overall air quality in Australia. The problem is that unless we do something about and rapidly about climate change and reducing emissions transmission to clean energy, the situation is not going to improve, but even if we did it from today to tomorrow, still we will be in that situation for quite a long time. So the problem is how to adapt ourselves to this, and here the role of buildings is extremely important because buildings should protect us. But you may remember there was a photo during the bushfires 2019-2020 of journalists broadcasting from outside the studio because inside the studio, a smoke alarm went off because of smoke that penetrated, which means that that studio and many other buildings do not protect us from smoke. So this is something extremely important, how we will protect ourselves inside buildings against bushfire smoke.
Eric. So next over there, one here and then back there. Eric.
Thank you for a really fantastic talk, Lidia, and congratulations too. My question is, it's really great to have sensors, obviously, to detect air quality, but imagine if you've got one in your home or in your school or in your workplace and it indicates very bad levels of whatever in the air. What can you actually do about it?
This is a great question. It is the question, what can I do about the problem? Let's say I'm here, actually, it's quite good. But if I detected a problem here, what am I going to do? I'm going to complain to someone, but this is not resolving the situation. The point is that this is not individuals' responsibility to make sure that indoor air quality in public buildings, that's what we are talking about, is good. But this is a responsibility of the operators of the society. So therefore, this knowledge of the sensor is not just to alarm you, but it is to be a signal to the building operators that something either is not working because it's a fault, which we see, or something has to be improved, or if nothing can be done in terms of improving ventilation, delivery of clean air, so-called equivalent ventilation measures should be taken, and here we are talking about air purifiers or something like this. But as I said, this information is to be used to do something by experts and not by individuals. But it helps individuals, and how many times I will go with my sensor complaining in different situations about sometimes they look at me like an alien.
Perhaps you've partially answered this question, but there are many domestic and commercial air purifiers available which claim to remove particulates. Do you have any comment about whether they're effective?
This is another very good question. Well, in principle, yes, they are effective, and we are talking about air purifiers which work based on filtering the air. So they filter the air, and in the process, they remove particles of any origin, whether it's viruses, bacteria from human respiratory activities or bushfire smoke. The issue, however, is that to use them, still some knowledge is necessary. First of all, is it needed at this place here right now? If there's no threat, if ventilation is already good, let's say we have whatever, windows open, it's not needed right now. If it's on also, if it's too loud, it's not going to be used in the classrooms. So the question is when it should ideally turn itself on and at what level to be useful. So this was, I remember there was at the beginning of the pandemic, some authorities were buying en masse purifiers and delivering them to schools. I said then they will become electronic junk because this school teachers, principals, they didn't know what to do with them, and that actually came to be. So yes, they are effective, but still some knowledge how to use it, and ideally autonomous operating of the systems is necessary.
I might jump in with a question there. You mentioned, I wasn't aware of the issue with laser printers, but there's a proliferation now of 3D printers, both domestic to industrial. Are there studies on that? Do you want to comment on the particular issues with 3D printers?
We haven't looked into this. The other issue we discovered by a chance, so it became a project on its own. Well, they are very likely emitters as well. So that would be an interesting little study to find out and do the same what we did with the previous issue.
Very good talk, thank you very much. Could I follow up that last one, the unexpected printer pollution? With cars, we all know just how bad diesels are, and petrol cars are bad too. But is there any pollution from electric cars? I ask as an electric car driver.
This is a very interesting question where there are no emissions from the electric engines, but there are increased concentration of larger particles resulting with interaction between heavier car tires and the road. There's not really a huge body of information about this, but because the cars are heavier, so because of this there's a larger, not resuspension, but emissions of these particles. To what extent this is a problem, it's very difficult to assess right now. Are the cars going to be heavier? Because the weight of the cars is the size of the car. So I've seen some trend in Europe, for example, that electric cars are smaller than the petrol cars. I don't see this trend here, but hopefully with time we will realize that we don't need to drive so massive cars when driving 10 kilometers from home to work, and therefore the cars can be smaller, can be lighter, so that this problem will disappear. But if anything, this is the problem with electric cars.
Okay, I think we should probably wrap it up there, and let's thank Lidia again and congratulate her on her award. Congratulations. Thank you, Lidia. And I'd like to now call Professor Helene Marsh to the stage.
Thanks, Malcolm. I'm delighted to be here today to introduce the Ruby Payne-Scott Lecture for 2023. The Ruby Payne-Scott Medal and Lecture is a premier career award that recognises women researchers of the highest standing in the physical or biological sciences. It's one of the most prestigious career awards of the Academy and honours Ruby Payne-Scott's pioneering contribution to radio physics and radio astronomy. The 2023 winner of the Ruby Payne-Scott Medal and Lecture is Professor Jenny Graves. So many of you will have seen Jenny get her medal last night. Jenny is an international leader in comparative genomics of vertebrates, arguing that Australian animals are particularly valuable as independent experiments in evolution. She exploits the biology of Australian marsupials, monotremes, and reptiles to dissect conserved genetic structures and processes, pioneering a comparative approach that has led to many fundamental discoveries. Jenny produced unique data that successfully challenged accepted ideas, leading to new hypotheses about the origin and evolution of human sex chromosomes and sex-determining genes. She showed that human sex gene and sex chromosomes evolved quite recently, and that the Y chromosome is degrading rapidly and will disappear in a few million years. Sorry, guys. Jenny made fundamental discoveries about how the X chromosome is genetically silenced in female mammals, and shows that genes on the inactive X are not copied into RNA, and that DNA methylation suppression suppresses transcription. She initiated and guided collaborative research on the epigenetic control of environmental sex determination in Australian reptiles. Jenny got her medal last night, as we know, at the dinner, and now it's my great pleasure to welcome her to deliver the 2023 Ruby Payne-Scott Lecture.
Well, thank you, Helene. Well, sex is a splendid thing, and I've spent a good deal of my working life looking at the genes and the chromosomes that make animals either male or female. The first 30 years of my career, I worked on mammals, and I looked at the gene on the Y chromosome that makes you male and at the peculiarities of the X chromosome. And I was rewarded for this work by receiving the Macfarlane Burnet Medal in 2006. But after all that time, it seemed like time for a change, and I was very fortunate to strike up a collaboration with Arthur George's group at the University of Canberra. And this happened because I looked at their a draft of a grant application to look at sex in a lizard, and I thought, what this grant needs is me, because I had a lot of experience with molecular genetics and cytogenetics, and I'd looked at genes and chromosomes down the microscope and at the molecular level. And so that's began a wonderful 20-year collaboration looking at sex in the dragon lizard. So we had a very successful grant called Sex in Dragons, and Arthur and I both used that in our titles, as you've seen. So this has really been my second career, and I hope I can convince you that it's another independent career, and I am very, very grateful for the award of the Ruby Payne-Scott Award for this work. Well, I need to explain to you about vertebrate sex determination. Here's an embryo, and it looks much the same with a human embryo or a chicken embryo or a lizard embryo. And it's these little green dots that are going to form a ridge of cells that becomes either a testis in the embryo, and this testis will make male hormones, and it's the hormones that make the baby a boy. The alternative pathway is that that same little ridge of cells becomes an ovary, and that embryo will become a female. So there's got to be a trigger that tells you which pathway these same cells are going to take. So this is quite a unique thing in biology, that a single lot of cells can go down two quite different pathways and turn into different organs. So the trigger can be either genetic, and it can either be a male determining factor or it could be a female determining factor. And in mammals, we know what that factor is. It's the SRY gene on the Y chromosome that makes the embryo a boy. Birds do it in a different way. They have different sex chromosomes and a different sex-determining gene, and there are all kinds of different sex-determining genes that we now know about. There's also some animals which determine sex in a different way. Fruit flies, for instance, have a gene on the X chromosome that can count. You know, you have 2 X chromosomes and you're a female fly, and you have 1 and you're a male fly. Silk moths have a gene on a female-specific W chromosome. So there's these triggers that we know about, and we know most about sex in these species. But the alternative is that sex can be determined by an environmental trigger. And particularly temperature is a trigger that is very often used. So as I said, we know most about sex in mammals and in humans and mouse. And we know that there's a gene, the SRY gene, which is the trigger. But it's part of a big web of other genes and other products, and it's a real push-me-pull-you, because SRY kicks into action another gene, and that kicks into action other genes, and it also suppresses female-determining genes. So you can see that it's a network full of checks and balances, and we know quite a lot about this pathway now, and we know that it is pretty much conserved in all animals, regardless of what the trigger is. Well, sex in mammals is terribly interesting, and still lots and lots of work being done on it. But it's essentially the same in all mammals. The sex chromosomes are very conserved. The SRY genes seem to have the same action in elephants and humans and everything else, and the pathway is pretty much conserved. And I'd have to say that this could be a bit boring. So I was delighted when I had the opportunity to go further and further away from humans instead of just comparing humans and kangaroos and platypuses. I would now be able to compare humans with reptiles. Now, reptile chromosomes are actually highly conserved, but the sex trigger is not very conserved. And there's also both genetic sex determination and temperature sex determination, and some species actually have both. So environmental sex determination, alligators are the pin-ups here. You've probably heard that when it's hot, the eggs all hatch as males, and when it's cold, they all hatch as females. Same eggs, but a different trigger pointing them in a different pathway. Turtles are kind of the opposite. When it's cold, all the eggs hatch as males, and when it's hot, they hatch as females. So these species have no sex chromosomes. It's not that the whole chromosome is missing, it's still there, but it does not determine sex. So you think after 30 years looking at genes and chromosomes, I wouldn't really like to work on turtles or alligators because they don't have any pretty sex chromosomes to look at. But I'm also very interested in epigenetic control because of my work on the X chromosome. Epigenetic control that means above the gene, there's something which is turning the genes on or off. Well, I said that the reptile genomes are conserved, and here's work that we published a few years ago looking at all the sequences of a number of reptiles, birds, turtles, and mammals. What you can see here is the size of the genome is on the x-axis, and you can see that the number of chromosomes starting at the bottom with the big chromosomes, and at the top, very tiny, tiny little microchromosomes. And you can see right across snakes and lizards and birds and turtles, there's tremendous conservation. So these chromosomes are virtually the same right across. They go kind of crazy in mammals, and it's very interesting to wonder what it is about being a mammal that your genome is completely scrambled up. However, the genome may be conserved, but the sex chromosomes are not. There's all kinds of different sex chromosomes in a whole bunch of lizards in particular. And what this says is that there's very rapid turnover of sex genes and chromosomes. And in just this one little phylogeny, over a couple of million years, we can see that the central bearded dragon and its closest relatives have genetic sex determination, but its more distant relatives have temperature sex determination, and its more distant relatives have genetic sex determination, but a different gene and a different chromosome. So you can see things happen very fast in reptiles, and this is why I say mammals are a bit boring compared with reptiles. So I was very lucky to become involved in the Sex in Dragons project, and I was particularly fascinated by the model species, the central bearded dragon, which has genetic sex determination. So over a whole range of temperatures, you get half boys, half girls, but it's got relatives, close relatives that have temperature sex determination. So we thought, well, the first thing to do is to find the sex chromosomes. Well, that's easier said than done in reptiles because there's 6 or 7 perfectly good big chromosomes that you can look at down a microscope. And then there's these tiny little things that look like specks of dust. And if you line up males and females, you simply can't tell the difference. But I was very fortunate to attract a very talented cytologist, Tariq Azaz, and he was able to use all kinds of fancy new molecular techniques to pull out sequences which were present in males but not females, females but not males, and he was able to show that there was a female-specific chromosome, which is a W chromosome, and we were able to then look at sequence differences between the sexes, and that was really key. So, of course, we wanted to know what was the dragon sex-determining gene? And we did have sequences that were female-specific, so we thought this couldn't be very difficult. We'll just look for differences at the sequence level between male sequence and female sequences. And there were quite a lot of them, but they all turned out not to be sex-specific. So it turns out that there's a lot of deletions in the population, and we were just picking these up. So that was a lot of work, but it didn't really tell us what we wanted. So instead, what we did was actually we pulled out the Z and the W chromosome by micromanipulation and looked at the sequences there. And we were one of the first adopters of sequencing protocols, and we were able to get a pretty decent sequence of the dragon lizard through BGI. What we could do then was pull out the sequences physically, look at them and figure out what genes are on them, and then we could go to the chicken genome and find out, well, what else is on that chromosome? So what we could do was line up the chicken genome on the left with the dragon genome on the right, and you can see there's a lot of commonality there, and we could predict what genes we would expect to find on sex chromosomes in the dragon lizard. And there were two that looked really interesting. One was this gene RSPO1, which we know is in the sex-determining pathway and it's a female-determining gene. And the other was NR5A1, which is you need to be becoming a male. So we thought, okay, we've got two candidate genes, so this needle in the haystack is pretty much solved. And we were excited because both these genes were in the conserved sex-determining pathway. So what we had to do was make sure that those genes were on the sex chromosomes, and RSP1, when we looked at it, and these red dots show you where the gene is, and the green dots show you which the sex chromosomes are, and bad luck. That's a wrong gene because it's not on the right sex chromosomes. There we go. But when we did the same thing with the NR5A1 genes, you can see that the green and the red dots are on the same chromosome, so we thought that's our candidate gene. The other good thing was that it seems to be expressed in the testis, but not in the ovary, which is what we'd expect. So that was our candidate gene. Okay, job done. We know that this gene codes for a very important steroidogenic factor that's very important in making males in mammals as well, so that was good. It was expressed in the right place. But the problem was it was actually on both sex chromosomes, on the Z chromosome and the W, so what was the difference going to be? And the sequence was identical for the copies of the gene on the W and the Z were identical. So we thought, that's a bit weird. That doesn't look good. And they're both expressed in testis. So we thought, well, we've got the wrong gene again, or maybe it works in some weird way. And that seems to be the case. This work was done looking at the RNA that was copied from this gene. And if you look at the RNA copied from the gene in ZZ males, it makes perfectly sensible. Oh, for some reason, we're going backwards. There. It makes perfectly sensible protein. It's got two bits. One bit binds to DNA and one bit binds to the ligand. And both these RNA transcripts made sensible protein. But if we looked at the females, ZW females, with the same gene, but it was copied into 18 different transcripts, and they were quite crazy. So there are all these splice points there that were joined together in different ways, and it couldn't make any sensible protein at all. It made protein that didn't have a DNA binding domain, or it didn't have a ligand binding domain, or it just went kind of crazy downstream. And so we thought, well, maybe that product actually inhibits the action of the gene in females. And so this is what we think is actually happening, is we're actually getting in a female, we're getting sensible products from the Z chromosome and crazy products from the W chromosome that actually inhibit it. And so these transcripts are all a bit mad, and the products, some of them are sensible products, but others are not, and they actually inhibit, they bind to DNA and stop the proper copying of the RNA. So here we have quite a new way of doing sex, not by differing the genes, but by differing the way the gene is expressed. So it's a genetic mechanism, but it's actually an epigenetic function. So what this means is the conformation of the W chromosome, which we know to be different, you can actually see it's different down the microscope because it's full of repetitive sequences, this somehow messes up the way that the RNA is copied. So that was pretty interesting. We thought, okay, it looks like we've got the right gene now, and the dragons have genetic sex determination. We think we know what gene is doing it, and we think it's genetic, that is, at every temperature you're going to get half males, half females. That turns out to be wrong. If you push the temperature up even further, they're all girls. So half the girls are ZW, which is normal. The other half have ZZ, which is a male genotype, but they develop as females. So they're sex-reversed females. The great thing, and you can see by the pink flower on this gene, that it is a sex-reversed female. The really great thing was that these ZZ females are fertile. In fact, they lay more eggs and they have a better hatch rate than their normal sisters. So what we could do then was to mate these sex-reverse ZZs to the normal ZZ males, and all the progeny, of course, have only Z chromosomes. We've got rid of the W chromosome entirely. We've completely changed the sex-determining mechanism. So now, instead of a genetic sex-determining mechanism, we now have a temperature sex-determining mechanism. And the scary thing is it's happening in the wild. Over 10 years of sampling, the frequency of sex-reversed females has gone up to about 5 or 6%. So this is one of my favourite pictures of then postdoc Claire Holloway with her Nature cover story and her newborn son. So the lesson is this: yes, you can have it all. So what this told us is that sex-determining systems can change very, very rapidly in reptiles, from genetic to temperature, and then back again. And that's why you get such a lot of variation in sex in reptiles. Well, of course, we'd like to know, how does high temperature determine sex? How does it reverse sex when you've got these sex chromosomes and sex-determining genes here? And this is really what people are working on now. One thing we discovered a few years ago, and I think Arthur Georges showed this the other day, is that you get different transcripts made at higher temperature that retain an intron. And we think probably what this does is actually wipe out the function of that gene. it looks like the genes that are affected and retain introns are part of a very important repressor complex, and for some reason I'm having trouble. It's advancing when it should be going back and that so forth. There we go. So what's this polychrome repressor complex 2 seems to do is to push the whole pathway towards the male, and we think probably what's happening with the heat is it's affecting it's changing the epigenetic marks, so instead of male you now get female. So we don't know what the point at which temperature sensitivity occurs, but it could be any of these points. So I'm ecstatic, of course, because a lot of these epigenetic markers are the same ones that I studied on the X chromosome 30 years ago. So this is ongoing work. It's getting very exciting. We'd like to know how heat interacts with the genetic pathway. So our conclusions are that we know that dragons have a ZZ male ZW system, and we think that the gene on the Z and the W is the male factor SF1, but it works in this really weird epigenetic way. And at high temperature, the ZZs develop as females, and this means that you can very rapidly turn over the sex-determining system, and that intron retention is a big part of this, and so is probably a calcium-regulated way. So I hope you agree that this is indeed a second career, and I think it's been a particularly interesting direction for me to take it. I'm extremely grateful to Arthur and his group. Here's a picture with our famous paper here showing Arthur, and of course he's handling a lizard, as he often is, and he has a wonderful group with postdocs and whom I've interacted with many times. So in case you want to ask me what my third career is going to be, I have to show you that very recently I have written the libretto for an oratorio, which is the creation story from science, which has been performed and which I'm spending a great deal of time getting people to listen to and think about. Thank you.
Thank you, Jenny. That was a wonderful lecture. I definitely think that you've had 2 careers and therefore deserve 2 career medals from the Academy. I'm not quite sure how our medals are going to cope with a 3rd career, but I'm sure the awards people and the council will be working on that. So we now have the opportunity for a short Q&A session, and if you have a question for Jenny, please raise your hands, and we've got a lot, so I'll let the runners decide who goes first.
Jenny,
Maybe
thanks
you,
very
Bob?
much. Wonderful lecture. In cardiomyocytes, soon after birth, the metabolism changes from glycolysis to fatty acid oxidation, and with that you have a decrease in alpha-ketoglutarate, which directly affects Jumanji-3, and therefore you switch off the ability to proliferate. And I'm wondering whether metabolism is being changed in these organisms, and that's controlling Jumanji-3, which is a histone demethylase.
I don't know about metabolism, and there's all sorts of differential growth that occurs in testis differentiation versus ovary differentiation. Is differential growth of different parts of the genital ridge. So that's very interesting. I didn't know that. Yeah, Mike.
Yeah, thanks for the great, interesting talk. I just have a question about the fact that if every ZZ female gives birth to ZZs, and half of the progeny of a ZW is ZZ, then doesn't that mean the proportion of Ws in the population has to go down and eventually will disappear?
Absolutely. It looks like the W chromosome is losing out. It's becoming less frequently in the population, and it could completely disappear. Arthur's done a lot of modelling and says, no, it probably won't disappear, but it depends on the ambient temperature, but you could be driving the whole system to temperature dependent.
Thank you very much for a fantastic talk, and congratulations too. I just wanted to know, why has actually this evolved at all?
Very good question. I call it dumb design, which is, of course, different from intelligent design. Sex is, everything's all wrong. It breaks all the rules of evolution. Evolution should be giving us fitter Y chromosome. So why is a Y chromosome disappearing? Well, it's a part of the job of the Y chromosome because it's always in a testis, and so it's always being damaged, and it can't repair itself. And so you can see that evolution sort of gets parts of the genome in a bit of a bind, and this happens in fruit flies as well. So there are general rules that drive down parts of the genome. So it doesn't seem like a sensible, if you or I were designing a sex system, we'd do a much better job. But it's intrinsic because some things are expressed in males and some things are expressed in females. So it's an intrinsic part of the system that there will be these drives to eliminate genes.
Francis?
If it's me, Jenny, a lot of evidence points the finger at perhaps chromatin modification enzymes as being the temperature-dependent step, enzymes that cease working at a certain temperature or work better at other temperatures. Would you agree with that? And are there any candidates? And if so, can that be tested biochemically in terms of their temperature sensitivity?
Absolutely. So we know that a lot of the epigenetic regulators regulate chemical modifications to chromosomal proteins called histones. And there's a whole histone code that we're still finding out about. And if one of these enzymes that puts the modifications on or takes them off were temperature sensitive, we could explain it. There's almost an embarrassment of new data now so it looks like at least 2 of these processes are definitely temperature dependent. The difficulty is that we don't know, is it a direct effect of that enzyme or is it something further back in the pathway? But this is going to be the next very exciting phase of figuring out exactly what these pathways were. But I think we're dreaming if we think it's going to be simple.
Can I ask one? Yeah. Thank you so much for the very intriguing talk. So, can you see me? Okay, yeah, I'm here upstairs. Yeah. So I'm very intrigued by the fact that temperature can determine sex. So I was wondering, so I think I have missed this point. So you mentioned like this cold and hot determining male or female. Is it always the case that cold always gives male versus hot temperature giving female?
A very good question. No, it's quite inconsistent. Alligators, the hot gives you males, and marine turtles, hot gives you females. So with global warming, we can look forward to a world full of male alligators and female turtles, and that's not going to work very well.
And when you say hot and cold, what's the kind of quantity, how much the temperature dependence, temperature difference that kind of leads to this reversal in the process?
I'm having a bit of difficulty hearing you, but I think what you're saying is, how much heat. There's a huge literature, a very old literature, where people have exposed eggs to temperatures over a long time or a short time, a variable temperature, as Arthur talked about yesterday. And this is very crucial. It looks like there's some sort of accumulation of heat induced product. but we don't know what that product is.
Thank you so much.
So I think it's just one last question. Thank you,
Just
Francis.
quickly, Jenny, that was lovely. I'd like to know if the ZZ females have any other characteristics other than a pink flower and more eggs. For example, are they bigger or are there any other obvious differences?
The ZZ females seem to be bigger and fiercer. So, as Arthur said yesterday, they make better mums than their normal sisters do. And I think that that's an interesting point because I think we know in humans that 2 X's are better than 1. You know, boys suffer for all sorts of sex-linked diseases that girls don't because we have a backup X chromosome. And the same would be true of ZZ. If 2 Z chromosomes is better than 1, you're always at risk if you've only got 1 copy of the gene because if it's mutated, bad luck, there's no backup. So that is probably why the ZZ females are very robust and lay better eggs.
So thank you very much, Jenny. That was a fantastic lecture, and thanks everyone for the great questions. So can we give Jenny another round of applause? So that now concludes the morning session. We're going to have a morning tea. As we leave, we ask all the 2023 awardees to move immediately, it's underlined, to the main foyer for a group photo, and this includes all awardees who received an award in 2023, and a reminder that all awardees are asked to make their way to the Becker Room for their individual portraits sometime today. And so please join us for morning tea in the Jaeger Room, the Dorothy Hill Room, or outside under the marquee, and we'll reconvene at 11:15 for our next award presentations. Thank you very much.
Kathleen Folbig has always insisted she never killed her 4 young children.
Did you kill Caleb?
No!
Did you kill...
No!
Did you kill Sarah?
No.
Did you kill Laura?
That's a position she maintained while exhausting all of her legal appeals and during 2 subsequent judicial inquiries. 20 years into a 25-year prison sentence, the justice system has now accepted there is reasonable doubt about her guilt.
I think the nation will look at the case of Kathleen Folbig as a major miscarriage of justice.
Ms Folbig was convicted of smothering her babies Caleb, Patrick, Sarah and Laura. The 4 died on separate occasions between 1989 and 1999. But at the time of her conviction in 2003, there was no way of knowing if gene mutations played a role in any of the deaths.
In fact, it was only in 2003 that the first draft of the human genome sequence was completed. Now, since then, it has become possible to sequence whole human genomes and exomes in a much faster and affordable way.
That's Professor Carola Vinuesa, who analysed sequences from DNA extracted from old heel prick samples of 2 of the Folbig children.
She was concerned that the analysis of that genetic information had not been fully heard during the first inquiry into the convictions of Kathleen Folbig. And it was those concerns and her resolute understanding of her science and her scientific integrity that in some ways didn't let her sleep at night.
Professor Vinuesa took those concerns to the Australian Academy of Science, where she is a fellow.
The more we looked at it, the more we too became concerned around the way science had not been heard in that first inquiry.
The science was complex. Research undertaken by Professor Vinuesa and complemented by independent experts around the world showed that Ms. Folbig and her 2 daughters possessed a rare gene mutation known as CALM2G114R,
which encodes for calmodulin. Calmodulin mutations typically are associated with cardiac arrhythmias that can cause sudden unexpected death in children and adults, both while awake and while asleep.
The second inquiry, which was instigated at the urging of prominent scientists, including 3 Nobel laureates, also heard medical evidence about epilepsy and respiratory problems, which could help explain the deaths of Ms. Folbig's 2 sons, Patrick and Caleb.
There is a reasonable doubt as to Ms. Folbig's guilt.
Retired Chief Justice Tom Bathurst KC accepted that the weight of the evidence cast reasonable doubt about Ms. Folbig's guilt, recommending her pardon. A key difference in the way the second inquiry considered medical evidence was the role played by the Australian Academy of Science.
I think now that with this new evidence, it's beyond reasonable doubt.
This is a clear indication that the independent scientific advice has an important role to play in the justice system.
The Australian Academy of Science was granted leave by the inquiry to act as an independent scientific advisor to it. I think unique to this case, and the role the Academy's been able to facilitate, is that the inquiry's been able to hear from a range of experts from the right subdisciplines to give a complete picture of the sort of science that is complex, and some of it's still emerging, that's been able to shed light on the possible causes of death of the 4 Folbig children.
The Academy believes the case serves as a dramatic example of the need for a more formal mechanism to re-examine cases after appeals have been exhausted when new science is revealed.
Science is always emerging. Nothing is going to stop the advancement of knowledge, nor would we want to do that. It would be an enormous missed opportunity if we couldn't bring about some of the law reform that's needed so that science can more effectively inform the justice system.
People think that reconciliation is about Aboriginal and Torres Strait Islander people reconciling with the rest of the population, but it's actually not. Reconciliation is about how non-Indigenous Australians recognise and celebrate that Aboriginal and Torres Strait Islander people were here 65,000 years ago. We've had a continual relationship with the country. We are the longest continuing cultures in the world. How do we reconcile in the last 230 years? We've had a dramatic impact since colonisation on Aboriginal and Torres Strait Islander people. There are people that say, why should Aboriginal and Torres Strait Islander people have a say in these matters? And I just point out that whenever we have a natural disaster, be it a flood or a fire, the government might think that they can go in and make changes and redevelop those communities, but they cannot do it without the engagement of those communities. So, if we can see that as being good practice for the rest of the population, we have to see it as good practice for Aboriginal and Torres Strait Islander people. Why should we differentiate?
These 2 scientists have something they need you to hear about the world's oceans.
My name's Matthew England. I'm a professor of ocean and climate dynamics at the University of New South Wales. I study how the oceans respond to climate variations and climate change.
My name is Adele Morrison. I study oceanography at the Australian National University in Canberra.
At the centre of their work is how water moves around the planet. Surface currents are powered by wind patterns, but deep underwater currents are driven by different layers of density in a process known as overturning circulation.
The ocean plays a big role in global climate by moving heat and carbon and also nutrients around, which affects ecosystems in the ocean. Changes that happen in one small location, like say near Antarctica, can then have a global influence because those waters move all throughout the whole planet.
And it's the Antarctic overturning circulation that has these scientists worried. They're two of the authors of a landmark study published in Nature that makes a dire projection closely related to the melting of the ice caps.
And what we see in that simulation is that the meltwater rate that we expect to play out over the next few decades is enough to slow down that overturning at a very dramatic rate, about 40% reduction by 2050.
Meltwater is a clear consequence of climate change. As ice caps melt, huge amounts of fresh water enter the oceans. The resulting reduction in salinity makes the water much less dense, meaning it doesn't sink with the same force.
So the dense water is important because it's a major driving factor in the global overturning circulation, which is this conveyor belt of waters that move around the whole ocean. And so this formation of dense water pushes that water down into the abyss, and then throughout the rest of the global ocean that moves up again in different places around the globe.
A slowdown in the overturning, particularly by 40%, would have a profound impact on marine ecosystems.
The thing about the oceans is that all of the marine life that we have at the surface, when it dies off, it sinks to the bottom of the ocean. So there's a lot of nutrient-rich water in the ocean abyss. If we slow down the overturning circulation that brings that very bottom water back up to the surface, we cut off a way that nutrients get back to the surface to regenerate marine life.
Their projection is based on advanced modelling that doesn't just factor in meltwater, but captures key regions of Antarctica with unprecedented skill.
And this is particularly important around Antarctica because there's a lot of small-scale processes that are important for this dense water formation.
A slowdown of the North Atlantic overturning circulation has already been documented. But this latest study shows that overturning in the Antarctic region could decelerate at twice the rate.
I should also say there's actually evidence in the ocean abyss that the mechanism we've described is already playing out. There's been significant warming of the ocean abyss, and that's exactly what our model projections show, that as the ice melt slows the overturning circulation down, you do get a warming of the ocean abyss. And if anything, our models are a bit behind where the observations are already tracking. So all the evidence we have suggests that we will have a slowdown of this overturning circulation in the next couple of decades.
There's so many people working in science doing great work.
We use research innovations to solve the world's biggest problems.
The link between science and a happy and healthy society is as important as it's ever been.
That's why I do what I do, because I want to see my science make a difference.
And I think that this is a great opportunity for recognition of that work.
To now be awarded for my contribution, for me, that's just truly incredible.
I highly encourage anyone to apply for these honorific awards. It's a good way to get our science out there.
Receiving this medal is a real honour.
It means a tremendous amount to be acknowledged by the Australian scientific community, because it means other people have seen there's a reason I do what I do, and they want me to keep doing it.
I would absolutely encourage anybody who is passionate about their science to apply for one of these Australian Academy of Science honorifics.
Professor Louis Morawska has some planets on the boil.
It's a big block coming down. Watch.
He often encourages his students to make lava lamps like these ones as a way of demonstrating Earth's evolution.
The density of the hot versus cold liquids, they play off against each other in these really complicated ways. So the blobs would be maybe continental crust, and the background liquid, we think of that as maybe being a little bit like the mantle of the Earth.
Professor Morawska is an expert in planetary scale geodynamics.
My interest is in how the Earth formed and how it became how it is today, with plates moving, turning itself inside out, creating mountains, creating the oceans.
Earth is covered in tectonic plates. Interactions at their boundaries can lead to major geological events such as earthquakes and volcanic eruptions. Professor Morawska builds computer modelling tools to better understand the formation of those plates.
Now, the reason we're interested in that is because the process of plate tectonics stirs the whole Earth in 3 dimensions. So the plates going round into the interior and coming back out again, that recycles a lot of interesting elements, and those might well be elements that are important for sustaining life in the long term.
He also wants to know why Earth appears to be the only planet in our solar system that has tectonic plates.
And the really interesting question, I suppose, is if you look at exoplanets, planets around other stars which might be a bit larger or a bit smaller, or maybe have a different history, maybe they have a different heat content, maybe they're made of something very slightly different. Would those planets potentially have plate tectonics? So the study of plate tectonics has very big implications for understanding whether distant exoplanets may be suitable for life.
One of Professor Morawska's software creations is called Underworld, which can be used in both laptops and supercomputers to study geological problems. He insisted on the source code being available to researchers everywhere.
It's very important to me because I really do actually believe in open science from that perspective, that if you want to do something, you want other people to be able to reproduce your work really quickly. You want other people to build on your ideas. So if they have access to the best tools, and I have a bit more time to think about how to improve those tools, then it's a win for everybody.
Born in London, Professor Morawska moved to Australia more than 25 years ago. Today, he's based at the Australian National University, which is also home to one of Australia's most powerful supercomputers.
It is sort of the national university, which makes it a little bit special. And, although we can access computers from anywhere, we can also access the people, we can talk to people. There's natural collaboration that develops with other people who you run into.
He sees becoming a Fellow of the Australian Academy of Science as valued recognition of the earth sciences.
It also gives a sort of an opportunity to let people know about the importance of the Earth as a planet, the planetary evolution, to be able to explain a little bit how the Earth evolves and why it's important to understand that.
Professor Mark Krumholtz uses computers to help solve the mysteries of our universe.
My particular focus is on how stars form, where do they come from, when they die, where does their material go, how are galaxies assembled.
He models the gas between stars that form part of the interstellar medium. These interstellar gases collapse together under the pull of gravity, forming new stars. But when those stars inevitably die and explode as supernovae, they drive the gases back out into the galaxy.
So you have this constant tug of war between gravity trying to pull everything in and the newborn stars that try and push everything back out.
These processes take place over millions of years, making them hard to observe and measure. That's where Professor Krumholtz's work as a computational astrophysicist comes in.
We can't watch this happen in real time, but we think we understand the basic physical principles that govern, for example, the flows of gas, the way stars work. And you can build those physical principles into a computer simulation.
Simulations that could help inform new observations or paint a picture of what galaxies millions of years into the future might look like. It's research that requires immense computational power, more than the level offered by current infrastructure here in Australia. In 2022, Professor Krumholtz was awarded an Australian Laureate Fellowship to develop new software to drive the nation's supercomputers to peak performance. But he says Australia still lags behind in terms of hardware.
If you want to be at the cutting edge, you actually have to replace the computer every 5 years or so. So it looks like that.
As a kid, Professor Krumholtz wasn't particularly interested in astronomy, but loved maths and physics.
I grew up with a view of the New York City skyline, so there were no stars. It was that I sort of at an early age, found it fascinating that you could use math to represent reality. It seemed like magic.
In 2016, he moved across the globe to his current role as professor at the ANU Research School of Astronomy and Astrophysics. He's proud to now also be a Fellow of the Australian Academy of Science.
It feels like it's a sign of acceptance that, you know, I'm not viewed anymore as an outsider, that I'm actually now a member of this community, and that's nice.
If you think soil is boring, you haven't met Professor Buddhaman Manasni.
I find soil amazing. It's an amazing topic to study. Hello. This one is from Narrabri, so this is from our university farm.
Soil plays a vital role in sustaining agriculture and ecosystems. Around 97% of our food comes from soil. It also purifies our water, helps prevent flooding, stores nutrients, and contains extraordinary levels of biodiversity.
So that's why I think that we should take care of our soil to ensure that we have a sustainable future.
Professor Manasni is recognised as a global leader in digital soil mapping. He's developed detailed maps of soil properties using underlying soil processes and machine learning supported by satellite and field data.
So all of these sensors are just like taking a fingerprint of the soil. From the fingerprint, then we can try to identify what are the constituents of the soil.
His methods are used by soil scientists around the world, from the paddock level to the global scale. The ability to rapidly and precisely predict and map soil carbon levels is shaping up to be a vital tool in the fight against climate change.
Soil is a carbon sink. Soil is the largest carbon storage on the land.
He wants to safeguard and enhance this carbon storage potential by improving soil quality. Here in Australia, his maps are helping to assess and improve land management practices.
With the help of remote sensing images and statistical tools, we are able to extrapolate this data, letting us know how much, for example, carbon is stored in every 30 metre in Australia.
Most Australian farmers now practice a technique called minimum tillage, where they don't remove the residues of previous harvests, such as stubble and roots, before planting new crops. That prevents a substantial amount of carbon from being released into the atmosphere, while also making the soil itself more robust.
So now we need the next step to increase further carbon in the soil. So there are trials now being conducted by our researchers trying to put cover crops in between the season and different types of varieties of cover crops to increase carbon back in the soil.
Professor Manasni grew up in Indonesia before moving to Australia to undertake his Master of Agriculture at the University of Sydney. 30 years later, he's still at the university and can now also proudly call himself a Fellow of the Australian Academy of Science.
It is an honour, and also it is a recognition towards the discipline that we know Soil Sciences Australia is changing the world.
Professor Emma Johnston investigates how humans interact with marine ecosystems.
My work has primarily been in coastal marine ecosystems, which is where humans have the most impact because we tend to love living near the coast and we use coastal ecosystems for all of our transport and our recreation, and we get a lot of food from them.
Global shipping means that none of those ecosystems work in isolation.
We actually have this mechanism for transporting species from one country to the other on ships, and that could be either in the ballast water or connected to the hull of a ship.
Some of Professor Johnston's research included laboratory and field experiments, which identified new drivers of invasive species success and clearly demonstrated their ability to rapidly evolve in new environments.
In many of the ports and harbours that I work in, there are more than 300 different species that you can see of marine organisms that are introduced. They're non-native.
Understanding this paves the way for better management strategies, such as dredging up contaminated sediments to help improve outcomes for native species. Professor Johnston believes in translating research into action, particularly evidence-based public policy. She was chief author of the Australian State of the Environment report in 2021.
We are at the forefront of climate impacts, and without rapid, drastic, urgent, unimaginably fast carbon emissions reductions, we stand to lose a lot of those precious ecosystems.
Professor Johnston is Deputy Vice-Chancellor Research at the University of Sydney. She's become a highly influential voice for science, having served as President of Science and Technology Australia and Dean of Science at UNSW. She's determined to make the sector more open and inclusive, but says that requires a whole-of-society approach.
It's the schoolyard where they're encouraged to do particular activities. It's their role models that they see as they go through high school and the choices in subject that they're encouraged to make. And then in their actual undergraduate and their career, it's about building in the flexibility so that people can have a diverse array of different cultural backgrounds, but also lifestyle choices, and come in and out of scientific careers.
Research funding is another key priority for Professor Johnston. She says government and industry investment is proportionately much lower in Australia than in many advanced nations.
We have incredible minds and incredible fundamental research being done, but sometimes we cannot follow through because the investment in the translational work, in the innovation, in the bringing the product to market, the money is just not there.
Professor Johnston sees becoming a Fellow of the Australian Academy of Science as not just an honour, but an opportunity. She wants to work with other Fellows in promoting the value of science and guaranteeing a vibrant sector.
Science is a method that has enormous potential for benefits for the whole of society.
Helping the environment was something Professor Kylie Catchpole always wanted to do.
I really didn't see myself as an environmental activist, though, and I didn't really know what to do with it. So I studied physics. I got to the end of my degree and I thought, well, what shall I do now? It wasn't really clear.
A question that ultimately set her down the path of solar energy research. Today, she's a world expert on all things solar and jointly leads a group here at the Australian National University on advancing perovskite solar cells. While silicon has long been the dominant material used in solar cells, perovskite has emerged more recently as a low-cost alternative with potential to be as efficient as silicon. A potential proven with world records set by Professor Catchpole's team. In 2022, they took perovskite solar cell efficiencies to 22.6%, breaking their previous record of 21.6%.
Perovskite cells now have efficiencies that are similar to silicon solar cells, so just a single perovskite cell, they have efficiencies, the top efficiencies around 25%, whereas the top silicon cells are around 26%.
Professor Catchpole has also led the development of more efficient tandem solar cells. These systems involve stacking a perovskite cell on top of a silicon one, an advancement that could push the solar efficiency ceiling even higher.
The perovskite cell will absorb the blue light really well and give you a higher voltage, and then the red light will actually pass through the perovskite solar cell and be absorbed by the silicon solar cell, and then that will give you additional voltage. So together, that can give you a higher efficiency.
In theory, the efficiency limit of a two-cell tandem system sits as high as 44%. But more efficient and affordable solar power isn't just the end game for Professor Catchpole. She also sees it as a means to help produce another type of renewable energy, green hydrogen.
We'll need hydrogen to produce various materials to refine metals, for example. And if you can produce those using solar energy, then you can reduce the cost of hydrogen.
In another project, her team connected their solar cells with a catalyst to directly generate hydrogen at high efficiency. Efforts that Professor Catchpole hopes will accelerate our path to decarbonisation both globally and here in Australia.
There's an extremely bright future for renewable energy in Australia. There's just so much opportunity we're really blessed with the amount of renewable energy resources we have, and we should make use of them.
A message she now shares as a new Fellow of the Australian Academy of Science.
Deep thinking, where every move counts. That's a skill set Professor Andrew Wilkes has refined through chess tournaments like this one.
Chess taught me how to think tactically and strategically. In the sciences, those sorts of skills are incredibly important.
He was amazed in 2017 when Google DeepMind's AlphaZero AI program triumphed at chess against world-leading specialist software within hours of teaching itself the game.
If you could apply these sorts of tools to a similar process, drug discovery, it's a process that has some rules, many unknown unknowns, then you might be able to advance your programs that much quicker, that much more effectively.
And that's what brings us here to the Bio21 Institute at the University of Melbourne. Professor Wilkes co-founded a company based at the institute called Synthesis MedChem.
There are big machines, big toys, big equipment that can do amazing things here. And the sort of information that comes out of that, structural biology, analytical chemistry, those sorts of things are incredibly important for us as we develop our drug discovery programs.
Professor Wilkes is a molecular and cancer biologist. During his time at the Ludwig Institute for Cancer Research in the 1980s and 90s, he discovered and named the Jacques family of kinases, enzymes that play a critical role in how cells interact with their environment. That discovery significantly advanced our knowledge of immunity, opening new avenues for therapeutics. Professor Wilkes is also very much an entrepreneur. Before Synthesis, he founded one of Australia's most successful biotech companies, Cytopia.
Making the connection between the science and cash or investment, that's a little bit of a skill. There's still a translation gap in some way, where the potential value of a piece of science is still somewhat opaque to the people who have cash to invest.
Professor Wilkes' mission these days is to generate enough investment for early phase drug discovery before then passing the projects onto larger pharmaceutical companies to complete clinical development. That mission is enhanced by machine learning technology.
The computer will develop an algorithm by which it can improve the way the molecules behave, and you kind of go along for the ride.
It's not all left to the computer. Ultimately, each hypothesis must be subjected to biological validation.
But you can get a long way down the path much more quickly, simply using these AI programs and computational chemistry. We're all cyborgs now, in many respects.
Professor Wilkes says becoming a Fellow of the Australian Academy of Science is a massive honour, and one he hopes will encourage more investment in the complex chessboard of translational science.
The impacts of climate change are being felt around the world, from extreme weather events to biodiversity loss. We've been put on notice to take urgent action. Countries, including Australia, have committed to limiting global warming to well below 2 degrees above pre-industrial temperatures. But right now, we're on track for 2.1 to 3.5 degrees of warming by 2100. That's beyond what scientists consider manageable and could have potentially catastrophic impacts for the environment, people, and the economy.
So what we have is a world which is increasingly stressed by human activity.
The solution is to address the tens of billions of tons of greenhouse gases being pumped into the atmosphere every year. That requires not just dramatically reducing future emissions, but removing greenhouse gases already released.
This means drawing down or removing these historical emissions from the atmosphere for, if you like, geological time.
Natural systems such as our oceans and forests are highly effective carbon sinks, but they need help, especially given the further pressures from events such as bushfires and ocean warming. We don't yet have the right toolkit to effectively remove greenhouse gases at the scale required. So, the Australian Academy of Science brought together the country's leading experts to identify opportunities to expand research efforts and start the conversation in Australia. Beyond strengthening existing carbon sinks, new technologies will need to be developed.
I think the scale of the problem that we're facing means that we can't discount anything, and in fact, everything needs to be on the table.
Including things such as direct air capture, a technology that mimics trees.
We can develop these nanoporous materials that are really highly selective for capturing carbon dioxide from the atmosphere and other greenhouse gases. So in the same way that a tree uses photosynthesis, it takes in carbon dioxide and processes that carbon dioxide and spits out oxygen and energy at the other end, we've got a way to do that, if you like, using technology.
The first air capture units in Australia, designed by Southern Green Gas, are now in the prototype stage. But there are many other emerging technologies. This Hawaii-based startup, for example, uses solar power to make seawater less acidic, enabling the ocean to absorb more CO2. Scientists are now working to better understand the implications of the various emerging technologies.
There's a big gap between doing things at a lab base and doing them at a real-world scale significant enough to actually make a difference to climate change. That jump brings a whole series of uncertainties, risks and costs.
Once we have a better understanding of those consequences, the technologies could well be used alongside other strategies to help fight climate change.
I don't think there's any such thing as a silver bullet. So this is going to take a mammoth effort across the world, including a portfolio of approaches in order to get us to this point where we can indeed stabilise our climate and avoid a climate catastrophe.
Professor Rochelle Bookbinder has two jobs. The first is running her small rheumatology practice in Melbourne.
You put your chin on your chest, good, your head back.
And the second is as a distinguished researcher in the School of Public Health and Preventive Medicine at Monash University.
I get great satisfaction from doing both of them, and the patients just give me ideas for what needs to be studied.
But her career as a researcher got off to a shaky start when, as part of her PhD, she was tasked with giving lab rats arthritis.
When my supervisors were surprised that none of the rats had developed arthritis, I actually had to come clean and say I couldn't actually bring myself to inject the rats.
She went on to do a master's and later PhD in clinical epidemiology, the basic science of clinical medicine.
There's a lot of good things about modern medicine. We've had a lot of wonderful discoveries that have cured disease and increased people's longevity. But the current
Welcome back, everybody. I hope you were able to enjoy some morning tea. You would have noticed for morning tea was our 70th anniversary cake, which was cut yesterday and served today here at Science at the Shine Dome. I hope you enjoyed that. Nalini Joshi's in the audience somewhere and was feeling aggrieved that we were putting a knife through Pythagoras' theorem because it's everlasting, and I think she's right, but we did cut it up and enjoy it, so the theory lives on. Thank you and welcome back. Our awards program, as you know, recognises and elevates the important work of the science community, ranging from early and mid-career researchers, as well as those who have undertaken lifelong research. One critically important area that currently does not have an award is in information, communications and technology. This morning, President Jagadish announced at our donors' breakfast that we are delighted to have established an award, the Brian Anderson Medal and Lecture for Information, Communications and Technology, generously supported by Professor Brian Anderson and Mrs. Diane Anderson. This donation will allow us to establish recognition and support for early career researchers for this critically important discipline that is growing rapidly. Please join me in thanking Brian and Diane for their support. to continue with the award presentations this morning, I invite Professor Lynn Beasley, the Secretary for Education and Public Awareness, to the stage. Over to you, Lynn.
Thank you, Anna Maria, and good morning, everyone. I am delighted and honoured to be here standing to introduce to you two mid-career awardees from 2023 to give their award presentations. Firstly, it's the Jacques Miller Medal for Experimental Biomedicine. It was established to honour the contributions made by Professor Miller, including the discovery of the function of the thymus and the identification in mammalian species of two major subtypes of lymphocytes and their functions. The award recognises research of the highest standing in the field of experimental biomedicine and is open to researchers 8 to 15 years post-PhD in the calendar year of their nomination, except in the case of significant interruptions for their research career. I'm delighted to say the 2023 winner of the Jacques Medal is Professor D. Yu. Professor Yu is an immunologist whose research focuses on the function of T cells. He's internationally renowned as a leader in follicular helper T cells, a specialized subset of T cells that essentially control B cells to produce antibodies. His landmark discoveries reveal the key molecules and pathways for T cell function in health and in disease. Based on his fundamental research breakthrough, Professor Yu partnered with physician scientists and led clinical research on lupus, rheumatoid arthritis, allergic rhinitis, influenza, and HIV infections, which have established new and improved diagnoses and therapies for autoimmune, allergic, infectious diseases, and the improvement of the human vaccine efficacy. Wow. Please welcome Professor Yu to present his work, and as we say, congratulations on your award of the 2023 Jacques Miller Award.
It's my great pleasure to be here. First of all, I would like acknowledge Nagurana people on the land we meet today and pay my respect for their ancestors and descendants. So here at Shining Dome, we probably have the most diverse scientific audience. But I think we share something in common fundamental. We love equations. This is probably one of the most famous equations, E equals mc squared. As an immunologist, I think our immunology also have our own equation, which I called I equals m cubed. Immunology equals mechanism times monitoring times modulation. It's a great pleasure. I present my research upon receiving the 2023 Jacques Miller Medal for Experimental Biomedicine. I will use my equation and give some example to show how the evolving understanding of CD4 helper T cells can help us to assess the immune responses and use this knowledge to innovate some new strategy to control the function of the immune system. And this is also very, very fitting coincidence because Jacques Miller, his discovery in the 1960s has really opened this very important role of CD4 helper T cells in this immune regulation. So as you can see, I will continue working on this. So you can see there's a diagram on my left side. This was a very brief summary for Jacques Miller and his fellow American immunologist Max Cooper. Both of them received the 2019 Albert Leska Basic Medical Research Award for their discovery and the origin of the two important lymphocytes for the adaptive immune system, T cell and B cells. Before Jacques, people consider thymus, which was on the decoration of the Jacques medal, was a useless organ, is left from evolution. But Jacques has discovered the thymus is well T cell development. As we heard from Georgina yesterday, T cell play a critical role to fight against the cancer. Not only Jacques discovered T cell have this cellular mediated immunity, they also fundamentally helping B cell to produce antibody for humoral immunity. So I will give you some of our discovery and thought about which T cell really help a B cell and how they do so, and can we assess their function and their activation and understand immune response, eventually how we can innovate some strategy to control the T cell help and antibody response. So time fast forwarding, this is the current model after Jacques over the 50-60 years. And this is a tissue section from mouse and lymph node. We have a T cell in the middle surrounded, we call by the B cell follicle. And in an immune response, this B cell follicle develops this called germinal centers, labeled in red here in the middle of follicles. Let's zoom in to see what's happening in an immune response. We get the T cell activated, we all call priming. And this T cell mobile and can migrate into the B cell follicle where they meet with B cells and they provide the soluble factors or surface receptor, allows them to support B cell proliferate and eventually form these germinal centers. For most of us, we consider somatic mutation is a bad thing that's related to the cancer. But in the germinal center, B cells undergo rapid and frequent mutations much faster than the cancer cells. And the advantage benefit of these mutations allow the B cell particularly mutate their surface receptor. So some of the surface receptor can have much better, higher affinity to recognize the antigen. And again, we need these follicular T cells, TFH cells, to select only small percentage of these cells to eventually become plasma cell or antibody secreting cells or memory B cells to provide much higher quality of antibody response. So over the years, this model has been gradually established from lots of work. I'm sorry for this very busy slide. You can see the dendritic cell interaction with T cells allow them to generate and eventually help B cells for antibody production. I will highlight a few we think very important. Of course, the first one comes from Jacques to observe the thymus is important, allow T cell to help B cells. And really, it takes several decades to discover this CXCR5 from colleagues, Australian, immunology in American, to discover CXCR5 allowed the T cell migrate into the B cell follicle. And then 1 year later, to categorize these human CXCR5 positive T cells, helper B cell especially. And when I started my research, I was lucky to involve in the characterize the mouse, these CXCR5 TFH cells, and show through this co-stimulation TB interaction, if we have excessive of this function can drive too much autoantibody, eventually attack our own body in terms of autoimmune disease. And we later discovered the major soluble factor is called IL-21, interleukin IL-21. And eventually, I think you can see in the center of this diagram is a molecule called BCL6. It's a transcript factor. As we heard from Jose yesterday, this is how the cell programming to become their specialized function. And this is the identity of TFH cells. So how this knowledge can help us to really assess the immune response or controlled immune function is challenging here. The reason for that is the T cell B cell interaction occur in the lymph node or other lymphoid organ. There's no this phenotypically TFH cells, mature TFH cell in the blood. So using mouse model and also human samples, we were able to identify a set of markers shown here by flow cytometry to able to identify a related population in the blood and reflect the TFH cell activation. Following this, we can see in the vaccination, 1 week after vaccination, we can see a clear increase of the function, and that correlates with the protective antibody response. And also we can see in autoimmune disease, in the lupus patient, for example, too much of this TFH activation correlates with the more autoantibody production and more severe disease. So this method has been widely adopted globally, and we have used this method to looking into the TFH function in infection, allergies, and cancer. So how we can use this knowledge to potentially treat disease? I have touched on this autoimmune disease. Systemic lupus erythematosus is a systemic autoimmune disease affecting multi-organs and can be life-threatening. And by this knowledge of TFH cells, we actually able to repurpose an existing drug, interleukin-2, which was the first effective immunotherapy for human cancer back to the 1980s. But instead to use this to activate the immune system, we use 100 times lower dose to change an immune activation agent into an immune suppressive, largely immune tolerance inducing agent. So you can see, even the patient just gives 3 doses of this short cycle of L2 therapy, they can have a broad efficacy and a large response. You can see the facial remission very clear. And immunologically, they can suppress follicular helper T cells, pro-inflammatory Th17 cells, and expand Treg cells. And following this, we do the controlled randomized clinical trial to confirm efficacy and safety. We also can repurpose this drug for other autoimmune disease, such as Sjogren's syndrome. Let's change the gear a little bit. How about enhanced antibody response in vaccination? Our study, along with many other researchers, has demonstrated TFH function. If they got a defect, they also associated with much reduced inefficacy of vaccine response. By studying the biology of TFH cells, we was able to discover, rather than general center, that's the B cell side, undergo apoptosis discovered by Suzanne, Cory, and Jerry Adams sitting in the audience. The TFH cells really susceptible another type of cell that's called ferroptosis. The reason for this is these T cells have continued interaction with B cells, and this constant interaction leads to they have a reactive oxygen species eventually trigger this we call lipid peroxidation and stress. And the cells need this enzyme called glutathione peroxidase 4, GPS4, to mitigate this process, and this is selenium protein. So in the mouse model, as a proof of concept, we was able to show give the selenium supplementation can enhance TFH function and vaccine response, not only in the animal model, but is also in the human being setting. And now we further studied this, as found this pathway also critically regulated by lipid metabolism, and we can find a further way to control this. So I have shown some of our use of this equation to understand why these diverse immune cells are so specialized, how this dynamic immune system can be assessed, and eventually innovate some of the modulating strategy. But one of the most dynamic immune systems what we have is our childhood. Unfortunately, we actually know very little about this. And 2 years ago, we formed this Ian Fraser Center for Children's Immunotherapy Research. We try to understand children's immune system and innovate more therapy, collecting data, make the analysis platform embedded into the clinic. And we have this particular flagship project called Pediatric Immune Cell Atlas for Children's Immunotherapy Research. Very long name, but they can abbreviation as a Pikachu. So hopefully even children can know the name means. With that, I would really acknowledge lots of people and other things. And first of all, I think we are standing on the shoulder of a giant, Jacques Miller, which really opened the field, which is a center for my research for the past 20 years. Landing in Australia 2003, 21 years ago in September, I was very fortunate able to get some of the most brilliant immunologists as my mentor, my PhD superior Carola Venus and Chris Goodenow, my post-mentor Charles Mackay, and most recently Ian Fraser has been a mentor for my research and strategy. Equally, luckily, I get a very diverse and very passionate PhD students and fellows working in my lab, and they have do a lot of great things. I have very proud to show some data to generate. I highlight some of them name. And also, I'm grateful for the 4 institutes I've been working with, ANU, Monash, and Garvey Institute, and UQ, and of course, our collaborator, particular clinical researchers, and also, of course, our founding body. Last but not least, I would like to thank my dear family, and particularly my parents are sitting in the audience, and they trigger my curiosity for the science and my love for the life, and also my brother's family. Thank you.
Just great. Thank you. I'd like to add another couple of M's, and that is you're a wonderful role model, and I know already you're a wonderful mentor to others in your team and far wider than that. Congratulations. Let me open this for questions, please. I'm looking forward to them. Now, while people are thinking, I'd like to ask a burning question, and that is, how much of what you study comes from talking to clinicians and telling you their needs and their gaps in their knowledge, and how much comes from you to reach out to our clinical colleagues?
Wow, that is a question continue to be discussing in the research. I think it's 2 ways. I consider we as an investigator sometimes like a detective. So we go to talk to the clinicians, they have lots of questions, see every day in the clinic, and we try to help them to solve some of the questions, find some clues, but eventually has been tested with them to see whether our explanation is true or false. So that is really the 2-way connection and the discussion.
It seems to work brilliantly. Please, Frances.
Oh, yes, that was brilliant. So I wanted to ask about the selenium. I think it was GPX4, and you mentioned selenium supplements. So how often is selenium deficiency a problem in immune, you know what I mean? How often is selenium deficiency a problem?
So this is a great question. Thank you for asking this. We haven't particularly studied the selenium deficiency in our own project, but in the literature, there's a clear selenium deficiency associated with more infection and a reduced immune response. And the selenium supplementation is to tuning the level of GPS4, and we have shown in the mouse model, even you lack 1 copy of GPS4, it's very subtle reduction. You only got half of the antibody response.
You're happy with that response? I can see the nods. Thank you so much for a marvellous presentation and brilliant work. Gosh. Our next award is the Nancy Millis Medal, honouring the contributions made to science by the late Professor Millis and recognises her importance as a role model for women aspiring to be research leaders. The award is open to women mid-career researchers, 8 to 15 years post-PhD in the calendar year of their nomination, except, of course, in cases of significant interruptions to a research career. It's in any branch of physical or biological sciences, and the recipients will have established an independent research program and demonstrated exceptional leadership. I'm proud to announce the 2023 recipient of the Nancy Millis Medal is Professor Renae Ryan. Professor Ryan's research focuses on neurotransmitter transporters, nanoscale vacuum cleaners that suck chemical messengers back into cells after they have sent their message on to the next cell. In diseases such as Alzheimer's, epilepsy, stroke, these vacuum cleaners can break down, lead to confusion in the cellular communication mechanisms, and ultimately to cell death. Your internationally recognized research has revealed molecular architecture and cryogeography of these minute vacuum cleaners, allowing us to start to understand why they stop working in disease conditions and providing the basis for the development of new medications to treat previously intractable brain disease. Professor Ryan is a globally respected leader and advocate for gender equity, diversity and inclusion, and a sought-after supervisor, mentor and role model for women in science. Please welcome Professor Renae Ryan to accept the medal and deliver the lecture.
Thank you very much, and thank you to the Academy for giving me this award. I didn't know Nancy Millis, but I heard she was an amazing woman, and from what I've read about her, she was a real trailblazer and had to fight for her right to do research and find many different ways to do that. But I also have heard from many people that were mentored by Nancy that she was a wonderful mentor and leader as well, so I'm very proud to get this award. So I work on membrane transport proteins, and cells from all forms of life are surrounded by a lipid membrane that contain these proteins, and they control the movement of everything inside, into and out of a cell. They're vital for energy production, cellular communication, obtaining nutrients, and extruding waste. The proteins I've worked on my whole career until now are proteins from the glutamate transporter family, and I will use this acronym, excitatory amino acid transporters, and that's because in addition to glutamate, they can also transport aspartate. And while they're found throughout the body, really in all organs, they're most widely studied in the brain, and so I'll talk about their role in the brain today. So this is a cartoon of an excitatory synapse, and glutamate is represented by these red circles. And so glutamate is released from the presynaptic terminal, and it will traverse the synaptic cleft and activate its postsynaptic receptors for neurotransmission to occur. But for this whole process to work efficiently, that glutamate needs to be removed really quickly from the synapse once it's done its job. And that is done by these little vacuum cleaners, as was just mentioned, the EATs. And mainly the glutamate is moved into astrocytes or glial cells surrounding these excitatory synapses. And they do this by coupling to other ions because these proteins have to move glutamate against a very large concentration gradient. There's a lot of glutamate in cells, and the resting glutamate concentration in synapses is predicted to be as low as 10 nanomolar. And so to do this, they couple to the inward movement of 3 sodium ions and a proton and the outward movement of a potassium ion, because that's where these ions want to go. The downregulation or dysfunction of these proteins has been linked to many different diseases because glutamate is such an important neurotransmitter in the brain, both acute conditions like ischemic brain damage following a stroke, but also some more chronic conditions as well. But in addition to this well-studied process, these proteins can also activate a chloride channel. And so we know that this chloride channel is found in all members studied thus far. We don't really know what it does, but it's been proposed that it could play a role in charge neutralisation or ionic homeostasis. But we are learning that it seems to be linked to this brain disease, which I'll talk about a bit later, called episodic ataxia type 6. And so back when I did my PhD with Rob Vandenberg at the University of Sydney, we understood that there were distinct conformational states that carried out these two functions, but we didn't really know much about how the protein looked at this time or how one protein could do both of these things, because channels and transporters mechanistically are quite distinct. Channels are open pores, and once they're open, the ions will flow down them. Transporters, on the other hand, have this alternating access mechanism where they can only be open to the one side of the membrane at a time, because if it was an open pore, glutamate would just flow out of the cell. So I've been really interested in trying to understand how these proteins can carry out these two functions. So to do this, structural biology is a great tool to understand what these proteins look like at the molecular level and how they work and how they move. And so this is a glutamate transporter. They exist as trimers. So here we're looking down the bottom of the slide in the plane of the membrane, and here we're looking top down on the top of the cell. And this red circle indicates one protomer, so 3 of these come together, and they're made up of 2 domains, a scaffold domain in brown and a transport domain in blue. And we know from many structures, from our group and others, that they have this elevator type mechanism where 2 sodium ions come in, followed by the substrate and the last sodium ion, and then that whole transport domain actually rotates and moves down to the inside of the cell, while the scaffold domain remains relatively static. And you'll see this happen here in this little video. And then it opens up and releases its cargo to the inside. So this has been wonderful, and that's many, many years of work, really over about 15 years of work understanding these mechanisms. But all of these different structures could not explain the chloride channel component, which we were really interested in. There was a bit of a hint in one of these structures where you could start to see an aqueous pore open up at the interface of the scaffold and transport domain. And we thought this was really exciting because many of the residues we had studied that specifically affected chloride permeation were found facing into this pore. And so we hypothesised that as that transport domain moved down, maybe there was a point at which a continuous aqueous pathway opened up, and this could account for the channel activity. So this was a very long study, and I'll thank the people at the end that did it, but we used a disulfide cross-linking strategy because it's a very transient state. The transporter likes to be at the two end states, and it's very hard to capture these intermediate states. And we used a range of biophysical techniques to study both the function and the structure of these. So out of 30 cysteine pairs, one actually worked, which is why I'm telling you this story today. But we had to use single particle cryo-EM. We had traditionally been in crystallography lab, but that wasn't working for us with this project. And it was around the time that cryo-EM was becoming much more accessible for membrane proteins of this size. So I won't go through the details, but these are the little particles. You can actually see them on that first, sorry, my mouse isn't working, on that first one right on the left. We can get the structure of the protein. And what we had was actually two different states. We had about 85% were in this intermediate state we already knew of. So we're a bit disappointed initially because we already had this structure. And you can see that that's blocked here. There's the beginning of the aqueous pathway, but it's blocked by protein. But what we did get was about 15% was in this new structure, or in this new state where the transport domain had moved along, not very much, and then we saw this continuous pathway. So it really was on the elevator pathway, and you can see we could do molecular dynamics simulations and see that water could go through this pore and also chloride. So that was really exciting, but we were really interested in how this then can be linked to this disease, episodic ataxia. So episodic ataxia is a heritable neurological disorders. There's 8 subtypes, and subtype 6 is linked to the glutamate transporter EAT1. And there are a range of symptoms associated, motor symptoms mostly, and you can have migraine and seizures associated as well. And so this is a multidisciplinary collaboration with collaborators at McGill University and University of Illinois. And what we do is we study the function of these patient-derived mutations in Xenopus levosauricide, so we use electrophysiology. And we have these really cool Drosophila models, so Drosophila larvae models, where you can knock out the Drosophila's normal EAT1 and they can't move. This is these little larvae using infrared tracking. But then if you put in the human EAT1, they completely recover. So we can use this quite cheap and simple and without animal ethics tool to put in these human mutations and look at their motor phenotype. We can also solve structures of these proteins and do molecular dynamic simulations as well. And so when we initially started this project, there were only 7 patient-derived mutations known. So this is a rare disease. And what we found was the common theme was that it was actually a disruption in the chloride channel function rather than the transport, which is not what people had expected. They assumed that these transporters weren't clearing glutamate, and that was what was causing the neurotoxicity. And the other interesting thing we saw was we saw both increases and decreases in chloride gave the same phenotype. Which is still confusing, and we're still trying to understand this. Since we published this paper, many more of these variants have become known to us, including clinicians have reached out to us, and Nick Abreu is one from New York University. And he has a multi-generation family with episodic ataxia, and he has found they have a mutation in EAT1. And the yellow outline here indicates the people that have been confirmed with the mutation, and the black indicates people with these episodic ataxia symptoms. And so we've shown that there is no change to the glutamate transport properties for this mutation, but it has an increase in chloride conductance. And Don has shown in his larvae model that these larvae are very unhappy. They really can't move at all, and so they don't develop into human flies. They die because they can't eat. So our hypothesis is at the moment is that EA6 is caused by mutations in EAT1 that somehow disrupt the chloride homeostasis in glial cells, because remember these transporters are expressed on. So hopefully I've convinced you it's all about the chloride and that the EATs are cool dual function transporter channel proteins. Here you can actually see a chloride ion there in purple moving through the interface, and we can look at every interaction as it's moving through with every single amino acid residue, and we've done a lot of follow-up work to show this pathway. So the EATs obviously do play an important role in clearing glutamate, but we also think that this chloride channel has an important physiological role. And it's activated during this twisting elevator movement. So it explains how one protein can have a controlled transport mechanism, but also a chloride channel at that interface. And we think that this chloride channel dysfunction, rather than maybe the glutamate transport dysfunction, could underlie diseases like EA6. So in future work, we're characterising many more patient-derived variants at the moment, and we've been getting in contact with clinicians all over the world. So if anyone works on this disease or knows of anyone, please point them in my direction. And we're also looking at other diseases linked to other members of this family, like DEE, which is a developmental and epileptic encephalopathy, but also some other spasticity and neurodevelopmental disorders. And lastly, what we're doing, which is quite exciting at the moment, is developing functionally selective inhibitors, because all of the current inhibitors we have block the transporter in the outward-facing state, so they inhibit both functions. But now we have this new state. We've been using that to do virtual screens and had some good success actually in identifying a range of inhibitors that specifically block the chloride conductance. And so these will be really useful just as experimental tools, so we can try and understand the physiological role of these two processes, but potentially as therapeutics. And I just have to do my acknowledgements. And many people in my career have been amazing. My team members, the people up here who did most of the work I talked about today, previous PhD students and postdocs, the current team as well. Also Rob Vandenberg, my PhD advisor, and Eric Goa and Joe Mindell, my postdoc advisors. Collaborators are amazing, our molecular dynamics collaborators and our episodic ataxia collaborators, and we also did the cryo-EM work in collaboration with Alastair Stewart and Meghna Sopti at Victor Chang. I'd like to thank the NHMRC and ARC for funding and all of the access to the microscopes and synchrotron that we use. And lastly, I'd like to thank my family, who are absolutely amazing and wonderful supporters of me. And I think Mum and Dad are watching, so thank you to the wonderful support of my whole family. Thanks.
What a lucid description of beautiful work. Thank you so much. Opening it for questions, please, Suzanne, and then Eric.
Congratulations on your beautiful work, Renae. I was quite fascinated to see this dual function, and I was wondering, are there mutations in humans that affect both aspects of this protein, or one or other of them, and how do they differ in their phenotypes?
Yes, I most probably should have explained that a bit better. So the chloride conductance needs glutamate and sodium to be bound to be activated. So we always knew that, but now we have the structure, we understand why, because you need the transport domain to close and move about halfway across the membrane for it to become activated. So if you have mutations that prevent the substrate or sodium ions binding at all, you just have a dead transporter. It won't transport glutamate and it won't activate the chloride channel. So it doesn't have to go through the whole cycle though. So as long as it can get into that chloride conducting state, you can still have the channel. And we see an increase in channel activity when it's a much slower transporter, which can be impacted by mutations. And some of these patient-derived mutations, we think that's what they're doing. So that it can work, but it might be a bit slower, and so it just spends more time in the conducting state. There's not as many known patient-derived variants of just, dysfunctional transporters, but these are the ones that we're seeing that are functional transporters but have differences in their chloride permeation, a bit more interesting. I don't think that the others will most probably be found if they have a completely dead transporter.
And we can have one quick question and a fairly quick answer, please.
Sorry.
No, exactly. Just keeping to time. Eric, please.
Thank you for your talk. In horses, we see a number of ion channel transport dysfunctions, PSSM, HYPP, you're probably familiar with, and we use therapeutic interventions to assist in terms of feeding, low-carb diets, etc. Do you see the same in this in terms of therapeutics, or what would you predict that could be done? They're also heritable traits as well, and we genetically test.
Yeah, I mean, this is a fairly rare disease. EA1 and 2 are mutations of potassium and calcium channels, and they are more common and very widely studied. There's not really any good medications for episodic ataxias. They use acetosolamide, a carbonic anhydrase inhibitor, which about 30% of people respond to, and actually that family we have put on acetosolamide and it is helping them. And so we're also trying to understand how that happens. It's maybe something to do with pH regulation in the synapse. The dysregulation of the glial cell membrane potential could be affecting many different proteins in those cells. So we're looking into that now, but I just don't think there's enough known about dietary interventions or anything, but yeah, it's useful to think about.
Thank you so much. And other questions I know were there. Am I allowed one quick extra one? If it's very quick, Eric, please.
Actually, it is very quick. I was just curious. I assume that this transporter is energy craving.
Yes. So it uses the energy from the sodium and potassium gradients, which are established by the sodium-potassium ATPase.
Right, so it's an ATP consumer.
Yeah, and that's why it's linked to that. So that's why in stroke, when the sodium ATPase runs down, it stops working. All of those neurotransmitter transporters rely on the sodium gradient mainly.
Thank you. Thank you so much. Again, congratulations and a marvellous presentation. We move now to the presentation of the honorific medals for 2023. These awards will be introduced by the former Secretary for Physical Sciences, Professor Malcolm Sambridge, and the former Secretary for Biological Sciences, Professor Helene Marsh. Over to Malcolm and Helene.
Thank you, Lynn. We'll begin the medal ceremony with the David Craig Medal. This medal is awarded in honour of the outstanding contribution to chemical research of the late Emeritus Professor David Craig. It recognises contributions of a high order to any branch of chemistry by active researchers, and the recipient of this medal delivers several public lectures across Australia. The 2023 winner of the David Craig Medal is Professor David Craig. And it is my pleasure to invite David to the stage to accept this award.
Play is also that there is too much medicine, and we know from studies that about 30% of medical...
In the 1960s, a Norwegian doctor called Lawrence Gran was working as part of a Red Cross relief effort in what was then known as the Republic of Congo.
He noticed that when women went into labour, that the labour would be very fast. And what was actually happening is that they were making a tea from a local plant called Kalata Kalata, ingesting that tea, and it would accelerate the childbirth process.
The active ingredient was a molecule or peptide that Gran named Kalata B1.
But at the time he couldn't determine the structure of this molecule, and it was really 20 years later that I came along and found that the structure had this very interesting head-to-tail cyclic structure that made it super stable, and that explained to us why this molecule could withstand the boiling and then the oral ingestion.
By working that out, Professor David Craig became the first scientist to document circular peptides of this size.
Most peptides, like insulin, couldn't be boiled or couldn't be orally ingested, but this molecule could be, so it led us to this whole new area of super stable molecules.
And a whole new world of possibility. He called these circular peptides cyclotides. Today, thousands have been discovered in plants around the world. Much of that work has been done here at Professor Craig's laboratory at the University of Queensland.
We know that plants make these molecules very effectively. What if we could re-engineer a plant to make a medicine so that, in principle, you could grow medicines in your backyard?
His goal is to cut the costs associated with pharmaceutical development and dramatically increase access to medicine, particularly in developing countries. One example his team is working on right now is to integrate a multiple sclerosis drug into tomatoes.
So that people with chronic multiple sclerosis could maybe have their Greek salad and have some level of medical protection from the disease in their everyday diet. So there's lots of exciting possibilities for turning foods into medicines.
Professor Craik believes there's a big opportunity for Australian farmers to set up biofactories for innovative peptide-based drug production.
Rather than growing a crop that might be worth $100 a tonne, it could be worth $100,000 a tonne by producing a high-value medicine in those plants.
These peptides also have applications in agriculture. One Australian company, for instance, has already developed an insecticide from butterfly pea plants containing cyclotides, which is now being used to protect crops such as cotton. The product is non-toxic, making it safe for pollinators such as bees. Professor Craik says this is just one example of how his circular peptides can be utilised.
We've been working in this field now for 20 years, and when we started there was nothing known. In fact, people used to say this can't be true, you can't have cyclic peptides of the size that we discovered, they just had not been seen before. And then we went on travelling around the world looking at many different plants to find out just how common these molecules were. Really it's come a long, long way from the initial stages where this was a little hobby project 20 years ago, but it's basically become a passion and an adventure for the last 20 years or so.
An adventure that's now been recognised with the prestigious David Craig Medal from the Australian Academy of Science.
Next to the Hannan Medal. This is a career award that recognises outstanding research in any of the fields of statistical science, pure mathematics, applied mathematics, and computational mathematics. It honours the contribution to time series analysis of the late E.J. Hannan, Professor of Statistics at the Research School of Social Sciences at the Australian National University. The 2023 winner of the Hannan Medal is Professor Richard Hartley. Richard was not able to join us today, but please join me in a round of applause to congratulate him, and we'll play the video.
You're looking at a digital reconstruction of Piazza San Marco in Venice. Three-dimensional models such as this one are underpinned by complex mathematical theory.
A lot of my work has been in the understanding of geometry from scenes as taken from images.
Work that began in the early 1990s when Professor Richard Hartley was a researcher for a division of General Electric in New York.
What they wanted to do was simulate flights where they would see models of what they were seeing, particularly the ground and airports, so they could learn to fly into these things.
He used satellite images to work out the shapes of terrain on the ground.
I look at the models that I was building there and feeling how primitive, incredibly primitive they are. One could do better here by saying, what happens if you had two or several views of the scene?
That led him to developing with Andrew Zisserman what's now known as multiple view geometry. Mix his theories with modern-day computers and the possibilities are endless.
There's a famous paper called Rome in a Day, which shows that you can build Rome in a day, or at least you can build a 3D model of Rome in a day from pictures that you have of Rome, thereby disproving the proverb.
Professor Hartley is a rock star in the world of computer vision. He was co-author of a book that documents the geometric principles of 3D reconstructions.
People describe it as the Bible of, or sometimes just the Bible, by which of course they mean the Bible of geometric computer vision.
He says he never takes his copy with him because he knows he can find one in every computer vision lab in the world. You can often spot Professor Hartley riding his scooter around the campus of ANU, which is renowned for its innovations in computing.
And computer vision is an area which is just exploding in importance and the amount of activity in it, and it's nice to be part of a very busy research area.
Professor Hartley is the recipient of the Hannan Medal for an outstanding career in the field of computational mathematics.
Next is the Jaeger Medal. This is a career award made in honour of the contribution of the late Professor John Conrad Jaeger to the earth science. The award is made to a scientist for investigations of a high order into the solid earth or its oceans carried out in Australia or having some connection with Australian earth science. The 2023 winner of the Jaeger Medal is Professor Matthew England. Matt, please join us to accept your award.
Professor Matthew England has been coming to these Sydney beaches for as long as he can remember.
I love the ocean. Even as a 3, 4, 5-year-old, I actually once terrified my parents by running off at the beach and going to a rock pool and spending an hour looking at all the marine life in this rock pool. And they couldn't find me. And it was a big surf day, and they literally, they'd called the police and they thought they'd lost their kid.
But that experience didn't keep him away. He visited the beach most weekends, even while studying science at the University of Sydney.
And I had no idea that my passion for marine life and marine ecology, which was really just born out of being a little 5-year-old kid studying starfish in a rock pool, that 15 years later I'd be at a university studying maths and physics, and somehow the two fields could come together. And it was a great discovery for me. It really changed the course of my degree. Suddenly, I went into a classroom where equations were being written across the board, but they were the equations of motion for the ocean. It was the mathematics of the ocean.
Professor England is now regarded as one of the world's leading experts on the role oceans play in regulating the world's climate.
The summer to winter cycle is made more temperate by having oceans on our planet. The oceans absorb huge amounts of heat during summer, and that buffers the heat extremes we would otherwise see. And then in winter, that heat gets released back into the atmosphere, and so we benefit from a free air conditioning service globally by having the oceans absorb heat during summer, release that heat back into the system during winter.
But that air conditioning system isn't working as it once did. By loading the atmosphere with greenhouse gases, we're forcing oceans to absorb even more heat.
There's a tremendous benefit to the oceans absorbing heat during climate change because it takes heat away from the atmosphere, but there's payback through these processes like sea level rise, ice melt, coral bleaching, and it's those processes of payback where my research sits. It's trying to understand exactly how this heat uptake occurs, where it's happening, how it affects the ocean currents, and how those changes to the ocean currents feed back into the atmosphere.
His research is painting an alarming picture. Professor England says our oceans are effectively being hit by a sledgehammer.
And the reason I call it a sledgehammer is that, we can go back into Earth's past climate and we see very subtle variations in forcing, so Milankovitch cycles and paleoclimate changes that are quite subtle compared to the big perturbation we're imposing today with greenhouse gases. And so globally, greenhouse gases are changing the textbook description of the oceans. Regionally, each current itself is also changing.
In the southern hemisphere, one of the biggest impacts is playing out in Antarctica, where warming water is melting ice shelves.
This warm water that gets to the ice shelves and melts the ice shelves, that itself doesn't raise sea levels, but the ice shelves buttress the ice sheets behind them. And once you melt those ice shelves, it's well known that you greatly accelerate the stream flow of the actual land ice.
And if that happens, the rate of global sea level rise will also dramatically increase.
We do have some deep knowledge of some of the thresholds globally and the ones we shouldn't pass, but it's not like there's an exact number. Cross 2 degrees Celsius and we're doomed. Keep it below and things will be okay. Even at 1.9, there are a whole number of risks. It's just every fraction of a degree Celsius stacks the odds of even higher of some of these tipping points being passed.
Professor England's lifetime of dedication to understanding our oceans has seen him awarded the prestigious Jaeger Medal from the Australian Academy of Science.
As scientists, it's just nice to know when you put so much effort into trying to discover something, trying to understand how the oceans work, that it's made a difference and that somebody else has noticed.
Next is the Suzanne Cory Medal, which honours the contributions made to science by molecular biologist Professor Suzanne Cory, who's in the audience today. The medal recognises outstanding research in all the biological sciences, being awarded in alternate years in the biomedical sciences and in all of the biological sciences, excluding the biomedical sciences. So we're in the second phase here. So the 2023 Suzanne Cory Medal is awarded jointly to Professor Terry Hughes and Professor Catherine Lovelock. Kath was unable to join us today, but please join me in a round of applause to congratulate her. And may I please welcome Terry to accept the award. This is very special for me because Terry nominated me for the Fellowship of the Academy, so I'm really delighted, Terry.
You're looking at pictures taken by Professor Terry Hughes during his recent study trip to the northern parts of Queensland's Great Barrier Reef. It's an area he knows well.
I first visited there in 1990, so I revisited earlier study sites, and yeah, the changes there are quite dramatic. There's a lot less coral now than there used to be.
The reef has had to deal with cyclones, an invasion of the coral-eating crown-of-thorns starfish, and most damagingly, the impacts of climate change. Professor Hughes has spent the past 3 decades documenting those impacts.
We saw the first mass coral bleaching event on the Great Barrier Reef in 1998, and that was really the first wake-up call we had here in Australia about the threat from global warming.
But that wake-up call did little to stem global emissions of greenhouse gases. Since 1998, the reef has endured multiple bleaching events.
Bleaching is a stress response by corals. If you take a coral into the laboratory and torture it by changing the salinity or the temperature or adding sediment or testing toxic chemicals, it will bleach. The whiteness is actually its skeleton, showing through the translucent tissue like an upside-down jellyfish.
Bleached coral is sick coral, and back-to-back bleaching events in 2016 and 2017 meant many couldn't survive.
In 2016, we lost about 1/3 of the corals on the entire Great Barrier Reef. Most of that loss was in the northern third of the Great Barrier Reef, where more than half of the corals died over a period of just a few months following record-breaking high temperatures.
Professor Hughes and his team saw it happen.
And the best way to do that at the scale of the Great Barrier Reef, which is 2,300 kilometres long, is to get in a small airplane and crisscross the length and breadth of the reef. It takes about 8 days to do that. We also have to ground truth that data underwater. So in 2016, I led a team of about 100 people who spent about a month underwater measuring in situ on the reef the amount of bleaching and the amount of mortality subsequently.
He also told the world what he saw. One of his social media posts was retweeted by actor and environmentalist Leonardo DiCaprio, reaching an audience of millions.
We no longer have the luxury of waiting a year or two from collecting data, analysing it, publishing it after peer review in a journal. The timeline for that might surprise people. A year's really fast. It's more typically about 2 years. And that's a big gap if you're trying to communicate important information to a general audience.
Professor Hughes says reaching a mainstream audience is critical in securing the political will needed to enact the kind of changes that could save coral reefs around the world.
I think it will probably get worse before it gets better. We often use the term running the climate gauntlet for coral reefs. What we really need to see is global temperatures stabilise, hopefully at somewhere close to 1.5 or 2 degrees. If we can do that, then we will still have coral reefs in the future. They'll be quite different from the coral reefs of today, and certainly the coral reefs of 30 years ago. But I think if we can curb global warming at a reasonable level and give the corals time to respond evolutionarily, which will take a couple of decades, then we will see a rebuilding of coral reefs if we can stabilize temperatures at a sufficiently reasonable level.
His career studying, understanding and advocating for coral reef systems has now seen Professor Hughes awarded the prestigious Suzanne Cory Medal from the Australian Academy of Science.
Next is the Thomas Rankin Lyell Medal. This is a career award that commemorates the contribution of Sir Thomas Rankin Lyell to Australian science and industry, in particular to his own fields of physics and mathematics. The purpose of the medal is to recognise outstanding achievement by a scientist in Australia for research in mathematics or physics. The 2023 recipients of the Thomas Rankin Lyell Medal are Professor Susan Scott and Professor Nick Wormald. Firstly, I invite Susan to the stage to accept the award.
I'm Distinguished Professor Susan Scott, and I'm an expert in general relativity theory, cosmology, and gravitational waves.
That means Professor Scott's day job is to unlock the secrets of our universe by studying the fabric of space-time. But the answers don't come easily.
The theory which covers that is Einstein's theory of general relativity. Singularities are places in space-time where the laws of physics go badly wrong. An example of that could be at the centre of a black hole, where we have things like the curvature of space-time going infinite. So we know that there's a problem there, and we know that we need a new theory and new insight to cover what's happening inside those areas that we believe to be singular.
Her work has been greatly enhanced by the detection of gravitational waves.
One of the best examples of information we can get from gravitational waves that we can't get from other things is, of course, the collision of two black holes, because by their very nature they're black and they don't emit electromagnetic radiation. So we can probe the formation of those systems using gravitational waves. But very importantly, and this is perhaps the holy grail of what we do with gravitational waves, is that one day we want to be able to probe right back to the start of the universe.
Professor Scott is a member of the international team that achieved the first ever direct detection of a gravitational wave. Her mathematical models help to determine the violent sources that generate these waves. Professor Scott started working on gravitational waves some 18 years before the first confirmed waves were received by the massive LIGO detectors in the United States in 2015.
We were very convinced during all that time that we were going to achieve this goal. It was just a question of making the instruments sensitive enough to be able to do that.
Professor Scott believes Australia could be the perfect place to build a third-generation gravitational wave detector.
We are looking at possible sites in Australia, and we're very fortunate in this country that we do have the space to have a lot of possible sites. We have to look at things like seismic quietness. So it could be a LIGO-like instrument, but with much longer arm length. Instead of 4 kilometres, maybe 40 kilometres.
And Professor Scott is well positioned to make the case. She's the first Australian to be elected a Fellow of the International Society on General Relativity and Gravitation, and is now the recipient of the prestigious Thomas Rankin Lyell Medal for outstanding achievements in mathematics and physics.
And I now invite Nick to the stage to accept his award.
From the World Wide Web to the roads connecting our cities, networks are just about everywhere. In mathematics, random graphs are used to model these complex networks.
With random graphs, you take a set of objects and you are not told which ones are related, but the relationships come randomly.
Professor Nick Warmold is a world leader in random graph theory and one of Australia's most renowned mathematicians. While his work is largely theoretical, many of his methods have been used by other scientists to solve real-world problems. One of his major projects has been about developing a way to apply differential equations to random networks. While networks are made up of nodes linked together as discrete steps, differential equations describe continuously moving processes such as a car accelerating. Because of that difference in context, the two were rarely combined prior to Professor Warmold's differential equation method. But it turns out his method has many uses, including in the area of mobile phone communication. Messages are sent between phones as ones and zeros, and some of these bits can get lost.
The way it works is you send actually more information. So a very simplistic way of doing it is if you send every message twice. So then if one of the messages gets corrupted, you can use the other one. But that wastes too much space.
So a group of US researchers created algorithms that used random networks to make this process much more efficient.
Part of their research, they come to a question, well, what would this algorithm do on this network? How would it behave? Then they can throw in the analysis by the differential equation method and get some answers.
Other research he's done on how to generate random networks has also provided a robust framework for researchers to map and test their work in different networks and contexts. That's important, not just in telecommunications, but in other fields such as ecology and computer science. More recently, Professor Warmold also worked with collaborators on how to use network theory to optimise underground mines.
One of the intriguing questions that came up there was with certain constraints on how you can dig tunnels and underground mines, what's the cheapest way to make a network of tunnels that reaches a given set of locations underground? There are a lot of constraints on the tunnels, like a maximum angle they can be at for the trucks to drive up and down. But the version of the question I heard didn't mention underground mining. It was just, here's a problem. And the problem sounded interesting.
Interesting problems have driven the direction of Professor Warmold's research for decades now. He says there's nothing quite like the excitement of finally cracking a case. For his outstanding career-long achievements in pure mathematics, Professor Warmold has been awarded the Thomas Rankin Lyell Medal from the Australian Academy of Science.
This sort of recognition doesn't come along very often in a lifetime, so when it does, it really gives some sense of achievement.
Next is the Anton Hales Medal, and I'd like to acknowledge Denise Hales in the audience today. Welcome, Denise. The Anton Hales Medal recognises distinguished research in the earth sciences and honours the contributions to the earth sciences by the late Professor Anton L. Hales. Professor Hales was the founding director of the Research School of Earth Sciences at the Australian National University. Please welcome the 2023 recipient of the Anton Hales Medal, Professor Teresa Awardee.
Volcanic eruptions can be awe-inspiring to watch. But their unpredictable nature means they remain a threat to many.
Today, almost 10% of people around the world live within 100 kilometers of an active volcano, and so we need to understand how volcanoes work and why they erupt.
Associate Professor Teresa Ubide is a volcanologist at the University of Queensland. She travels the world collecting volcanic rock samples so she can analyse their chemistry back in the lab.
I am particularly interested in the crystals that grow inside the volcano and are transported to the surface during eruptions.
That's because these crystals serve as a time capsule of what was happening inside the volcano before it erupted. She uses an ultraviolet laser to remove thin layers from the crystals, which she then analyzes for changes in trace chemicals.
And I build an image of the distribution of elements in the crystal so that I can see what happens inside the volcano before the eruption. And so we can access the guts of the volcano that we may not be able to see otherwise.
And by doing so, Associate Professor Ubide is unlocking some explosive secrets.
The more we study volcanoes, the more we realize how complex they are inside and how actually they are fed by a very complex network of reservoirs and conduits that are responsible for bringing the magma from depth to the surface.
Understanding that process could help us better predict when they will erupt. For example, her research has shown that as magmas travel towards the surface, they are filtered to a consistent composition in many volcanoes around the world. That may indicate many eruptions are caused by magma reaching a specific set of physical properties, or sweet spot.
It's a bit like shaking a bottle of Coke, and this ideal mix of properties propels the magma all the way to the surface.
Associate Professor Ubide also explores the link between volcanoes and the critical metals needed for the development of renewable energy, such as wind and solar. Copper, found at the boundaries of tectonic plates, for instance, is known to be associated with the magmatism caused by one plate diving beneath another.
So if we want to understand the Earth and its resources, we need to do research into those critical processes, including volcanic processes that drive accumulation of metals for a sustainable future.
Associate Professor Ubide says it's a great honour to receive the Anton Hales Medal and is thankful to those who nominated and supported her. She hopes to inspire other women to pursue a career in STEM.
I'm very excited to contribute to building role models for future girls to study STEM. I think it is really important that we build a diverse and inclusive STEM workforce to make the discoveries for our future.
Next is the Christopher Heide Medal, which honours contributions to mathematics by the late Professor Christopher Heide. In recognition of Professor Heide's broad interest in mathematical sciences, the award is offered in one of three fields of mathematics on a rotating basis. In 2023, it recognises excellence in elliptic and parabolic partial differential equations. Congratulations to the 2023 recipient, Dr. Valentina Wheeler.
Ever wondered why bees build their honeycombs as hexagons?
And the answer is because it's a way to minimize the amount of wax that they use to construct those hexagons, but maximize the amount of honey volume that they put inside. Nature is an exceptional mathematician in terms of minimizing effort and creating the best outcome.
Finding ways to model different phenomena in nature is part of what Dr. Valentina Wheeler does as a geometric analyst. She studies a class of equations known as curvature flows.
Curvature flows basically take this object and they say, I'm going to modify this object in time, so it's changing its shape in time, and I want to give it a speed to which it modifies.
This framework lends itself well to describing moving interfaces, making it useful for things such as bushfire modelling. In her own research, Dr. Wheeler and her colleagues have used curvature flow theory to create a model that predicts how a fire front will move.
If you have a corner fire, so you have two flat lines, front of the fire, which are merging, they form a corner. At this corner, the fire will develop into a jump. So the speed of the fire in the corner will be, I would say, increasingly bigger than the one on the flat sides. And this type of jump behavior is similar between phyrons and then curvature flows.
This isn't the only application that Dr. Wheeler is exploring. She's also looking into collaborations with biologists to model red blood cells in patients with spherocytosis, a disease where someone's red blood cells are spherical instead of disc-like.
If somebody can understand the cell and why it becomes and when it becomes spherical, then we can create targeted drugs. And that would be something that you can bring in a team of biologists and work together with. We can tell you when these blood cells become spherical based on the parameters, which you can control.
Finding these applications for pure mathematics isn't always easy and could take decades or even centuries. But Dr. Wheeler says it's the pursuit of fundamental knowledge that motivates her the most.
All mathematicians take their work home. We never stop thinking.
Her outstanding research in pure mathematics has now seen her awarded the Christopher Hyder Medal from the Australian Academy of Science.
Next up is the Dorothy Hill Medal, which honours the contributions of the late Professor Dorothy Hill to Australian earth science and her work in opening tertiary science education to women. Please welcome the 2023 recipient of the Dorothy Hill Medal, Associate Professor Raffaella De Michaelis.
Associate Professor Raffaella De Michaelis was never what you'd call a computer geek. But these days she spends a lot of time using our national supercomputing facilities at NCI in Canberra and here at the Pawsey Centre in Perth. She needs the massive processing power for something she is passionate about, improving our fundamental understanding of materials.
I make models of minerals and other materials to understand how they are formed, how they grow, what's their properties, and how they interact with their environments.
Associate Professor De Michaelis is a computational chemist at Curtin University. Her mission is not just to understand nature, but to mimic it. She believes that could be key to overcoming one of the big environmental challenges of our time, climate change.
In nature, mineral surfaces undergo all sorts of chemical transformation that are able to sequester carbon dioxide, to transform carbon dioxide into fuel, so into other organic molecules. And all of this happen in nature. We know it happens, but we don't know the chemistry.
Her models are trying to change that. Not only could her findings help efforts to remove greenhouse gases from the atmosphere, but they could also assist with the production of new materials and even clean energy.
Probably the most important part of my work is to make sure that the models that I develop are realistic, because with a computer and a bit of coding and a mathematical equation, you can make basically everything happen. But does that make sense? Is that realistic? So a lot of my time is spent in challenging my models.
Associate Professor De Michaelis grew up in Italy. She clearly remembers the moment her schoolteacher explained how water molecules rearrange themselves during freezing and melting. That was the day she decided to pursue a career in science.
I saw all of these pretty pictures and I said, wow, I want to do that. I've always been very much interested in the theory of things.
But understanding the theory of things isn't always straightforward. A lot of her experiments end in failure.
The curiosity of finding out, I think that's what drives most scientists to overcome the failure. It's very important also for the community, because you tell people don't do that because that's not going to work. So failure is a scientific result.
But Associate Professor De Michaelis has had no shortage of success, including being awarded the Dorothy Hill Medal for the critical impact of her work in the earth sciences.
Next is the Fenner Medal, which is awarded in honour of the outstanding contributions to science by the late Professor Frank Fenner. It recognises distinguished research in biology. The recipient of the 2023 Fenner Medal is Associate Professor Emily Wong. Please welcome Emily to accept the award.
It seemed a far-fetched idea. Could the DNA from a Queensland sea sponge shed light on one of the biggest mysteries in animal genomes?
I was in a lab which was working in the sponge, so I guess that's where that idea came from.
Associate Professor Emily Wong and her colleagues were determined to better understand a certain part of DNA sequences called enhancers.
They are regions in the genome which control when and where genes are expressed. We think around 30% of the genome has some sort of annotated enhancer function.
Enhancers perform a critical role in influencing cell identity and certain traits such as size, coloration, and disease susceptibility in multicellular organisms. While they take up a huge proportion of the genome, they're particularly hard to find.
A lot of these sequences, they change really rapidly, so they evolve really rapidly. So the methods that we use to identify protein coding genes across genomes don't work well with enhancer sequences.
That means they're still poorly understood. In fact, they could be considered the dark matter of the genome. This may be why scientists hadn't found any enhancers that were similar across the animal kingdom until the sea sponge experiment.
When we first saw the results, I was very skeptical.
I didn't believe it.
Associate Professor Wong and her collaborators at the University of Queensland extracted enhancer DNA from the sea sponge and injected it into a single cell from the embryo of a zebrafish. While the enhancer sequences found in the sponge were very different to those in the zebrafish, they still successfully drove the expression of a protein in zebrafish cells. The findings, published in Science, suggest humans, mice, zebrafish, and most likely the entire animal kingdom share enhancer regions that drive the expression of certain common genes. And given sponges and humans last shared an ancestor more than 700 million years ago, it means the functional mechanism has been preserved throughout evolution. Associate Professor Wong says this discovery means there's still a lot more to uncover about enhancers.
We really need to focus on understanding these regulatory elements and understand how they function.
That's the mission behind her research at the Victor Chang Cardiac Research Institute in Sydney. Solving the puzzle will help scientists better understand basic biology and genome evolution, as well as predict diseases.
We are looking especially in congenital heart disease. We're trying to understand how genetic variants at enhancers could impact gene expression and that could lead to a disease.
Associate Professor Wong says she feels incredibly honoured that her work has been recognised with the prestigious Fenner Medal from the Australian Academy of Science.
So next is the Gottschalk Medal. This award recognises the contribution to science by the late Professor A. Gottschalk, and it recognises outstanding research in the biomedical sciences by researchers up to 10 years post-PhD. With us today, we have the award recipients of 2022 and 2023, Dr. Ailsa Glukova and Professor C. Ming Man. Please welcome first the 2022 recipient, Dr. Ailsa Glukova, to accept the award.
Dr. Elisa Glukova is a structural biologist. She uses techniques such as X-ray crystallography and cryo-electron microscopy to closely examine proteins within cells.
It's almost like seeing is believing, so you can actually see how things look, and from there you can infer how they work.
Specifically, her group studies G-protein coupled receptors, or GPCRs. Embedded within the membranes of cells, the receptors serve almost like inboxes for outside information, helping to regulate a range of functions in the body. That makes them critical for modern medicine.
Right now, about 35% of all drugs on the market target these transmembrane receptors.
But not all GPCRs are the same. Humans have almost 800 different types, with each responsible for specific signals. Dr. Glukova is trying to better understand a group of adenosine GPCRs.
And they specifically respond to a small molecule which usually gets released when cells are dying, for example, from hypoxia.
When the receptors bind to adenosine, pro-survival stimuli are sent within the cells, usually stopping the cells from dying. Her group was the first in the world to get structural insights into the A1 adenosine receptor. And it's that fundamental knowledge that could lead to better therapies for things such as heart disease and inflammation.
As a result of my research, somebody else can come in and design better small molecules, better drugs to target multiple diseases.
A passionate advocate for basic research, Dr. Glukova has been awarded the Gottschalk Medal for outstanding contributions to biomedical science.
Now please welcome Professor C. Ming Man, the 2023 recipient of the Gottschalk Medal, on the stage to accept the award.
Inflammation is one of our body's natural defences to outside invaders.
Inflammation can be good because it can help us fight infections, but inflammation can also have a dark side, which means that it can cause diseases like sepsis and cancer.
Good inflammation is generally acute, happening quickly and resolving as soon as our immune system is happy. But bad inflammation tends to be chronic and prolonged.
Prolonged inflammation can eventually damage your cells and tissues and induce all types of different stress signals.
The work of immunologist Professor C. Ming Man centres around finding the right balance of good and bad forms of inflammation. His team at the Australian National University has uncovered the underlying process that triggers acute and chronic inflammation.
We have identified different immune sensors, just like different alarm systems within our body, which can be activated in response to different types of pathogens. And what's exciting about this research is that we can harness the power of this immune system and turn them into drugs.
Drugs in the form of antimicrobial peptides that can destroy different types of pathogens, which are the cause of some types of inflammation. The peptides are highly selective, targeting only bad bacteria, including the ones that are increasingly becoming resistant to existing drugs.
I believe that our drugs can kill multi-drug resistant bacteria, which is what we have been showing in the lab. We believe that these peptides can bind directly to the surface of the bacteria and basically rip open the envelope, the outer surface of the bacteria, so it can no longer survive and replicate within the cell.
The team has shown this process works. The next step is to improve the safety profile and stability of the drugs before moving to clinical trials.
Stability comes with how long the drug lasts in the body and how many doses you need to be able to effectively treat the infection.
Professor Mann's team is also working with clinicians at the Canberra Hospital to improve early detection methods for bowel cancer. They can do that by looking for inflammation biomarkers within a patient.
Often people get diagnosed when it's too late, so it's kind of like a prediction tool.
Professor Mann says he knows the innovations will benefit humankind, calling this a massive incentive to work even harder. His contribution to medical research has now been honoured with the Gottschalk Medal from the Australian Academy of Science.
I think this medal will continue to inspire me and other young scientists to continue to take risks with their research and dream big.
Next is the John Booker Medal in Engineering Science, which recognises outstanding research in the engineering mechanics that addresses problems in the static and dynamic response of physical systems within engineering and applied mathematics disciplines. It honours the memory of Professor John Robert Booker, who worked in the area of theoretical geomechanics, was Professor in the Department of Civil and Mining Engineering at the University of Sydney, and held a personal chair in engineering mechanics at the University of Sydney. I'm pleased to invite the 2023 winner of the John Booker Medal to the stage, Dr. Amelia Liu.
For most of us, the glass used in windows or our phone screens hardly seems like a mystery. But it turns out the nature and structure of this material still baffles scientists to this day.
Glasses are essentially the problem child of condensed matter physics. Physicists at some level don't really understand why glasses are solid at all.
Dr. Amelia Liu is a physicist at Melbourne's Monash University. And here at the Monash Centre for Electron Microscopy and the nearby Australian Synchrotron facility, she's uncovering the beginnings of an answer to the so-called glass problem. Using highly sensitive instrumentation, she's developed new techniques to analyse disordered materials such as glass at the atomic level. Unlike crystals, which have an ordered lattice structure, the atoms in disordered materials are arranged more haphazardly. This makes them challenging to study with conventional methods such as X-ray crystallography.
For crystalline materials, this is basically a solved problem. The diffraction patterns are highly symmetric, and we have all the tools we need to solve the structure uniquely in terms of the diffraction pattern. But for glasses that have a very disordered structure, the problem is not solved. In my research, one of the major advances that I made was shrinking down the beam of X-rays or electrons to the size of a coordination polyhedron in a glass and collecting a diffraction pattern from that very, very small volume.
By zooming in to the level of individual atoms and their nearest neighbours, Dr. Liu discovered that while glasses have disordered structures on the larger scale, they have local symmetries. Her research found that this local symmetry gives glass its rigidity, while any structural deformation that can result in fracture corresponds to a reduction in that symmetry.
Glasses have one very well-known Achilles heel, which is that they are brittle. So when we deform them beyond their failure point, then they fail catastrophically, and a crack goes right through the whole material. And so one of the challenges with glasses is how can we engineer them from the atomic level to mitigate this brittle behaviour?
She hopes her techniques can ultimately address this challenge and help engineers design tougher materials. For her outstanding research in engineering mechanics, Dr. Liu has been awarded the John Booker Medal from the Australian Academy of Science.
Next is the Lefebvre Medal, which is the research in chemistry, commemorates the work of the late Professor Raymond James Wood Lefebvre. Its purpose is to recognise outstanding basic research in chemistry. The 2023 recipients of the Lefebvre Medal are Associate Professor Rana Chandrawati and Professor Tianya Ma. Firstly, I invite Rona to the stage to accept the award.
This tiny sensor could help to overcome a very big problem.
In Australia, we waste $10 billion worth of food, and 70% of that food is still edible.
Many of us throw away food once it passes the best before date shown on the packaging. But those dates don't offer an accurate real-time indicator of the food's actual quality. Associate Professor Rona Chandrawati and her team at UNSW Sydney may have the solution to this. They've developed highly sensitive nanosensors that react to compounds generated by food that's gone bad.
For example, when meat is boiled, they produce ammonia, and our polymers will react with this ammonia. And then it changed the backbone of the polymer and changed the way it absorbs light. So in our eyes, they change color.
Blue indicates the meat is fresh, but a red patch means the meat shouldn't be eaten. The sensors have been shown to work on not just meat products, but milk and fruit. They don't need any specialized equipment to work, and the team hopes the cost to consumers will be minimal.
The food sensor that we developed in our lab here is low cost. They are approximately 0.2 cents per sensor that can be attached onto food packaging. And we make this low cost because we want it to be accessible.
Associate Professor Chandrawati is a chemical engineer and materials chemist and believes the best ideas often come from personal experience.
I feel guilty as well when I waste food when it probably can still be safely consumed. So I thought, with my background and understanding materials chemistry and understanding engineering and polymer chemistry, how can we use this knowledge working together with the team to develop a solution to this problem?
Talks are underway with packaging companies to roll the sensors out at scale. But the technology has potential applications beyond reducing food waste.
By changing the chemistry of the polymers, we can develop the sensors to detect many other target compounds, for example, in environment, in agriculture, or monitoring health.
That might mean detecting heavy metal contamination in soil samples or certain chemicals in a patient's blood. Associate Professor Chandrawati's growing list of honours now includes the Lefebvre Medal from the Australian Academy of Science for outstanding research in chemistry.
Now please welcome Tianyi to the stage to accept the award.
Professor Chen-Yi Ma is a globally renowned renewable energy scientist who's all about chemical reactions, especially making them faster and cleaner. Here at Melbourne's RMIT University, he's developing technologies to address challenges in the energy sector.
It uses functional materials to refine renewable energy into whatever we need, like fuels, clean fuels, compounds, fertilizers, and medicines.
One of the inventions he's proudest of is a 2D device that can float on the surface of seawater. The device directly converts solar energy into chemical energy to speed up the process of splitting water into hydrogen and oxygen. It's estimated 100 metres of this floating device could generate 1 litre of pure hydrogen in 1 hour. While hydrogen has long been proposed as a clean fuel that could be used to power households, industries and cars of the future, much of it is still produced from fossil fuels. But Professor Ma's device offers a greener alternative.
We built a single unit of 1 square meter that can generate hydrogen, and then we replicate the 1 square meter unit. We just simply fabricate 100 of it. We can scale the technology up for 2 orders of magnitude.
Professor Ma isn't solely focused on creating eco-friendly energy sources for the future. He's also working on ways to tackle our current carbon emissions. He's now in the process of building a device that captures exhausted carbon dioxide and uses an electric current to upgrade it to useful components such as methane and formic acid.
The traditional way is we convert fossil fuels by high temperature, high pressure, thermochemical processes. And these processes, you can have to build a huge reactor. By using the advanced catalytic processes, we can do at ambient condition, which is user-friendly, very safe.
The benefits of his technologies haven't gone unnoticed. Every year since 2020, Professor Ma has been named by analytics company Clarivate as one of its global highly cited researchers, a title given to scientists who've demonstrated a broad and significant influence in their research areas. In 2022, he was the highly cited researcher in both the chemistry and materials science fields.
I'm quite passionate about what I'm doing because they are not only fundamental science breakthroughs, but also they have far-reaching industry impact.
That impact has now seen Professor Ma awarded the Lafleur Medal from the Australian Academy of Science.
Next is the Moran Medal, which recognises the contributions to science of the late Patrick Alfred Pierce Moran. It recognises outstanding research by scientists in one or more of the fields of applied probability, biometrics, mathematical genetics, psychometrics and statistics. The recipients of the 2023 Moran Medal are Associate Professor David Fraser and Dr. Rachel Wang. Firstly, I invite David to come to the stage to accept the award.
Ever heard of Bayesian statistics and probability? Even if you haven't, you most likely practice it daily.
The joke, in a sense, is that all humans are Bayesians because we have this sort of subjective belief about probability that we're assigning to the events around us. And once we observe an event, we then use that event and our subjective beliefs to update what we think is going to happen about the future.
Associate Professor David Fraser works in the Department of Econometrics and Business Statistics at Monash University. He's spent years researching the Bayesian paradigm of probability and says he can explain this key idea with a simple example. Imagine a group of parents who drop off their children for school each day, and every morning the kids are safely escorted across the road thanks to the school crossing guard.
And you watch them cross every single day. Then that is your existing data set. And what a Bayesian does is they say, okay, I have that data set. Now I'm going to update my beliefs about the likelihood of them successfully crossing the road tomorrow via what I've learned and my prior beliefs about that. So my posterior beliefs on the observation of all these data points of them crossing the road is going to lead to a very high probability of them successfully crossing the road. We do this sort of calculation thousands of times a day. We just don't call it Bayesian probability.
Bayesian statistics now plays a significant role in machine learning and artificial intelligence. Billions of data points can be crunched in real time to deliver probable outcomes on things that impact our everyday lives. For instance, services such as Netflix and Spotify use algorithms based on so-called approximate Bayesian methods to suggest shows or songs users may like. Associate Professor Fraser's work has focused on understanding the behaviour of these Bayesian methods and finding ways to ensure they deliver the most accurate predictions possible. He says this is crucial when the predictions can affect much more than the media we consume.
A good example of this is, say, self-driving cars, right? A lot of self-driving cars rely on what are called variational Bayesian methods. Those are methods of approximating an exact Bayesian learner for microsecond updates as to when the car should stop. Robotics uses these methods a lot to try and make sure that when they have self-driving vehicles or drones or something like this, they make sure that they're not going to crash into anything.
Associate Professor Fraser says working at the cutting edge of Bayesian probability has shown him just how important the language of mathematics is to the world we live in.
Every single aspect of our lives now is driven in some fashion by a model. What I do essentially is I use mathematics and statistics and data to test and make sure that those models that we base every single aspect of our life on are in some sense robust.
This work has now seen him awarded the Moran Medal from the Australian Academy of Science in recognition of an outstanding contribution to applied probability and statistics.
Now please welcome Dr. Rachel Wang on the stage to accept her award.
Every day, more than 2 billion people around the world log on to Facebook. Beyond the endless cat videos and status updates, each of those users has helped create a social network which can be modelled to identify communities and find out how information spreads online.
With a lot of simplification, of course, you can abstract this representation into a graph, a mathematical object. And you've got people as nodes and their relationships, whether they are friends or not, as edges.
The University of Sydney's Dr. Rachel Wang is an expert in statistical network models. She finds them everywhere and not just online.
In ecology, you have the food web, which is not an example of network. It's a predator-prey relationship. And in molecular biology, you've got the gene networks. So you have these transcription factors and the target genes interacting with each other, forming the gene regulation mechanisms which govern how cells function.
Highly sophisticated algorithms have been developed to assign the various components of a network into subgroups or clusters. In the case of Facebook, for example, you could place people into implicit social circles based on common interests or backgrounds.
Finding these groups is likely to give us clues or information about, for example, how information spreads on the social network, how infectious disease spreads throughout a social network.
But reliable outcomes rely on valid statistical inference and the quality of algorithms. And that's where the work of Dr. Wang comes in.
My job, of course, is to study the theoretical behavior of these algorithms, whether there is any theoretical guarantees for the performance of the algorithms under certain generative assumptions on the model.
That's a vital mission, given the increasing use of so-called black box algorithms in which key decisions are left to machine learning without deep understanding of how those decisions are reached. Dr. Wang is currently working to strengthen conformal prediction tools, which offer a way to assess the certainty of the predictions made by machine learning models. Her statistical methods have also been critical in extracting new biological knowledge from genomics data and improving our understanding of the inner workings of cells. It's diverse work, but a career path she could have missed out on had she allowed a stereotype she encountered at a younger age to discourage her interest in mathematics.
When I was growing up, I kept hearing the message from other people around me that boys are better than girls in science, maths. Girls are better at more artsy subjects. I just discovered I liked doing math, so I didn't care.
Dr. Wang says she hopes being awarded the Moran Medal from the Australian Academy of Science will encourage other girls to pursue a career in STEM.
And our final award for this ceremony is the Palsy Medal, which recognises distinguished research in physics and commemorates the work of the late Joseph Palsy. The recipient of the 2023 Palsy Medal is Professor Yue Liu. Please welcome Professor Liu to the stage to accept the award.
Professor Larry Liu says it's easy to sum up nanotechnology.
Nanotechnology allows us to do big things from a tiny world.
You'll often find him here at the Australian National Fabrication Facility in Canberra, developing the nanofabrications needed for the next generation of technologies.
We can make extremely thin, high-speed, and focus-tunable microlenses based on those ultra-thin materials. So those kind of properties cannot be realized using conventional semiconductors.
Professor Liu leads the Nano-Electromechanical System Lab at ANU. He says the nano world has fascinated him since he was an undergraduate student.
It's so interesting because you cannot see it by your eyes, but we can develop different types of techniques to see it, touch it, and engineer it.
Techniques such as mechanical exfoliation, where crystals grown in the lab are thinned down layer by layer until they're about one or a few atoms thick. It's a material so slim that his team has successfully used it to create the world's thinnest lens. These micro lenses have a wide range of potential applications.
It opens the door to flexible computer displays and miniaturized cameras.
He says those cameras would have the potential to improve the way medical screening is performed.
The device could be small enough to be inserted into the blood vessels to provide high-quality images. Those images will help specialists to better understand the causes of heart attacks and help understand how to prevent them.
The technology is still a few years off being ready, but it's the possibilities that keep Professor Liu motivated.
The most exciting part is that we can make discoveries that can potentially change our future life.
For his outstanding research and contributions to applied physics, Professor Liu has been awarded the Pawsey Medal.
And before I sign off, I believe we have two members of the Pawsey family in the audience today, Hastings and Liz. Where are you? There you are. Welcome.
Thank you, Helene, and thank you, Malcolm. That brings to a conclusion this part of the day, where we now have lunch in the Jaeger room, the Dorothy Hill room, and outdoors. Dietary requirements are in the Dorothy Hill room. May I also acknowledge the donors who have made the awards possible, to the Pawsey family, Hales family and others. Thank you so much for being here, Brian and Diane. It really makes, I think, it very, very special for everybody involved. I hope for you, and I know certainly the award recipients very much enjoy meeting you and thanking you for your generous contribution. I'm not sure if TJ Higgins is in the audience. Are you with us, TJ? If anyone spots TJ, it's his birthday today. It's a significant birthday. Is that him right at the back? You're hiding, TJ. There you are. Happy birthday. We will continue with the awards presentations after lunch at 1:30 p.m. Due to popular demand, we've had lots of calls, especially from our fellows, for more presentations, not through video, but in-person presentations from our awardees. So we have been able to incorporate that back into the program at Science at the Shine Dome this year. So you'll have an opportunity to hear directly from those awardees then. Enjoy your lunch, and we'll see you back after it.
immune system plays a key role in protecting us from microbial invaders, but its overactivity can lead to complications.
The capacity to respond to a pathogen is just as critical as the capacity to turn off the response when it's no longer required. When an immune response is not turned off, then the dysregulated immunity and the ensuing inflammation can cause a lot more problems than those caused by the actual virus or the pathogen itself.
Professor Maria Pia degli Esposti studies that delicate balance. As an immunologist, she has made pioneering contributions to understanding natural killer cells and their role in the immune system. These white blood cells have long been recognized as a first line of immune defense, the early assassins that promptly kill infected cells.
We now know that NK cells have functions that are much more sophisticated than we previously thought. And our work has contributed to a body of knowledge that identify the regulatory function of NK cells. These functions are well beyond their capacity to attack and kill, and they extend to their ability to actively regulate immune responses and to adjust various facets of the immune system to ensure that the pathogen, the threats, is effectively handled while also turning off the initial response once the issue has been addressed.
Her team developed innovative preclinical disease models based on infection with a prevalent virus known as cytomegalovirus, or CMV, to better understand how the immune system functions. CMV is unusual in that once it infects a person, it is never completely eliminated. In most cases, the immune system seemingly manages the virus so that it doesn't cause symptoms. But when the immune system is compromised, for instance in transplant patients, the virus can reactivate and cause significant illness.
And it is in those clinical situations where we need to be able to intervene and take action.
Professor degli Esposti and her team found that using antibodies, specifically ones that matched the particular strain of infecting CMV virus, offered the best protection against CMV reactivation. That discovery won them the Eureka Prize for Scientific Research in 2019. It's impactful research that speaks to why Professor degli Esposti fell in love with science and immunology in the first place. Growing up in Italy, she had initially wanted to be a pediatrician, but she soon realized her passion lay elsewhere.
What I was much more excited about was the potential to understand the origins of diseases, as this would really enable us to alter their trajectory. And the best way to achieve this is by fundamental research, because that allows us to understand and ultimately target the processes that are at play.
Her critical work has now seen her elected a Fellow of the Australian Academy of Science. She says she accepts the honour on behalf of her entire team.
The scientists say this probably means they are dealing with some new deadly sexually transmitted disease.
It's been 40 years since the first description of AIDS-related illnesses. We still don't have a cure or even a vaccine, but experts in the field say that doesn't tell the full story.
40 years seems a very long time. However, the progress in HIV has been extraordinary. I mean, in many ways, it's to me the biggest and most important scientific advance in the last century, because we developed a way of managing a virus that had a universal death sentence and make it a completely manageable chronic illness, all through science and the availability of antiviral drugs.
HIV is a virus that gradually destroys cells used by the body to fight off disease. It can severely damage your immune system, making it much easier to develop diseases such as tuberculosis and some cancers. The most advanced stage of HIV is known as AIDS.
Even in Australia, about 1 in 5 people diagnosed with HIV are diagnosed so late their immune function has deteriorated to a point where they have AIDS.
But overall, new cases of HIV in Australia have halved over the last decade. In fact, Sydney is on track to become the first place in the world to end community transmission of HIV by 2025.
It's time to make history.
Effective elimination is defined as a 90% reduction in new cases over a decade. The big improvement is largely due to high testing rates and preventative measures taken by gay and bisexual communities. But infection rates in the heterosexual population haven't fallen as sharply.
The Kirby Institute's Professor Rebecca Guy says the data shows why regular sexual health checkups are critical.
The test is like the gateway to a whole range of things that are important for a person. So you think about HIV, if you have a test and you find out it's positive, you can start treatment straight away and you can live the same longevity of life as everyone else and remain healthy.
While accessing an HIV test is relatively simple in Australia, the same can't be said for many places in the world.
People at risk of HIV, men who have sex with men, for example, there are still countries that criminalise that activity. And so how can you get a public health message out if the activity is considered criminal?
Even where it's not illegal, the stigma around HIV can sometimes drive down testing rates. That's one reason the Kirby has led the push for more screening options, including self-testing kits for people to use at home.
They're not quite as accurate as a lab test, but I guess the way I always think about it is having a test, even if it's less accurate, is better than no test at all.
Professor Sharon Lewin is a Fellow of the Australian Academy of Science and President of the International AIDS Society. As head of the Awardee Institute, she played a key role in fighting COVID-19, a virus that we managed to have vaccines for within 12 months. So why haven't we been able to do the same with HIV?
Well, HIV is a very different virus to COVID or influenza, which are viruses that can be completely cleared from the body. HIV is unable to be cleared from the body, and that's because it's a virus, part of a family, that's able to become part of a person's DNA.
And that happens within hours of a person becoming infected. Treatments allow the immune system to recover and can even stop the virus from being passed on. But the virus doesn't disappear. It's just hiding and will re-emerge if treatment stops. Professor Lewin has dedicated her career to understanding this phenomenon, known as HIV latency.
I've been on the hunt for understanding where the virus hides, how to mimic latency in laboratory models, and ways to coax it out of hiding.
While we don't yet have a vaccine, we do have access to a medicine that can help prevent HIV, known as PrEP. If taken correctly, the daily tablet can lower the risk of infection by 99%.
In Australia, we have a public health system that makes medicine free, essentially. We have a fantastically mobilised, vocal and articulate gay community that have done a lot of the education around accessing PrEP, and our uptake of PrEP is one of the highest in the world.
PrEP doesn't offer protection from other sexually transmitted infections, so doctors recommend the use of condoms. Professor Lewin says PrEP is just one of many advances in HIV research that goes to show the power of science. She believes it's only a matter of time before HIV is cured once and for all.
It's become a very important, high priority area of research because we now have treatment, we now have widespread access to treatment. Treatment's relatively simple. The next big challenge is finding a cure.
You're looking at the largest single dish radio telescope in the world. It's called FAST and located within the mountains of southwestern China.
And that is by far the most sensitive radio telescope in the world. I first went there while they were putting the first few things on the ground, and it was amazing to see how quickly in China you'd come back a year later and the whole dish would be built.
At the heart of FAST is a primary multi-beam receiver designed and built by Australian scientists. China chose to collaborate with Australia because of our experience with the Parkes radio telescope in regional New South Wales.
The Parkes radio telescope is by far one of the greatest telescopes in the world. It's been upgraded many times, so it keeps getting upgraded. It's not a 60-year-old telescope. The most recent upgrade was just a couple of years ago with a very wideband receiver system that allows us to observe the radio waves over a much wider bandwidth than any other telescope on the Earth.
Parkes has special significance to Dr. Shi Dai. His first visit was as an international student.
I mean, the second you see the 64-meter big telescope there with wonderful weather, blue sky, I was really shocked. I thought this is the way to do astronomy.
He later returned to Australia to complete his PhD under the supervision of George Hobbs, before accepting a teaching position at Western Sydney University, where his research focuses on rapidly spinning neutron stars known as pulsars. The Parkes Radio Telescope has detected more pulsars than any other facility in the world.
Radio astronomy in Australia is really strong. The experience and expertise here are really the most attractive for a student in China.
That's thanks in part to the work of people such as Professor Elaine Sadler. For more than 30 years, Professor Sadler has been at the forefront of research into astrophysics. Her team discovered a 5 billion-year-old galaxy using the ASCAP radio telescope in Western Australia.
Now, 5 billion years ago, the Earth didn't exist. So in the time that signal's been travelling to us from this galaxy, the sun has formed, it's made planets, the Earth has settled down, it's got oceans, life has evolved, we've got a technology, we've built telescopes, and there we are just in time to catch this signal.
Professor Sadler started working regularly with Chinese scientists 7 years ago. She says international collaboration is critical to understanding our universe.
I think one of the great things about working collaboratively with people is it allows you to build up the trust and the common interests that allow you to make advances in science.
Astronomy is a very collaborative science. You can't just do your own research simply on your own.
It's about actually exchanging your idea with different people, different background, different expertise. If we don't have that, we're not doing the best science.
Now to growing concerns about the deadly coronavirus.
Can the spread of this highly infectious disease be contained?
As word spread of a potential global health emergency, Professor Sharon Lewin was up early every day. She'd jog while listening to COVID-19 updates from the New England Journal of Medicine.
I actually do really like physical exercise or ways to be completely distracted from all the other issues that cloud your mind in a busy work week.
And as director of the Peter Doherty Institute for Infection and Immunity, there was plenty on her mind. A joint venture of the University of Melbourne and the Royal Melbourne Hospital, the Doherty played a key role in formulating Australia's pandemic response. The Institute's scientists were the first outside of China to develop a lab-grown version of the virus.
And we made a decision very rapidly to share it globally, and the virus was available to over 70 different laboratories around the world.
The Institute also created the first COVID PCR test in Australia and diagnosed the first patient.
Professor Sharon Lewin is the Doherty Institute's director. Sharon Lewin.
Professor Lewin became a familiar face on Australian television, sharing findings from COVID modelling and updates on vaccine development.
I never thought to myself, we're under immense pressure here, we must deliver. I was very conscious that science, public health, it's a real group effort.
She says there are plenty of lessons from the pandemic, including the vital need to effectively communicate science to mainstream audiences.
Science plays an incredibly important role, but science is only useful if it's done in partnership with the community and also our political leadership.
And she believes the world's leaders must take steps to ensure low and middle income countries aren't left behind in the next global health crisis.
So we have to make sure we've got a system that can produce 7 billion vaccines or vaccines for everyone very quickly, and there's equitable distribution.
Fighting to improve health outcomes globally is a day-to-day job for Professor Lewin. She spent more than 2 decades unlocking the secrets of HIV and is now president of the International AIDS Society. Professor Lewin was at the epicenter of HIV science in the late 1990s, undertaking a postdoctoral fellowship alongside the famed AIDS researcher David Ho in New York.
He had just become Time magazine's Man of the Year after pioneering the use of combination antiretroviral therapy, which transformed HIV from a terminal disease into a chronic disease. There was genuine hope it would pave the way for a cure.
And within 3 weeks of me arriving in New York in December 97, HIV latency was discovered.
HIV latency is when the virus goes into hiding within the body's cells, preventing its eradication. Her research has focused on not only finding where it hides, but coaxing it out so it can be destroyed.
About 1 in a million cells carries a virus that we actually care about. So you've got to sift through 1 million cells to find that infected cell. But we've now got ways to do it.
More a marathon than a sprint, but Professor Lewin, now proudly a Fellow of the Australian Academy of Science, remains determined to reach the finish line.
We now have widespread access to treatment. Treatment's relatively simple. The next big challenge is finding a cure.
Distinguished Professor Xin Huoyu is at his happiest when he's making things. He built his first transistor radio at the age of 10.
Little radio I can hear. I was so excited.
Professor Yu grew up in China during the Cultural Revolution. Universities were closed, so he figured that once he finished school, he'd become a village electrician. But his plans changed after the death of Mao Zedong.
I had the opportunity to enter the university exam. Did pretty well, so I went to this University of Science or Technology of China.
And so began a long and successful career in electrical engineering, one that brought him to Australia in 1989 for a research fellow position at the University of Adelaide with legendary applied mathematician, the late Ren Potts.
I was so surprised. My English was very poor. Written was all right, but English so poor. But I think he probably sees some value in me, which I couldn't see.
Professor Yu was determined to experience everything Australia could offer, so accepted a lecturing position at what was then known as the Capricornia Institute of Advanced Education in Rockhampton. He was there when it became Central Queensland University and was soon named a professor. But Rockhampton was a world apart from his hometown in China, Harbin, known for its ice sculptures and winter days of minus 30 degrees.
That's the reason why I chose Melbourne. I went to Rockhampton so hot, and this is actually just right.
His time here at RMIT has seen him receive international recognition for his contribution to the theory and application of discontinuous control systems. A simple example of these systems would be a light switch, where the control action changes suddenly, as opposed to a dimmer switch where things change more steadily. Discontinuous systems have a central role to play in things such as robotics and smart energy grids. Complex mathematical theories need to be applied to properly understand how each component interacts.
How to ensure the system, because there's so many on and off, like switching, on and off, and how to maintain the systems continue to supply when you need it. That's the challenge.
Professor Yu considers mentoring students to be a major part of his role.
So I always tell them that first thing, when you come here, you think about 10 years later, what do you want to be? And we design the program for you to reach your potential.
It's clear that many of Professor Yu's students see his success as an inspiration. He's certainly had an incredible journey from making transistor radios as a child in China to being a Fellow of the Australian Academy of Science.
Michelle Hobbs spends a lot of time at some of Australia's most beautiful waterways. She's searching for one of the world's highly threatened and imperiled groups of animals, the freshwater mussel.
Typical day in the field, we rock up in the truck and we walk down and have a look, see if the site is suitable. We will deploy a water quality probe, take some measurements of that, and then we'll start looking for mussels. So that means sticking your hands in all of the dark holes and in the mud on the sides of the banks.
It can be laborious work, but it's all part of a critical effort to collect baseline data on mussel populations in Australia.
It's really important to get a handle on what the populations are like at the moment, so we've got something to compare to in the future.
It's important to be aware of any declines in numbers or changes in distribution given the enormous role mussels play in our ecosystems and communities. Not only do they clean the rivers they live in by removing algae and bacteria from the water, they're also culturally significant to many Indigenous peoples worldwide. But mussels are facing increasing pressure from threats such as dams, water pollution and extraction, as well as climate change.
We really have to turn to the First Peoples to incorporate their knowledge. There's an enormous wealth of information there that we can make use of.
That's an approach Hobbs is taking in her own research as a PhD candidate and associate lecturer at the Australian Rivers Institute at Brisbane's Griffith University. She plans on working with First Peoples across Queensland and New South Wales to bring their insights on water management to the forefront. She's also looking to travel to Canada and the USA to collaborate with Indigenous researchers there. Hobbs is one of 2 recipients of the 2023 Aboriginal and Torres Strait Islander Scientist Award, which recognises outstanding students and early and mid-career researchers in the physical and biological sciences.
It's a huge honour, it really is. I know I stand on the shoulders of everybody who's gone before me, so I just really hope that I can make a really positive contribution with my research.
Stephanie Beaupark describes herself as both a scientist and an artist.
When I started out at university, I just knew I loved chemistry and I loved being an artist. And so I just studied both, and I kind of let it lead me to where I've come to today.
And that's to study the chemistry of eucalyptus dyes, including the ones she uses in her own artworks.
I want to know what causes the colour, like what compounds are involved, what combination bind together to make the dye itself. I just want to know the bigger picture of how dyes work.
This curiosity first came about when Beaupark was doing an internship on Indigenous knowledges of climate and weather patterns in Western Sydney. The internship got her thinking about how natural dyes change across the Indigenous calendar seasons. To answer those questions, Beaupark is now making her own dye in her lab at the University of Wollongong, where she is a PhD student and associate lecturer. She's in the process of doing this with her favourite species, the Eucalyptus cinerea, which produces a bright red dye. Beaupark says she ultimately wants this process and her PhD project to be collaborative and community-led.
This project, I'm making it start off really open so that we can be driven in the direction that community wants it to be.
Beaupark says she's excited to receive the 2023 Aboriginal and Torres Strait Islander Scientist Award, but not just for the recognition. She has been searching for a way to reimburse artists and knowledge holders for their help on her project.
So this enables me to do that the right way and just make sure that people get the benefit that they deserve for their knowledge.
Taking medicine in the future could be as simple as eating a salad.
The example that we're actually doing at the moment is to have a multiple sclerosis drug in tomatoes so that people with chronic multiple sclerosis could maybe have their Greek salad and have some level of medical protection from the disease.
These drugs are based on molecules known as peptides. They are smaller than proteins and carry out important biological functions such as regulating other molecules, and some even act as hormones. Several peptides have pharmaceutical potential, including as potent painkillers. But because most break down if they're taken by mouth, Professor David Craik and his colleagues have engineered them into more stable circular peptides called cyclotides. Cyclotides are naturally found in some plants, so Professor Craik's team can embed them into edible plants such as tomatoes.
One of the issues with current painkillers is that often they don't work, and then we also know with opioids there are horrible addiction problems in the so-called opioid crisis. This is a different class of molecule that doesn't have those addictive effects.
Professor Craik says the potential applications extend beyond chronic pain and disease to areas including agriculture.
We could be creating a new biofarming industry in Australia where farmers could be, rather than growing a crop that might be worth $100 a tonne, it could be worth $100,000 a tonne by producing a high-value medicine in those plants.
The innovation isn't far from becoming reality, with the first crops potentially commercialised in the next few years.
Obviously there's lots of regulatory and social and ethical issues that we would need to address in doing this, but I think it does open up a new world for combining agriculture and medicine and delivering medicines in a novel way.
At first glance, this could be a plantation of Christmas trees in the Australian outback. But you're looking at what some are calling one of humanity's biggest ever scientific endeavours. This is an artist's impression of what's known as the SKA Low Telescope, currently being built in Western Australia. It's one of two critical components that, when completed, will together form the SKA Observatory, or SKAO. The second part is the SKA Mid Telescope, now under construction in South Africa. SKAO will consist of more than 130,000 antennas and almost 200 dishes across the two continents. That will make it the largest radio observatory in the world, promising to answer some of our biggest questions about the universe. ANU's Professor Naomi McClure-Griffiths is a Fellow of the Australian Academy of Science who discovered a spiral arm of the Milky Way and is chair of SKAO's Science and Engineering Advisory Committee. She says SKAO will enable science to take a giant leap forward.
It's the chance to be able to see the very first stars in the universe and when those turned on. And then everything from the very beginning to us here and looking at how planets form in our own galaxy.
For that to happen, radio telescopes must be located a long way from other human-made transmissions, such as TV, radio, and mobile phone signals, which can interfere with the relatively weak radio waves coming from space. That makes this site on Wajiri country ideal. It's already home to precursor telescopes like this one. The site itself is called Inyarimana Ilgari Bundara, the CSIRO Murchison Radio Astronomy Observatory, which translates to sharing sky and stars. The traditional owners agreed to host the science facilities on their land as part of a broader agreement that will ensure educational, social, and economic benefits for community. And the benefits to society as a whole can't be overstated. Science Minister Ed Husick says SKAO's cutting-edge technology will expose Australian businesses to new skills and capabilities. We will see those changes flow on for generations to come. SKAO will also enhance Australia's growing space industry, with scientists from around the world making regular use of the observatory. Some will even be on the lookout for intelligent extraterrestrial life. If an alien species were out there trying to reach out and say hello, by the time that radio signal got to Earth, it would have been so weak we wouldn't be able to detect it, says SKA Low Telescope Director Dr. Sarah Pearce, but that changes with SKAO. The observatory will start producing science before the end of the decade.
You're looking at what brought Professor Madeleine Van Oppen to Australia.
Coral reefs have such beauty, they're just amazing, and they're really important for the marine environment because coral reefs are home to about 30% of marine species, so they're incredibly important in that sense.
In 1997, she took up a postdoctoral position at James Cook University. The following year, the world witnessed its first global mass coral bleaching event, another stark reminder of the consequences of climate change.
When you see the damage, that is very frightening.
Professor Van Oppen started to investigate the relationship between corals and the tiny algae and bacteria that live within them, known as microbial symbionts. A relationship that we now know is vital to the coral's health and survival.
We also now understand that the microalgae that live in the coral tissues play an important role in the level of heat that the coral can cope with.
Coral bleaching occurs when intense heat waves cause corals to expel the algae living in their tissues.
The corals cannot live without these microalgae, so we often see a high rates of mortality following these intense summer heat waves that cause coral bleaching.
But Professor Van Oppen's research has shown it's possible to also harness those microalgae to help the corals cope better with heat.
So certain species of microalgae give the coral better protection against heat than other species. We're sort of playing with that. Can we make them more tolerant to heat by manipulating that relationship?
A question that has since paved the way to a whole new approach in coral restoration known as assisted evolution. The concept was first introduced in a paper Professor Van Oppen published in 2015 with her colleague and renowned coral biologist, the late Ruth Gates.
Sorry for the short lunch, but I hope everyone enjoyed lunch and you're able to connect with friends and colleagues during the break. So it's now my pleasure to introduce the Macfarlane Burnet Lecture for 2024. This medal and lecture recognises the scientific research of the highest standing in the biological sciences. It's a career award that commemorates the contributions to science by Nobel laureate Professor Sir Frank Macfarlane Burnet. The 2024 recipient of the Macfarlane Burnet Medal and Lecture is Professor David Lindenmayer. David is an international authority on conservation and landscape ecology. He has discovered novel ways in which key drivers of landscape change interact to affect biodiversity, ecological processes, and ecosystem conditions. Through a pioneering series of large-scale, long-term studies in forests, plantations, and agricultural environments, he has uniquely demonstrated how pre-existing landscape conditions combine with new kinds of landscape transformation to shape temporal and spatial patterns of species decline and recovery at multiple scales, from individual trees to sites, landscapes, and regions. David has also discovered mechanisms through which species respond to multiple natural and human disturbances. With his unique perspectives across an array of ecosystems, he has established innovative strategies for the management of biota and ecosystems in Australia and globally. He has also developed new conceptual models and approaches for designing experiments and other kinds of studies to quantify the effect of multiple interacting factors on biodiversity. Please welcome David Lindenmayer.
Thank you very much. It's an honour to be here, and in my next 20 minutes I want to go through some of the new findings associated with disturbance and how sometimes that begets more disturbance. So in this talk I want to talk about fires in forests, both globally and nationally, some of the mechanisms, how disturbance leads to further disturbance, this new theory about what we call disturbance-stimulated flammability, and some of the implications for management. So the talk is really based on the last 42 years of empirical measurements, both in forests and in woodlands, including post-fire environments across a series of large fires from 1983 to the Black Summer fires in 2019-2020. And these are often built around large-scale long-term studies, which I'll talk about in a moment. And as part of those studies, there's a lot of work that's looking not only at biodiversity and biodiversity responses to fire, but also the entire ecosystem response and multiple disturbances and interactions between those disturbances. So these are some of the large-scale studies that we do across temperate woodlands, coastal heathlands, and the tall wet forests of Victoria. And each of these long-term studies is associated with permanent sites where there's extensive empirical measurements made, not only of the biodiversity but also the vegetation structure and the disturbance responses. And here's some of the first measurements. Here I am disguised as a much younger man making these measurements, and they continue to this day, literally. Next week I'll be at Jervis Bay, we'll be measuring bird responses to not only fire but also weed control. So one of the key processes here is obviously climate. And there's no doubt that long-term climate and short-term weather are key drivers of fire. Not surprisingly, we don't see too many fires when it's cool and wet. But we need to account for climate and weather when we're analysing fire and other effects. And we also need to understand that forest management, plantation management and woodland management also has implications in how fire behaves. So one of the key things that we see is that the size and frequency of wildfires is increasing in many places around the world. Not all, but many places. And we've recently charted what's happening with the frequency of fire in many parts of the world, including Australia. And there are some parts of northern Australia which have burnt 42 times in the last 20 years. But there are also parts of the Amazon and small parts of the Himalayas that are also burning at extraordinary levels of frequency, which is outside the range of natural variability for these systems. And what we see in wood production forests around the world is a steep increase in the amount of that wood production forest that's burning. And some of the places that that's happening include Australia. So we see the burn trend in red that's increasing, for example, in North America and Australia. This doesn't include Canada in the recently, but that's also seen a substantial increase in the amount of fire that's taking place there. We can also see that there are some places where the frequency of fire has declined in the last 15 to 20 years. So that trend is also seen in Australia, where we see the size of fire and also the frequency of fire changing quite dramatically. So these red bars represent the size of fires in southeastern Australia, and you can see in the last 20 years we've had 3 megafires, where a megafire is defined as a fire that's more than 1 million hectares in size. Prior to that, there was only 1 in the previous century, that was 1939. So in the 20 years of this century, we've seen 3 versus 1 in the previous century. Oh, looks like we're stopped. Okay, so we can see this at a local level as well, or a regional level. This is for northeastern Victoria, and the green bars at the top represent the fire frequency between 2000 and 2020, and the red represent fire frequency from 1980 to 2000. And there's a highly significant change in the frequency of fire between those two times. And the important thing here is that we're starting to see areas that are burning 3 or 4 times in the last 25 years, when the natural fire regime should be 1 fire every 75 to 150 years. And so we're seeing these big changes in the mean fire interval, and sometimes in some cases 5 fires in less than 100 years, and in other cases 4 fires in less than 25 years, and more mega fires. But there's another component to this story that I think is really important, and that is that there's an effect of the pre-disturbance condition on the fire itself, and that's a pre- and post-fire interaction. Oops, we seem to be jammed. Okay, so this is a summary of the 2019-2020 fires, and the different colours on the graphs represent the forest fire danger index. Red being the most severe, or the most extreme, and green the least. And this is across 3 broad regions: Victoria, New South Wales, Southern New South Wales and Northern New South Wales. And essentially what we're doing here is comparing logged versus unlogged in terms of the fire severity, in terms of canopy damage. And what we see is that logged forests always burn at higher severity than intact forests, but the effect is so substantial that a logged forest burning under moderate fire weather conditions still burns at higher severity than an intact forest burning under extreme conditions. So this is a very substantial effect. The important thing here is that the effect is not linear. So what we see is a pulse of flammability that increases quite substantially around this period of about 10 years to about 40 years, and it takes the forest about 80 to 100 years for that effect to start to subside. So it's a non-linear effect that's skewed. And so what we see is that young, logged and regenerated forests are highly flammable, and they reburn at very high severity. Others are finding this. This is a statement from a paper by Wilson et al. In 2022 in terms of the propensity of these forests to burn when they're young. And now there are people seeing this all over the world, including most recently right throughout Canada. And this is right across a whole range of different ecosystems. So why is this occurring? Well, when we cut a forest, we leave a lot of debris behind. About 60% of the biomass in a forest is actually left in the forest as logging slash. The ground layer is dried out. We lose key things like tree ferns, which are very important mesic elements of the forest. The soil is drier. The architecture of the forest is changed with dense, almost wheat field-like young trees growing up. Often they're dominated by quite flammable young plants, and the microclimate is changed. We've actually measured that. It's drier, it's hotter, and the temperatures are more variable. So there are some key drivers of why we see this change in flammability. And it's interesting that others, in a practical sense, are already aware of this. So there's a safety note in the middle of this image. Safety note, high fuel loads forestry slash. So this is part of the fire grounds in 2019-20 down on the south coast of New South Wales, not too far from where we are right now. The other important thing is, beyond the local level flammability pulse, there's also increasing evidence for what we call spatial contagion in the way that fire moves across these already disturbed landscapes. And so this is a logging and fire interaction at a landscape scale. So if you see a classically cut forest these days, you'll see a logging coupe in one area, another one in another spot, another one in another spot again. And essentially what's happening is you're cutting pieces out of the landscape sequentially over time. And each of these areas then is cut and then regrown is more flammable. But what happens is that there's a spillover effect of that flammability on neighbouring areas, including ones that haven't been cut. And so this is really important in the way that fire spreads across the landscape. So with these extensive areas burning at higher severity than spilling across the landscape in this way, this forms what we call a landscape trap, where you have repeated fire that reburns the forest at high flammability, high severity. The forest is then reset to be young, in a young flammable stage, and it continues to reburn, so we don't see a recovery of mature forest where flammability is lower. And eventually, if you have enough repeated fire at a high enough severity, the system collapses into something else. And so this landscape trap, we've now seen all of the characteristics and the features that underpin landscape traps actually developing not only in our landscapes in Victoria, south, but also in places like Tasmania. So this comes from David Bowman's group in Tasmania. Widespread logging and wildfire increased the landscape scale of repeated fire consistent with a landscape trap. So with this information, we've started to come up with this idea of DSF, disturbance stimulated flammability. And what we see is that disturbance changes the biomass structure, the biomass regrows from the ground level. There's processes like understory thinning and changes in the traits of plants that are in the system, plant growth, self-pruning of trees. And so we see this pulse of flammability in the early stages of recovery, which then dissipates as the system gets older. So the least flammable, lowest severity conditions are actually in the oldest forests. Now, this is a really major problem because when we look at what's happening in forests across southeastern Australia, we've seen a 77% reduction in old growth forest across Victoria in the last 25 years alone. Major problem. And so we've recently published on this idea of disturbance-stimulated flammability, and other people are now coming to us and saying, well, that's exactly what we see in Canada, that's what we're seeing in Western North America, northeastern North America, Patagonia. So how are we going to manage these highly flammable forests? It's now a really big issue. Lots of people are recommending thinning. Maybe we should get in and thin the forest. Well, our response as scientists is to look at the data. Let's compare thinned versus unthinned forest in terms of what the severity of the fire was, its propensity to burn. And the answer from the empirical data, looking both at the 2009 fires in Victoria, the Black Saturday fires, and the 2019-20 fires, the Black Summer fires, was that thinning either had limited effect on fire severity, or in some cases it actually seemed to contribute to flammability. So that is not really going to be an answer, particularly as we can't thin forests above 18 degrees slope. That becomes a serious logistical problem. So then another issue becomes, what did the forest actually look like at the time of British invasion? And this is a very controversial area. This photograph shows one of our sites deep in the O'Shanassy water catchment. So it's part of Australia that has had no prolonged logging history at all. And you can actually see a photo of yours truly in a red jumper at the base of these trees. These are the world's tallest flowering plants. Quite remarkable places. It's site 450, just for some context. So in the popular literature, Bill Gamage's book and then Bill Gamage and Bruce Pascoe's book, there's a sense that these tall wet forests were open and park-like, they were farmed, and they were deliberately burnt, either by recurrent prescribed burning or in some cases where First Nations people walked up from the coastline, burnt down the forest and then walked back again. And this has big implications for management. It suggests that maybe we should manage these forests by burning them regularly and doing widespread thinning. Okay, so as scientists, we needed to look at the evidence. That's very important. That's why we're at the Academy. So we look at the historical testimonies from early explorers. We can look at paintings, we can look at photographs, we can look at carbon dating, dendrochronology, soil carbon, and look at the fundamental basic biology of how these plants respond to fire. And so when we look at early paintings, it's pretty dense, not open and park-like. Early photographs, and I can tell you I've spent many weekends in the National Library going through these images and through the testimonies. We'll come to that in a moment. And it's interesting talking to First Nations people about how these areas of old growth were actually very important for groups like the Gunnerkurnai to remind them of what their country was like at the time of British invasion. And it's very interesting to look at the early records. This contrasts very strongly with Bill Gamage's approach. What we see is from records from Strzelecki, Human Hovel, Broadrib and a whole range of others, is that the forests were often very dense, very wet, in many cases impenetrable. And we have hundreds of these testimonies from various diaries. And in terms of historical ecology, it's quite clear that Aboriginal people used these forests. They collected lyrebird feathers, the pith of tree ferns, the roots of sassafras and other plants, but they actually didn't live in the forests, which is really quite important. And you can see why. These people were not silly. These places get 2 metres of rain per year, and if you stand still for very long, you get covered in leeches. I can attest for that from the last 42 years. These people were using the forests, but they weren't living in them, and I think that's really important to understand that. So the overwhelming conclusion from the evidence, from multiple angles, triangulating the data, is that the forests are naturally dense and wet. They're not regularly burnt. The natural fire regime in these systems is high severity, rare fire, occurring on intervals of between 75 to 150 years, in some places up to 300 to 350 years. First Nations people that we talk to, in terms of that interface between Western scientific knowledge and First Nations knowledge, is that this was no fire country. And no fire is actually part of active management of these systems. They were certainly not farmed. There is no data, no evidence from LIDAR showing that these areas were extensively farmed. If you take LIDAR information from places like South America or Southeast Asia, underneath the canopy of the rainforest, you'll see diversions through irrigation, field boundaries, villages, all kinds of things. We don't see that in these forests. These are probably some of the most intact forests anywhere in the world, which is actually an extraordinary outcome in terms of land stewardship. That, I think, is an extraordinary celebration of stewardship of probably almost unparalleled relative to many other parts of the world. So these forests were not open and park-like. They weren't burned, they weren't thinned. So in this case, extensive thinning and widespread burning are really not appropriate management for these kinds of systems. I think it's really important to understand that. Prescribed burning in a forest that has 2 metres of rain per year is not appropriate for these systems. And sometimes that will either crash the system because there's too much fire, the trees aren't old enough to produce viable seed crops, so they're killed. If you do have a burn that manages to take in those forests, it will climb through the understory to the overstory and you'll have a major wildfire. So disturbance often in these systems makes the system more flammable, not less. And the fact that you have this pulse of flammability in the early stages of development from 10 years to about 40 or 50 years suggests inherently that these forests are systems where fire was relatively rare. Otherwise, frequent fire will be maladapted to how these systems work. So one of the key things to do is to actually help these forests to grow through to an old growth stage which is less flammable. Now, this is an enormous challenge. How are we going to do this? And our response to that is that we need to think about embracing new technologies to detect fires more rapidly and suppress them more quickly, and to use mathematical modelling through lightning strike modelling and the like to work out where we're most likely to have ignitions, be able to suppress those ignitions quickly, suppress fires when they're still small because that's when it's most effective, and to work with the system and work with what we call ecological controls on fire. This is a concept that my colleague Phil Zylstra has worked with, rather than fight against the forest in this context, against the fundamental ecology of it. You know, the sad reality is that if we can kill someone with a drone sitting in a wheelchair in Gaza, we can use that same technology to detect fires very quickly and suppress them very quickly. And it potentially is an industry that will help us around the world, given that there are problems with fire in Portugal, in Greece, in Scandinavia, in the Arctic and the like. And this opens fantastic opportunities for this country to make a contribution to other people's fire problems as well as our own. So I'm going to leave it there. I've got 1 minute and 31 to go. So thank you very much for listening.
Thank you, David. We'll now open the floor to a short Q&A session.
Sure. Sorry.
Maybe I'll just kick off and say, given that the flora is so different here in Australia than, say, the Amazon or in Canada, how does your wonderful work inform what's going on over there?
Yeah, that's a good question. So some of these other systems are also seeing pulses of flammability in that earlier stage. It doesn't occur in all systems, but it does occur in some of the systems such as the Nothofagus in Patagonia and some of the Douglas fir forest systems in Western North America. So we're not saying this is the model that's occurring everywhere. What we're saying is that this is one of the mechanisms that contribute as a positive feedback leading to these increased flammability systems. And the trick here is to look at the combination of traits, structure, composition that gives us these kinds of signals in the system.
Yes.
Is the cessation of logging in Victoria real or smoke and mirrors?
It's smoke and mirrors. So at the moment we have extensive fire breaks being put in across Victoria that will sum to about 6,000 kilometres of fire breaks. So that's up from 1,500 kilometres. Fire breaks are going to be somewhere between 40 metres and 100 metres wide. So that comes to 24,000 hectares over the next 5 years. At the time that Vic Forests was logging, they were logging about 3,000 hectares a year. So it's an increase from about 3,000 hectares to 4,800 hectares a year. The problem is that there's actually no evidence that the fire breaks are effective. They weren't effective when they were put in 2007, prior to the 2009 outcome. And so the recommendation for us is think about cost effectiveness. What is likely to be more cost effective? Are we well off spending $300-400 million a year putting in these fire breaks? Or should we be looking at new technologies and looking at moving our fire suppression activities close to where it matters? The data from work that we did in 2012, led by Phil Gibbons, shows that prescribed burning has some effectiveness, except under extreme conditions, but it's most effective when it's conducted close to where people live. The key thing to look at in terms of the response variable is loss of life and loss of property. And so prescribed burning needs to be done frequently and it needs to be done close. And so these activities with these industrial scale prescribed burns done in very remote areas is actually counterproductive. And so we can rethink with some science about how we can better focus what we're doing.
Suzanne?
David, first I want to thank you for your incredibly important work and congratulate you on this award. I wanted to make one comment and one question. The comment is that by burning our forests, we must be totally decimating the native populations, and so we're sterilising our forests at the same time. The question is, when you were mostly talking about old growth, wet forests, but a lot of Victoria, where I'm from, is more open kind of country. Is prescribed burning good for the more open kind of forests, or is it bad there also?
Okay, so I think we need to be careful here about ensuring that we think about what's called the fire regime, which is the sequence of fires in an area over a prolonged period of time. And the fire regime is different for each ecosystem. So what's an appropriate fire regime for a heathland or a dry inland colitrus forest will be different for a tall wet forest. So we need to make sure that we match the fire regime where we can manage it relative to the ecosystem that we're dealing with and relative to the particular species that we're concerned about. So what suffices, for example, in a tropical savanna in the Northern Territory, a la Alan Anderson, will be quite different to what happens in these tall wet forests. What we've seen historically is that the areas of inland Victoria, temperate woodlands and temperate grasslands, have a very different fire regime to the foothill forests and the wet forests and the cool temperate rainforests. So we need to be quite careful about what's happening here, and we also need to look at the total fire burden. And the total fire burden is repeated wildfires, prescribed burning, and on top of that now, increasingly cultural burning. So cultural burning is not the same thing as industrial hazard reduction burning. So this fire space needs a lot more nuance in it than is often the case. And it doesn't lend itself naturally to an 8-second soundbite, but it does lend itself to good science and good evidence to guide what's happening, and that's not always the case at the moment.
Yeah, look, we have lots of questions and not enough time. Eric, let me give you the last one, I'm sorry, and then maybe you could talk to David later on.
Thank you very much, David. Absolutely engrossing lecture. I'm just wondering, obviously, the reason why we've got disturbed forest is because of the forestry industry. And the question is, how does one balance the national need for timber with trying to protect forests so that they're more resilient in a warming climate?
Okay, what you do is you look at the structure of the industry itself. Okay, so the native forest logging industry is 90% of what comes out of forests. Native forest goes into the wood chip, paper pulp and packaging stream. 90% of our sawn timber in Australia actually already comes from plantations. But I think an even more difficult statistic is that 97% of our homegrown plantation eucalypt pulp logs gets exported. And sometimes we buy it back. So when you go to Coles or Woolworths, you look underneath, you'll see Vietnam, made in Vietnam. Now, some of that might be Australian material, or it might be Australian eucalypts being processed there and brought back. So we need to process more of our material, homegrown material in Australia for Australian jobs using Australian products. So there's a structural problem in the industry itself. The forest is actually worth more in terms of water production, for carbon storage, tourism opportunities, regional growth and renewal intact, and we need to focus where we get the highest and best value, which is in the plantation sector for wood products. So there is a solution here. We've thought deeply about that using environmental and ecological economics, and there are ways of thinking about how to get the best return on investment for Australian taxpayers from their forests and their plantations, because we're not doing that at the moment. Yeah.
Thanks very much, David. I think everyone will agree that Dave is an extremely worthy recipient of the Macfarlane Burnet Medal and Lecture this year for 2024. Thanks, David. So I'll now hand over to Professor Malcolm Sandbridge, former Secretary of Physical Sciences, to introduce the next award lecture.
Thanks, Bob. So I'm pleased to introduce the 2024 Ruby Payne-Scott Lecture. This medal and lecture is a premier career award that recognises women researchers of the highest standing in the physical and biological sciences. It is one of the most prestigious career awards of the Academy on honours, as we know Ruby Payne-Scott's pioneering contributions to radiophysics and radioastronomy. The 2024 recipient of the Ruby Payne-Scott Medal and Lecture is Professor Kerrie Mengersen. Distinguished Professor Kerrie Mengersen's 35-year post-PhD research career has focused on the development of new statistical methodology motivated by challenging real-world applications. As a pioneer and leader of Bayesian statistics in Australia, her first 25 years focused substantially on the research and translation of Bayesian methods and computational algorithms. In the last decade, her research has expanded further to embrace new types of data and data science, with the former including digital and citizen science data, and the latter focused on the intersection between statistics, machine learning, and artificial intelligence. Professor Mengersen's work is explicitly multidisciplinary, and all of her contributions have been jointly developed with collaborators. She has maintained a career-long focus on engaging with and mentoring women in the mathematical and applied sciences, and has more recently had the pleasure and honour of working with Indigenous Australian researchers. Please welcome Kerrie to deliver the 2024 Ruby Payne-Scott Lecture.
Thank you very much for the invitation and the honour of presenting this lecture. And I pay my respects again to all of the statisticians in the Academy, and also acknowledge all of my co-authors and colleagues in the work that I'm going to be presenting. The 2020s is an amazing time to be a statistician. As statisticians, we have a remit to unlock the value of data, to develop new mathematically rigorous algorithms, methods, theory that will enable us to break open the data and to reveal the patterns and the signals within those data in order to be able to understand better the underlying processes and systems. When I had my L plates on in the mid-1980s in mathematical statistics, data was small and relatively well designed. The methods that we used were appropriate for the type of data and also the computational abilities that we had at the time. That was then, and this is now. Data now has exploded. We know that we have data from satellites, from sensors, we have data from wearables, we have data from the genome, we have unstructured data from the internet, from videos, from text, from voice. And as statisticians, it's still our remit to understand those data, the nature of those data, to harness those data, to unlock the value in those data, and to then understand how they might contribute to our understanding of the world around us. And as statistical scientists now, we see that this data is really out there. The data is the oil of the 20th century. And as I say, it's the EV of science. And as statisticians, we also need to be out there developing new ways that we can tailor and use these data in the theory, the methods, the types of inferences, and the types of algorithms. And as amazing as it is to be a statistician in the 2020s, it's even more amazing to be a Bayesian statistician. Bayesian statistics really started in the 1760s with Thomas Bayes' treatise, which was published by Price, and supported with independent support through the work of Laplace. There were two main obstacles in the Bayesian world. One was that they needed to understand the nature of probability was really a debate at the time. Was probability a subjective understanding or measure of our uncertainty or our belief in a system, or was it actually a scientific objective metric that we could measure? Also, there was the issue of, if we want to look at Bayesian statistics, we're thinking about this in terms of inverse probability. We want to understand the probability of an outcome of interest, a prediction, an effect, a measure, an association, and we want to take that probability of that outcome given the data that we have. And we get at that by looking at the likelihood of the data and the prior. And so we have two parts of that, or three parts of that. One is the likelihood of the data under the parameters in the model, and that is then that needs to be developed as a model. We also have the prior information that comes into this, and we also have the computation. And so the two big issues there were about the nature of the prior and also the computation. So in priors then, the question was, how do you set up a prior for a particular model? And so this was debated, and in fact, there was almost a war in this particular area that lasted over a couple of 100 years, as it turned out. So the issue was whether we set up a prior to be an objective prior, which means that we have the principle of indifference. In other words, any particular outcome is going to have an equally likely chance of occurring under an objective prior. And there are all kinds of different ways of formulating this objective prior. And then also thinking about, well, actually, priors can be subjective. Priors can contain information. Priors can incorporate information from previous studies or from expert knowledge, and then learning how we can set up those kinds of priors and integrate them with the data. And so while this was going on as the war, there was also the issue of computation, the computational methods to be able to deliver on or to implement the methods and the models that were developed under the Bayesian paradigm. And so the solid line, you can see, had this initial interest in the Bayesian approaches, and then a decline as this debate about priors and computation continued. In the meantime, Fisher and Neyman were bringing forth the ideas about the likelihood-based approach, which really started to take off. We had people like Jeffries in the meantime that then started to develop a really strong theory of priors, and people like Guillemin and Guillemin who started to bring forth ideas about computation borrowed from physics, and now we have, for example, new tools that are around that have acronyms like MCMC and ABC and VI, and are ways that we can do the computation for Bayesian statistics. And so that solid line then has started to increase again, and Bayesian statistics has started to become immensely popular as a way of doing and thinking about probabilistic outcomes of interest given our data. So what we see now is this idea of Bayesian statistics taking the prior information that we have and the data and combining those to give us an updated understanding of the world, and then maybe using that updated information as our prior for the next tranche of data that comes along and updating that. What we see from this then is that this is the idea that we have about learning, Bayesian learning, and in fact mimics the way that we have human learning as well, basing our information at the moment as a prior and adding new information and updating our understanding of the issues of interest. And this type of Bayesian learning, this kind of human learning, obviously underpins artificial intelligence today as well. So we find now that we have this kind of Bayesian approach, and I would like to spend a little while talking to you about how we can use this Bayesian approach in different examples. The 21st century has enabled us to bring together the statistical expertise with the expertise of people in computer science, information science, e-research, and IT under the umbrella of data science. And that's a particularly exciting area, bringing all these different disciplines together. all to find new ways to unlock the value in data. But we also see that there is this really great opportunity to work with, finally, our Aboriginal and Torres Strait Islander researchers and colleagues. And it's a really exciting journey listening to how they deal with data as the earliest and oldest people gathering data, sharing data, analysing data, and then making decisions on the basis of data. And as we take their knowledges and our Western knowledge and merge them, then we can see new ways of developing statistical methods to try to address some of the really big challenges in health and environment and society. So we see, I'd like to demonstrate the way that we work with teams to be able to bring our statistical expertise to address some of these issues. And I'll do this through 3 examples, and I'll start from the outside by explaining the application, and then peel back the layers a little bit and talk about some of the statistical issues involved in those applications. And I'll focus the 3 applications and thread through the element of trust. And so the first example is a nod to the Aboriginal and Torres Strait Islander Community Health Service that you see the logo here. AICS is the second oldest and largest community health service in Australia. And they serve thousands of clients per year in a range of medical and social services. And the people in AICS came to us and talked about developing a data and digital strategy or framework to be able to understand their clients better and to be also able to give data to their clients that their clients could use to increase agency. And there's a lot of talk about agency through data and data literacy. And so agency here was about understanding the data that they were provided, that communities and Aboriginal people were provided, and to be able to make decisions on the basis of those data. And this kind of agency then provides hope for the communities. And so this is not really a scientific problem for the Academy, but what it does do is it illustrates the outcomes of our science for communities. AICS has done a fantastic job in implementing their data and data literacy strategy, and they've become leaders in the country for Indigenous community groups and the broader community groups as well. So peeling the layer back on what this collaboration has provided and what it means for statistical science is the issue of dealing with, one of the issues is dealing with private data. And so if we think about the issue of trust, we have the question about trust in data, the way that we would use data. And so we ask the question, how do we deal with people's data and private data? And one of the ways that we have been looking at doing this from a statistical perspective is through the idea of federated learning. So instead of trying to create the one database to include all of the information, including the private information from different silos, instead of trying to bring those data together, we can leave the data where they are and then bring together the aspects of the information that we need to create our models and do the analysis. So the data stay with the data custodians and we can do the analysis in the cloud. So this is a statistical question because it asks us to think about the way that we construct our statistical models to take advantage of this kind of architecture. And if we can do this, then it means that we can avoid a lot of the privacy issues and the delays in accessing data, which would then mean really a revolution in the way that we do analysis in the areas of health, in the areas of business, and in the areas of social systems. And so this is a question, a statistical question then, about how we actually construct the models and the algorithms to take advantage of this type of federated learning approach. The second example that I'd like to talk about is our Virtual Reef Diver project. Now, this is an environmental project. We work very closely with a number of organisations like the Australian Institute for Marine Sciences, and in this particular challenge was about the conservation of the Great Barrier Reef. Now, the Great Barrier Reef is 2,300 kilometres long. You can see it from space. There's lots of great modelling and monitoring that's done for the reef. Monitoring stations on the reef that have 20 years of data that have been carefully curated by groups like the Australian Institute for Marine Sciences. But the reef is so long and these sites are intermittent. So how do we know about what's happening in between those particular structured samples? Well, there's any number of people with their cameras diving on the reef. What if we built a virtual reef? What if we then took that information, those images from the divers, and we posted them onto the reef? And what if we then allowed citizen scientists to inspect those photos and annotate for coral, algae, sand? And if we took that information from the back end out and then combined it with our models, and so with the data that we have from the long-term monitoring. And so this is a lot of hand-waving, of course, but we structured this as a product, a platform, and we've had a lot of interest in acquiring photos and then annotations for those photos from around the world. So this kind of citizen science then invites a statistical question about how we actually deal with those data, how we analyse them and how we combine them through the architecture of Bayesian analysis with the long-term monitoring data. And so it's more than just a website. We have the images, we then do the classification, we extract the information, we combine it with professional data, we do the modelling, we get improved predictions for across the whole range of the reef. We also have to learn about trust in this particular issue, in this particular study as well. In the previous study, we talked about trust in the data. Here we're talking about the trusted analysis of the data as well. And so if we just look at how well we can predict with the long-term monitoring data alone, we go somewhere. But if we can use the citizen science data and we take account of the aspects of citizen science data that we know are involved with, for example, citizens have different skill sets. Citizens, how clean is their data? How much can we trust it? If we have the statistical methodology to understand or identify those citizens who have better skills and also adjust for their skills and the difficulty of the images that they might be annotating, then we can start to clean up that that citizen science data and increase the trust in the data. And if we do that and we include it with the long-term monitoring data, then we actually start to get much better predictions of the reef. So an ideal prediction would be where an ideal model would be where our observed data matches our predicted data, and we have just everything living, all the dots on the diagonal line. But you can see here that the inclusion of the citizen science data, this type of new data, unstructured data, can really help us to improve our analyses. The third example that I'd like to talk through is our Australian Cancer Atlas. Now, this is work with Cancer Council Queensland and a number of other agencies. And Cancer Council Queensland talked with us about 10 years ago to say, what are the patterns of cancer across Australia? What's the incidence and survival rate in different types of cancers? And how can we find patterns and signals in those rates? At the time, the data were siloed in the different states where the cancer registries are located. And so we brought those data together and we could map the data themselves, but we needed to be careful, of course, because it's private data. And so instead we created a Bayesian statistical model that would enable us to produce estimates of the cancer incidence and survival rates for small areas across Australia, about 2,000 small areas, taking into account prior information in this sense around for every small area, the areas around it are going to give some information for a particular area. And so that the information in those surrounding areas can be thought about as the prior information in this case. And so coming up with a Bayesian spatial model that would then allow us to estimate incidence and survival, have a probability of being above the Australian average, and compare different groups or different areas across the state. What we needed to do in this was also to give some ideas about uncertainty and some estimates of uncertainty and visualisations of it as well. Peeling back the layers on this, the statistical issues were not only the model that we created, but also trying to understand how we could create some trusted insights from the data as well. So for example, can we come up with areas that are similar, so clusters of areas in terms of risk or survival? And these kinds of clusters then require us to understand about clustering methods. And one of the underpinning methods is the idea of mixture models. And so these mixture models then give us the subpopulations or subgroups in a particular population, in this case, the regions here. And what we see from this is that we have to be able to understand how many components there are and the nature of those components. And that turns out to be a deep statistical question. We can learn from these data also about spatial inequalities. People who live in the city and live in the bush have different rates of survival from different kinds of incidence and survival from different kinds of cancers. In fact, people who live in the bush tend to have poorer survival than people who live in the city, and we can quantify how many lives could be saved if we didn't have that kind of spatial inequality. And in fact, that has then generated a whole lot of media attention and led to changes in policy for people who live in the bush to access treatment in the city. It's also, when we launched the last version of the Atlas in about a month ago, I talked about the word hope, how we do this. What are the outcomes? What are the partnerships? And what are the extensions? So this word hope is just a small word, but it really sort of translates across all of the studies that I've talked about and the other work that I do in terms of hoping that our small contributions in statistical science can make a real difference in our own area of statistics and mathematical statistics, but also in these much larger challenges that affect us in the world. And so I'd like to finish with the statement that we've come a long way. Alan Turing in 1947 said, what we want is a machine that can learn from experience. I think that statistical science has played a role in that and will continue to play a role in developing such a machine and such techniques for our world, but we still have a long way to go. Thank you.
Thank you, Kerrie, and lovely to see a picture there of Sir Harold Jeffreys, who the seismologist will also claim as one of the founders of modern global seismology. So we can open the floor to a few questions. Bob. Oh, wait, Bob, get the down here, the microphone.
Thanks very much. Thanks, Kerrie. Lovely lecture. I just didn't follow when you were talking about trusted data and you said you leave the data where it is and you only take up that one that informs whatever you're analysing. But I'm thinking in terms of our own work. in genome work, and we would take up then the particular data that, say, give you a GWAS hit, it still would potentially be a problem with privacy because you could still determine from that who the person is. How do you get around that?
Yeah, thank you. So, so this area is actually being, so federated learning is actually being considered for genomic analysis. And so when we say we take up the data, we're actually taking up some summaries of the data or some aspects of some elements of the information in that data set that's required for the analysis. So if you think about a model that you have and the kind of information that you need, you might just need the average, or you might just need the covariance structure, or you might just need some, X prime X bit of it. So that's what you ask for. And so then you work out how you can combine all of those bits that you need to be able to do the analysis as a whole.
Do you have a microphone? Oh, you do, yeah.
We've got one question from online, from Bernadette from the QTT Centre for Data Science. How can Australian-based researchers contribute to the current debate on safe and responsible generative AI that is poised to transform data-driven research?
Nice easy one for you.
Thank you. Thank you, Bernadette. So generative AI, as we know, is just taken the world by storm. It's one of the new forms of data that we need to really embrace, I think. And it's more than an algorithm to search across the web and bring information together. We're also able to now harness that and to use it and to understand whether we trust it, how we, what's the issue for like hallucinations in this, how we can incorporate this kind of information with our scientific information as well and scientific knowledge. So I think there's a lot that needs to happen with the understanding of the generative AI algorithm and also the information that we get from that and then how we use that for scientific discovery. So, and I think Australians can play a really big part in that. I think all scientists can play a really big part in that, but we really need to sort of embrace it as a new tool that we have.
Maybe we'll go for a final question down here, please. The microphone's making its way to you.
Thank you very much, Kerrie. Fantastic. One of the words you used was trust. And trust has become a sort of a nasty word in our society. Your work obviously needs people to believe you. In your research and your steps ahead, are you looking at how to inform people that they believe what you're doing and the results are accurate to help gain their trust to understand what you're doing?
Yeah, thank you for that question. I really think that we as scientists, we collectively, and the Academy is one place where this is really important, it's been talked about at the meeting a number of times, is that we provide a trusted voice. And so what I've been talking about here is how we can use mathematical methods and rigorous approaches to be able to work with these data and to be able to understand the nature of the data and then to be able to develop models and methods that use those data to describe systems and processes that we're interested in. I think that in order as to whether people believe or trust my approaches or what I'm doing, I'm a Bayesian, so I have a very strong belief that they will be quite happy with what I do. But also, I need data to be able to confirm that as well. And so, it's interesting to see. We have some doubts when we work in groups and teams, and then that's how you sort of then reconcile my understanding and other people's understanding of the world.
I think that was a lovely closing of the loop answer there, back to the prior. Thank you, and thank you for a lovely lecture. Let's congratulate Kerrie again. And we'll move on to the next part of the programme, and I'll hand over to Professor Julian Gale.
Thank you, Malcolm. We now have the award presentations to and from the other 2024 awardees. The David Craig Medal is awarded in honour of the outstanding contribution to chemical research of the late Emeritus Professor David Craig. It recognises contributions of a high order to any branch of chemistry by active researchers, and the recipient of this medal delivers several public lectures across Australia. The 2024 recipient of the David Craig Medal is Professor Justin Gooding. Professor Justin Gooding is an international leader in the field of surface chemistry. In particular, he is renowned as a leading authority in the modification of surfaces for the development of better sensing devices. Characterised by using molecular to nanoscale control, his science systematically addresses fundamental questions in electrochemistry and biology, as well as general challenges facing many sensors and analytical devices. He's made outstanding contributions to fundamental and applied research using self-assembled monolayers to fabricate molecular scale constructs on surfaces that provided new measurement tools. Professor Gooding's work has shown not only how to design and fabricate sophisticated surface architecture for sensing, but he has also changed thinking on both the level of control that is possible and the types of information that can be acquired using that control. Please welcome Justin to accept the medal and deliver the 2024 David Craig Medal Awardee presentation.
Thank you, Julian, and firstly I'd like to thank the Academy and the people that nominated me and also the people that chose. This is an incredible honour. I actually know what every single David Craig medalist has done when you look through the list, and so to be put in such illustrious company is quite an honour. I'm really going to talk to you about single molecule thick films. I'm a chemist. What chemists do is we typically create new molecules or new capabilities with the molecules that other people create. And I'm going to talk to you about how we can, some of that really basic science, but how it can go all the way through to a translation of a device. And so before I do that, I just wanted to acknowledge people that give us the funding, which is the Australian Research Council, National Health and Medical Research Council, and my home university, the University of New South Wales, plus a variety of other organisations. I wanted to thank the people in this figure, in the picture, which is the last Christmas party of the group, but when you start to break that down over the years, that comes down to an awful lot of people, and I get to be the dog and pony. They're the main show. They're the people that really do the hard work. All those people listed there are the bench researchers or our collaborators. I also wanted to acknowledge my families. Some of you may know my late wife, Katerina Gauss, Gottschalk Medal winner. Today would have been her 52nd birthday. But I'd also like to thank my wife, her innate love and support and goodness and belief that we should do good things because we have that opportunity, help rekindle the drive that you need to do cutting edge science, to do those things. So thank you. So what we do is we modify surfaces with a high degree of control. All those people you saw do that one thing. Sounds boring, right? No. What we do is we use single molecule thick films, sort of shown here in the, if I can find where my mouse is. These single molecule thick films, shown in this little red circle, we modify a surface with those. Why we do that is because you might see some of the molecules have a red dot at the end, representing a nitrogen atom, and a blue dot representing an oxygen atom. And what we basically can do is by taking a single molecule thick film and putting different chemistries within that, we can modify a surface with molecular level control. But because it's only a single molecule thick, we can exploit all the properties of that underlying surface, whether it's optical, electrochemical, but we can change how the outside world sees that surface. So what we actually do is then apply that mostly towards applications where that spatial control or the control of the surface, that molecular level control, allows us to make new measurement tools. Often we then hand off those measurement tools to the biologists or the people that do a whole lot of discovery science, or we move it towards a technology if the ideas are good enough to allow us to generate new devices. And so a lot of people think we make sensing devices, but what we really do is just that one basic thing, ask the question, how much control can we have? And what, with molecular constructs on surfaces, what new measurement tools can we make? And most of those, not the ones shown in the slide anyway, have actually made transitions towards commercial outcomes. In fact, the question mark, in a week's time, we'd start looking at using this new technology for looking at glucose in neonatal children, because if you accurately measure their glucose levels when they're young, when they're just born, you in fact impact an entire life. But basically we move towards applications. But I want to make this fundamental, or this really important point, something that bothers me in science, is we're trying to divide science into fundamental research and applied research. I just think it's research that answers really good questions. And if those good questions are new questions, you're going to have to do some really basic science to start answering them. And that's what I'd like to think we do. So what we're known for is measurement devices, biosensing devices. They really exemplify the importance of what you can do. So a glucose sensor, this is one of many on the market. It was actually my first PhD student's company. It's actually the first company to integrate a sensor with an iPhone and sell it in Apple stores. But really, it's an interfacial device. What a sensor is, a biosensor, is a solid surface with a piece of biology on that surface, so we can stick that surface into a complex solution such as blood, and rather than having to do any complicated analysis, we can just measure the concentration of something. And in the case of glucose, that single device, that class of devices, not that particular one, is the most successful and most used analytical device in the world. There are 1.5 billion glucose measurements done every single day. It's 10 times more than any other analytical device in the world, which the second would be the pH meter. So it can actually revolutionise lives. And of course, the other device you see is the lateral flow device. We used to think of it as a pregnancy test kit. Now we think of it as a rat test, because you've all used one, and it's now just taking a different piece of biology, putting it on a solid surface, and allowing us to do a measurement directly up your nose, we all love that, or wherever, and to allow you to do a measurement. So they're all interfacial devices, so how you design the surface is important. So we talk about these single molecule thick films. They're called self-assembled monolayers, because if you have the right conditions, a single layer of molecules will spontaneously form on a surface. And as I said, and it does that because the head group of that molecule loves the surface. The tails then order and form essentially, in some cases, a two-dimensional crystal. And so basically you've got these constructs of the end group determines how the outside world sees it, the head group determines how stable it is on the surface, and the carbon chains in between determine its order. And we can put different components into these films. You can have all sorts of different head groups, and one of the uniquenesses in our group is that we've developed, maybe not the actual coupling chemistries, but the learnings on how to form really good layers on a whole lot of different surfaces, from gold to indium tin oxide, glass, silicon, so semiconductors, and carbon. And so we can basically use all sorts of different chemistries for different applications. The principles are the same. A head group attaching to the surface, end group determines how the outside world sees it. And so to give you a feeling of the control we have, we designed molecules. There's actually a former David Craig medalist, Michael Padden-Rowe, one of the pioneers of electron transfer theory. We designed these molecules that have got 2 feet, so they're completely rigid. And then they're L-shaped. And the end of this molecule is actually a redox active species, so you can do electrochemistry to it. And by diluting that across the surface, you can imagine by changing the 2 components, you can control them laterally, but actually these molecules are rigid, so we can control them vertically off the surface. And we can space this head group, this electroactive group, above the surface at about 0.05 nanometer spacing. So incredible control if that flat surface is a single crystal. And we can show how the electrochemistry changes. So it's very basic science. And you might ask, well, who cares? Why do we want to do this? Well, because with the advent of monolayer chemistries and sensors, they're nearly all electrochemical, and they've nearly always got an electrochemical species near the surface, and that's going to determine how well the device works. And just to give you one example of what's possibly the next game changer if we pull off the technology, it's a method actually developed not by myself, but by a group in UC California, Santa Barbara, by a guy called Kevin Plaxco. He developed an idea where you have a sequence of DNA, and that sequence of DNA acts like an antibody and it binds to a small molecule. And when it does that, it changes shape. And by changing shape, you change the electrochemistry of that molecule. And they couldn't do that if they didn't understand how that electrochemistry would change. So that basic science goes all the way through to an application. But why this technology is absolutely game-changing, and it's being developed by a Melbourne company called Neutromics, who I work with, is because these molecules can do things reversibly, so they can bind reversibly. And so we can actually continuously monitor in the body in real time. So it's quite game-changing, because what the first product is to monitor a drug, put a patch on your arm, monitor a drug in your body in real time. And I'm massively running out of time. I was going to tell you a second example where we use surface chemistry to understand where cells adhere and how they find places on a surface. We use, again, monolayer chemistry, but where that extends is we then now make polymer systems where we have these adhesive molecules that bind to cells. The rest is a cross-linking system that we can then 3D print, where we've made a 3D bioprinter with a company in Sydney called Inventia that allows you to make cell cultures, 3D cell cultures in high throughput. Why is this important? Because producing 3D cell cultures, we are 3D, there are some features we can't mimic in a 2D microscope slide. Making 3D cell cultures allows us, takes a very manual process that cell biologists have to do and make it very automated. And so we can print these cups that you saw, print the cells in the middle, wait 2 days, and you have what's called a spheroid or an artificial cancer. And rather than make one every few days, we can make about 30 in 30 minutes, 96 in 30 minutes. So a microtiter plate well, all controlled size. The polymer system, we can control the environment. So it allows biologists to ask really basic questions about, well, how does the extracellular matrix influence the system, and so on. And I'm getting the push along. What we're now doing is probably even nicer work. Well, I think it's super cool. Artificial enzymes, detecting just a very few molecules for the liquid biopsy, which is understanding and monitoring cancer. And now we're developing methods of monitoring single molecules, all single molecules, quantitatively on a surface in high throughput. And with that, I'll thank you for taking the time, and I'd like to thank the Academy once again for this incredible honour. Thank you.
Thank you, Justin. I don't think anyone would accuse you of doing boring work. I'd say it's inspirational and amazing.
No, thank you.
We now have time for a short question and answer session. Let's see. Oh, there, please.
I was curious about how robust the single monolayers were. Can you engineer them so that they're not disposable?
Yeah, that's a really good question. So the reason we have 5 different systems that we work with, because say the ones on gold, we can put them in the body and they last for about a week, maybe 3 days to a week. The ones we do on silicon, we can throw them into hot boiling acids and bases, and they don't degrade. But gold is a super stable material, so it's only the monolayer chemistry we worry about. Silicon is an unstable material because it's silicon without any oxide layer. So the chemistries, it's a balance between the stability of the chemistries and the stability of the substrate. We still don't have the perfect monolayer system.
Francis?
Lovely. Could you explain to me how those wearable sensors work? How do they detect changes?
Yeah, putting a career into 10 minutes is not so easy. They've got a micro patch. So it's a micro patch with microneedles, so it goes and goes through the skin into the interstitial fluid. And so on the end of each one of those microneedles is an electrode with a sequence of DNA, an aptamer. So imagine my fist is the radioactive species. When the molecule binds, it flops down, and now we get more electrochemistry. When the molecule lets go, it comes back up again. And that's just sitting in interstitial fluid. The big challenge is, in fact, not doing the measurement, but relating the measurement to the blood levels. So with some molecules we know that, but in other molecules we don't yet know that. That's going to be probably the main challenge for the company, is actually calibration. We've already done it on people, we can already measure in people.
Okay, we have time for probably one more quick question.
Okay, just a very good collection of the work around the surface molecule. You only show us a little video about your recent Nature Photonics work, and my question is, how does your single layer of molecule contributing to the single molecule detection in photonics?
Yeah, so the innovation in the Nature Photonics paper is that we use electrochemistry rather than lasers to switch the fluorophores to do this super resolution light microscopy. And why that's important is it actually allows us to get quantitative switching, so we don't lose 2/3 of the molecules when we do that. But what that then allows us to do is actually count and look at surfaces under single molecules with single molecule resolution. So what I told you that, we have molecular level control over how we design these things and so forth, and I showed you lots of cartoons, I can't verify that's true. And so the new super resolution approach will then allow us to look at surfaces, design surfaces, and actually characterise them at the molecular level that we design them. That's why it's so important for us.
So I apologise that we have to leave it there, but please join me in thanking Justin for a truly worthy. Thank you. Congratulations. I'd now like to hand over to Professor Glenda Halliday to introduce the next awardee.
Next, to the Susan Cory Medal for 2024. This award honours the contributions made to science by molecular biologist Professor Susan Cory and recognises outstanding research in all of the biological sciences, being awarded in alternate years in the biomedical sciences and in all of the biological sciences, excluding biomedical sciences. The 2024 recipient of the Susan Cory Medal is Professor Peter Koopman. Somewhere? Sorry. Professor Peter Koopman's research focuses on how genes function as the blueprint of embryonic development. He is best known for his role in discovering the Y chromosomal sex-determining gene, SRY, widely acknowledged as a milestone in 20th century genetics. His subsequent work has exposed the molecular, genetic and cellular pathways by which the gonads form in the embryo, addressing the essential question of how males and females come to be. In parallel, Professor Koopman discovered several SRY-related SOX genes, including those that act as master regulators of skeletal and vascular development. These achievements have had broad and enduring impact in developmental biology, medical genetics and reproductive biology. Please welcome Peter to accept the medal and deliver the 2024 Susan Cory Awardee presentation.
Thank you so much for that kind introduction. I'm going to ask you, in a somewhat unusual step now, to completely lose yourself in this picture that you see above. And it's obviously a human embryo. The human body contains something like 50 billion cells. And during embryonic development, every single one of those cells has to be told what to become, all of the cells have to migrate to exactly the right position, group together with other like-minded cells to form functional organs. All of this needs to be done according to a very tightly orchestrated schedule. And so the focus of my life's research has been the question of where are the instructions for all of this? Who's reading the instructions? Who's orchestrating this fantastic symphony of embryonic development? And these questions are all the more remarkable when you think that unlike some of the very sophisticated structures, other structures that we have in our lives, jumbo jets or supercomputers or whatever, the embryo is constructing itself. And obviously the question is, how does that occur? Now, this is a galaxy-sized question, and to try to address this as a scientist, we obviously try to break it down into smaller chunks. And the chunk that I chose to focus on early in my career was the question of sex development. How is it that during embryonic development, the embryo gets to a certain fork in the road and then has to decide whether to turn left to follow the pathway of male development, or instead to turn right to follow a completely different pathway of anatomical development, that of female development. Now, Jenny Graves has kindly introduced this subject to you this morning, so I don't need to belabour it, but you know obviously that it's a function of whether your cells possess or don't possess a Y chromosome. And you see that on this slide. Now, the Y chromosome always receives a bit of flack because it's a small chromosome, it's rather puny, but I'd like to speak briefly in its defence. It's small and puny because it's thrown away a lot of the stuff that it doesn't need. What it really needs is a gene or genes that determine male sex. And so it retains this gene or gene, and that's its most important function. And so when I was a postdoc in London, I was a member of a biological experimental group, and we decided to try to find the sex-determining gene on the Y chromosome. And the way that we did this was to team with a human genetics group at the Imperial Cancer Research Fund under the direction of Peter Goodfellow. And the two groups worked together to try to find the sex-determining gene from the Y chromosome. And the way that we worked it was that the human genetics group would comb through Y chromosomal sequences to generate candidate genes, which they would then pass to us, which we could then test in a mouse experimental system as to whether they were or weren't the correct gene. So the human genetics group passed to us a chunk of the Y chromosome containing a gene which we called SRY, sex-determining region Y gene. They passed us this gene. We did all the tests in mice. We found that the gene was in the right place on the Y chromosome to be sex-determining. We found that the gene was active during gonadal development, which suggested that it was probably had a role in testis development and therefore in male sex determination. But all of this evidence was very circumstantial. We had to come up with an experiment that proved that SRY had a male sex-determining role. The experiment that we devised was hopelessly ambitious and almost certainly destined to failure. We took a solution of DNA containing this SRY gene. We had no idea whether we had all the parts of the gene, the enhancers, the promoter, the introns, exons, and so on. We injected this solution into fertilised mouse eggs. We had no idea whether the DNA would integrate into the egg's chromosomes and function. But the idea was we were hoping that injecting the SRY gene would change the course of development of XX mouse eggs towards a male fate. So we injected the eggs, we re-implanted them into foster mothers, and we anxiously waited for the birth of the mice to see if they were male or female. And miraculously, the injection of this gene was able to change the sex of the live-born mice to a male fate. Male mice were born. So this remarkable experiment told us some very important things. Firstly, that SRY is the only gene required from the Y chromosome required to make a male. and there are other things that are told us, but I've forgotten what they are. So we had proven that SRY was the male sex determining gene. This answered many thousands of years of speculation about the essence of the difference between males and females. But as a developmental biologist, we were very fortunate that the discovery of SRY gave us so much more. The bycatch of discovering SRY was very rich indeed, and the bycatch was that SRY turned out to be a member of a much larger family of structurally related genes called SOX genes. And we started fishing out these SOX genes and asking whether they might also have important roles in embryonic development. And of course they do, otherwise I wouldn't be standing here. One of the first genes that we pulled out was an SRY-related gene called SOX9. And when we did this test that you see in this slide, it's called in situ hybridization, what it does is light up the parts of the embryo in which a particular gene is active. It lights that up in blue. And clearly you can see that all of the developing skeletal elements of the embryo have turned blue here, meaning that SOX9 somehow is intimately associated with skeletal development. Again, we asked the very audacious question of could SOX9 be the master regulator, the gene right at the top of the regulatory hierarchy that results in the pathway of skeletal development? And the answer is yes, it is. SOX9 is capable of instructing undifferentiated mesenchymal cells in the embryo to take on the fate of chondrocytes, differentiate as chondrocytes, and of course that's the first step in the development of the skeleton. Other groups, human genetics groups, found that SOX9 is defective in a certain type of patient, some children with a rare genetic disease called campomelic dysplasia. And that's a disease that results in skeletal malformations. So SOX9 is a gene that also has important functions in human disease as well as development. And so this was a second example that we were able to identify of a master regulatory switch gene for the specification of cell type in embryonic development. And a third example that I'd like to talk about is yet another structurally related gene, a gene that we called SOX18. And once again, when we did this type of in situ hybridization experiment, we were able to find that SOX18 is active during the development of all of the blood vessels in the body. It's a gene that's required for the specification of endothelial cells, and it turns out to be a master switch gene for endothelial cell specification. Once again, other groups determined that structural defects in the SOX18 gene are causative for vascular anomalies in a certain class of human patient. SOX18 has a whole other story attached to it, which is that it's also a master regulatory specifier gene for a related cell type, which is that of the lymphatic endothelium. And that work was done by a postdoc in my lab, Matt Francois, who's now a professor at the University of Sydney, and I believe is here in the audience. Now, Matt found that SOX18 is critical for the specification of the lymphatic endothelium, and of course, the lymphatic vascular network is clearly very, very important in terms of human health and disease. Now, even more fascinating, or arguably even more fascinating, is when you think about the potential role of these genes in cancer. When you have cancer that is developing as a solid tumour, the tumour growth is very critically dependent on the development of a blood vascular system that can supply oxygen and nutrients to the tumour. In addition, the cancer is also very dependent on the development of lymphatic vessels into the tumour, which supply an exit route for cancer cells in the process of metastasis. And it's the metastasis that is the main contributor to the morbidity of cancer. So we had a gene that's responsible for both the vascularisation of tumours and the development of lymphatic vessels in tumours, and we figured that if you could possibly inhibit SOX18, you may be able to stem the growth and spread of cancer. And there is now a startup company in Melbourne that are exploring the possibility, the proven principle, based on the proven principle from work principally of Matt Francois, that you can chemically inhibit SOX18 to inhibit vascular development. I'm getting the wind up, but I want to briefly say that as a young student in the Department of Genetics, we used to go across to the Walter and Eliza Hall Institute and sneak into their seminars, and some of these seminars were given by Suzanne, Cory and Jerry Adams, who are in the audience, and they were talking about the emerging field of molecular biology, and I just thought I really have to have a piece of this action. I would love to do this sort of work. So it's certainly a great honour all these years later to be the recipient of the Suzanne Cory Award. Thank you very much to the Academy for that. And I'd like finally to acknowledge my family, without whom I wouldn't be able to have developed as a scientist with all the work that it entails, the time and commitment, and all of the postdocs and students that have contributed to my ultimate success. Thank you very much.
Thank you, Peter. The SRY, I think, has enormous implications. It's even somebody who hasn't really been delving into that in our neurodegenerative diseases, it's made a difference. So it's an amazing discovery, so well done. We can have some questions. While that's coming, I was going to ask, because you've got SOX9 and SOX18, and so there's how many SOX, and how many have you been studying?
There's 20 SOX genes, and honestly, this gene family is like the holy grail for developmental biologists. They are in charge of all sorts of extremely important decisions in terms of cell fate specification during embryonic development, and they are involved in many different types of human disorders. So it's really been a very, very rich payload for us.
Thank you, Peter. A fascinating subject. It affects all of us, I think, in different ways. I think you've answered the question. There are about 20, did I hear you say, SOX that may indicate some influence on the development of the body. Are any of these known to develop or change development in the brain? You've been very fundamental in the ones you've mentioned. I'm interested in whether there is a possibility of influences on the brain.
Definitely a possibility. Some are known to affect neural cell differentiation, but I would say there hasn't been nearly enough research in that area, but I think the answer to that is yes.
Can I ask a question? Upstairs.
Yes, yep.
Yeah. Thank you for the very nice talk. And what I'm going to ask is actually inspired from what I learned in the morning in the Sex and Dragons talk. And forgive me if the question is silly. So when you described the experiment that led you to confirm that it is the SRY gene that determined the male or female, I'm just wondering, how did you rule out a possible role of environment? Did you repeat the experiment for a sample of mouse at different conditions, and that's how you did it?
I'm not sure I fully understand the question. We certainly repeated the experiment many times and got the same result. Is that what you were asking?
Yeah, you actually answered the question. So I was wondering, how did you rule out any role of environment? Maybe that's not relevant for mouse, but I'm just curious.
Well, I mean, to us it was pretty straightforward. We were taking mouse eggs that had an XX chromosomal constitution, and those ones, if we implanted those, would never develop as males, and only the ones that we injected with the SRY gene changed their sex to that of male. So we took that as straightforward proof.
Okay, thanks.
Okay. Please, can we actually thank Peter again for a fantastic lecture, and congratulations on having him. I'll now pass back to Julian.
Thank you, Glenda. Next is the Nancy Millis Medal which honours the contributions made to science by the late Professor Nancy Millis and recognises her importance as a role model for women aspiring to be research leaders. The award is open to women, mid-career researchers in any branch of the physical and biological sciences. Recipients will have established an independent research program and demonstrated exceptional leadership. The 2024 recipient of the Nancy Millis Medal is Professor Anita Ho-Baillie. Professor Anita Ho-Baillie is a pioneer in developing next-generation solar cells, which will play a key role in the transition to a carbon-free economy. Professor Ho-Baillie's research centres on multi-junction solar cells, utilising a range of semiconductor materials to absorb different sections of the solar spectrum within a single cell, significantly enhancing energy conversion efficiency. Beyond research, Professor Ho-Baillie is deeply committed to inspiring young minds in STEM fields and science communication. Please welcome Anita to accept the medal and deliver the 2024 Nancy Millis Awardee presentation.
Good afternoon, everyone. Thank you so much to the Academy for awarding me the medal. So I'll charge along because there are quite a number of slides that we've got to go through. So how much photovoltaics are we going to have? According to the International Renewable Energy Agency, we need 5 terawatts of PV in 2030 and 14 terawatts of 2050 to reach keep the global warming to 1.5 degrees Celsius. So thanks to Andrew and Kylie and you setting the scene that we're going to be needing a lot of photovoltaics. So there are people who are experts in the field and they reckon we need even a lot more. So we need around 30 terawatts of photovoltaics in 2040 or 75 terawatts of photovoltaics in 2050. So 1 terawatt is 1 trillion watts, and that's 1 million times million watts of power. So are we going to get that? Possibly. So now there are 10 gigawatts factory being rolled out and 5 gigawatts of solar farm being rolled out. So to get to those levels of PV, we need 1 terawatt of PV a year. So that's hundreds of those factories and hundreds of those solar farms being rolled out every year. So that's quite a lot of solar modules that we're going to make, and that's quite material and energy intensive. It takes a week to purify, quartzite to metallurgical silicon, polysilicon, and we haven't even talked about how to make solar cells yet. So it's very important that we improve the power conversion efficiencies of cells and modules. So when we do that, we do 2 things. We can reduce the lifetime cost of solar system, provided they last as long, of course. And also when we improve the power conversion efficiency of the modules, you get more power out of the same material. So you're eliminating waste and you're eliminating pollution at the design and manufacturing level. So in a way, you actually improve the circularity of the product. Okay, so Solar 101, what do we need to get a solar cell to work? Well, we need the sun. That is made up of light, different color light at different energy levels, different photon energy, and is expressed in electron volts. So for example, blue light will be 2.6, and then you can move down 2.4, 2.2 across the rainbow. And to harness that solar energy to electrical energy, you need a semiconductor, which is usually insulator when you don't bother it. But if you give it energy, then the electrons will become mobile and the material becomes conducting. So the way to look at it is you can look at an electron usually sitting in a valence band, whereas in mobile, so it's a continual energy levels, and it's separated with a band gap. So there's a conduction band, valence band, and a band gap, which is what I always use the word band gap for a semiconductor material. So when the material sees light with photon energy that is bigger than the band gap, it will excite the electrons and make the material conducting. And when the material sees photon with energy lower than the band gap, the photons just don't get absorbed and just pass through to the next layer or whatever is underneath, and nothing happens. But if you have photons with energy way above the band gap, it will still excite the electrons, but you can see there's a waste of energy there. So we call it the thermalization loss. So if you have material that is made out of one material with a fixed band gap, you will get the thermalization loss and the sub-band gap loss. And that's why single junction solar cells got a theoretical limit of 33%, and for silicon is around 30%. So 29.4 or 29.6, depending on your calculation. And then with the double junction, you stack the the high band gap material on top of the low band gap material, so that the high band gap material will be more efficient in the way in convert the high energy photon, and then let the low energy photon pass through, and then you get the theoretical efficiency limit jumping up to 45%. And when you have a triple junction solar cell, you get 51% as the theoretical limit. So as you increase the number of junctions, unfortunately, the improvement gets incremental. So when you get up to 6 junctions, you actually don't necessarily get 60% theoretical efficiency limit. Anyhow, what sort of materials are we looking at? So in our lab, in a lot of labs, we look at metal halide perovskites. So this material has been talked about, and Nangzhou talked about it yesterday as well. Why is it so good? It's got a lot of photovoltaic enabling properties. So it's got very high absorption coefficients. So for silicon cell, you need 100 microns thick, which is as thick as a piece of human hair. And for perovskite, you only need 1 micron or less. So it's 100 times thinner. It's got good charge mobility, and is a compound semiconductor. That means we can change the composition and therefore tune the band gap. So you can make it a high band gap perovskites or low band gap perovskites. You can also stack the perovskite onto other cells because it's a thin film. So it's like a coating that you can coat it onto another cell, and it's quite easy to make actually. So tandem, so what does the industry think? Well, the industry think that some form of tandem in the form of silicon, maybe perovskites, silicon tandem, will come into the market post-2026. So how do you make tandem cells? Well, you can directly fabricate a cell on top of another, or you can just have 2 individual cells and mechanically stack on top of each other, and then you get these electrical leads coming out. So for the 2 junction, you will get 4 terminal, and then if you directly fabricate, you can just get a positive and a negative terminal out of 1 single cell. You can even split the spectrum using lenses and mirrors, and you can achieve tandem or multi-junction that way. Now for triple junction, it gets more complicated if you mechanically stack it. And then for spectrum splitting, I don't know how to draw it actually, so I just say don't want to know. So that's why in our lab we concentrate on the monolithic tandem, and we use perovskite and stack it on different kinds of materials, and we get 3 junction and 2 junction, and sometimes we make flexible cells as well. So it sounds easy, but it's actually not that easy. So every time you introduce a junction, you actually introduce 5 extra layers. So if it's triple junction and they're all perovskite, you've got 15 layers of materials that you have to fabricate. And we have people staying up till 10 p.m. at night. So you actually have to put 2 people working on triple junction. So this is a silicon perovskite tandem, turning a fan outside Hot Watts, which is University of Sydney. And you can see that over the years, our solar cell efficiency has improved, so is the quality of our graphics. You can see that the triple junction, we only just started, so it's still a 2D sort of more boring graphics, so we'll improve on that. And what do we do? So those materials are really, really thin, so they're nanometer layer thick. So we look at the non-classical behavior of that carriers being transported between the junctions. And theoretically, we predicted that the behavior will be nonlinear and also you get the oscillating behavior. But, how do I go back? Yeah, but experimentally, we haven't quite get there yet. So, it is my dream to be able to make those layers with sub-nanometer resolution to be able to observe those behaviors. And that will help us to really optimize the solar cell device design. Okay, so in our lab, we claim we can make really efficient cell, but are we? So what we do is we send our cells to an independent lab. They are very good at measuring cells and very careful with your solar cell. And we send it to the American National Renewable Energy Laboratory. And recently we got a 30% efficient solar cell, which is great because you already bypassed the limit of the silicon cell theoretical limit. And what is also encouraging is that the cell measure NREL is higher than what we measure. So what it's saying is when we say we got a 28% cell measured in our lab, we are not over-reporting it at all. So I tell the students you could possibly sitting on a 30% cell. So, hang on. And we got fierce competition around the world. So when we got it certified, there were only 8 groups in the world that joined the 30% club, and now there are 10 groups. So 3 companies and 7 research institutes joining the club. And oh wow, how much have I got? Only 30 seconds. Okay, so, and there are 49 research institutions working on this research area. So the technology has got to be durable, it's got to last long. So what we did is we use GCMS, gas chromatography and mass spectrometry to look at the cells, the outgassing of the materials out of the cells when we thermally torture them. So people didn't sort of think that this can be a way of characterizing perovskite cells, but nevertheless we did. And then we look at use GCMS to look at different designs, to look at different packaging method, and we came up with a method that can sort of reverse the reversible reaction and stop the outgassing, and therefore we can stabilize the cell to pass 3 industry standards. And that was like great news that we published in Science in 2020, and we transfer the learnings to tandems. Also, we're sending our cells to space. Why? Because they need 1 million square meter of space, cells in space. There's no water, there's no oxygen, so it's good for perovskites. And it's got a different cost structure. So they're a lot more expensive in space, so we can sell it at a high price. And the lifetime requirements is different. So we need to make the cells really efficient so we don't have to send so many cells to space and pollute the space. So anyway, we've got some cells that are on 2 cube satellites that were integrated onto a 12U deployer, then integrated onto SpaceX. And then it was launched on the 24th of August, 4:35 a.m. I was asleep, I didn't watch it. And then it got deployed into space in the low Earth orbit. So apart from that, we have great fun. We talk to the public about multi-junction cells, so you can see the boy playing a game. We just teach them what multi-junction cells look like. So he's got a dart gun shooting the different junctions. And we've got students involved, staff involved, PhD students involved. And what we're really proud is we supervise undergraduates, first year to 4th year students, and they all contribute to our world-leading research. This latest paper is contributed by many people, but Jasmine is in her first year. So she helped me look at the space debris. And there are also other papers by 2nd year students, 3rd year students, 4th year students. And finally, I'd like to thank University of Sydney for supporting me and all the collaborators and the Academy for this award. Thank you very much.
Thank you so much for sharing your exciting work. I mean, time flies when you're so passionate about a topic. Yes. And we thank you for your advocacy for solar, as illustrated by your contributions to the article on the ABC this week.
Oh, yes.
Which are on indoor solar cells.
Mm-hmm.
I'm sure there'll be lots of questions from the audience. Mm-hmm. We'll have time for a few. Andrew. Please.
Thank you very much, Anita. What do you think the limits are on multi-junction cells in terms of manufacturing? How many layers can we live with?
I think we can live with triple junction because we made them. Quadruple junction, it's doable because for group 3-5 materials, people have worked on it and, you know, they're used in space. In terms of 5, I think we're sort of just testing our limits a little bit.
Francis, a quick one,
Quickly. So congratulations on the 30%. No, I wanted to ask about the dimensions. So you're making cells that are very thin in the Z direction, and you've sent one up into space. So how many square meters can you do with perovskites on top of it?
Yeah, so our equipment is geared for 6-inch actually, but we don't usually make 6-inch because it uses a lot of materials. But all the certified cells are 1 square centimeter, and the one that we send to space are 1 square centimeter, so they're not 0.06 square centimeter that is typically reported in the papers. In the big ones, I would need some venture capitalists to help me with that.
Well, I'm very sorry we'll have to leave it there in the interest of time, but please join me in thanking Anita again. I'd now like to pass back to Glenda for the next awardee.
Next is the Gustav Nossal Medal for Global Health. This award honours the contributions made to the field of cellular immunology, antibody formation and tolerance of vaccine research science by Professor Gustav Nossal. The award recognises research of the highest standing in the field of global health. The 2024 recipient of the Gustav Nossal Medal is Professor Andrew Steer. Professor Andrew Steer is a paediatric infectious diseases physician and Director of the Infection and Immunity Theme at the Murdoch Children's Research Institute. He is an international authority on tropical infectious diseases. His research has established global community-based treatment for programs of tropical skin infections, influenced vaccine development for strep A disease, and introduced diagnostic technologies and control programs for rheumatic heart disease. Please welcome Andrew to accept the medal and deliver the 2024 Gustav Nossal Award presentation.
Thank you very much, and it's a real honour and a privilege to be awarded the Gustav Nossal Medal, and I'm genuinely humbled by being amongst so many amazing scientists during the last few days. It's quite inspiring. So I just want to talk a little bit about myself. I'm a paediatric infectious disease physician, so my main passion is the health and well-being of children and adolescents. But in fact, and I work in Melbourne at the Children's Hospital and I look after children with infectious diseases, but in fact, 90% of the world's children live in low and middle income countries. So my main work is actually in resource poor settings. And this is a photo of me in Uganda with one of our projects, where I'm often most happy, except with my family. And what's quite remarkable about this photo is this is the first time these children had ever played a jigsaw puzzle. And so you can imagine some of the challenges of explaining how you put the corner pieces in the corner and all those sorts of things. But it does sort of speak to the resource poor nature of some of the settings where we work. And my main passion is around tropical infections because tropical infections still cause the majority of deaths in our world for children. And in fact, tropical diseases are sort of divided into two. There's the big three, malaria, TB and HIV. And then there's neglected tropical diseases. And it's a bit of a David versus Goliath competition, particularly in terms of funding. So the NTDs, as I'll call them, may not cause death, but they cause considerable disability. And this is the statue that stands outside the front of the World Health Organization in Geneva. And it depicts really accurately the impact of NTDs. So the person here who has an NTD is the father who's been blinded by a disease called onchocerciasis. He cannot work because he's blind, but his son can also not work, can also not go to school because he has to look after his father. And this is the impact that we see with NTDs around the globe. So to jump to a new NTD, which is what I wanted to talk about today, is a mite called scabies. And you can see a picture there of the mite. And it burrows under the skin and causes that middle picture appearance. It's incredibly itchy, it keeps you up at night, makes it hard for you to go to school or participate in work. And it's frequently infected by by bacteria, which is the last picture, which is called impetigo. And that can go on to cause more serious bacterial infections, rheumatic heart disease, kidney disease. And what we do know is around 500 million people around the world are affected by scabies at any one time. So this is me as a younger person. During my medical student years, I was doing research on rheumatic heart disease in Samoa. And I know we've had some Pacific Islander colleagues with us here over the last few days, which is terrific. And whilst doing my research, I was expecting to see a lot of children with scabies, but in fact there were very, very few, and I started to wonder why. And what I learnt was that the previous 2 years there had been community-wide treatment or mass drug administration using ivermectin for the control of this other disease, which is called lymphatic filariasis, a parasite that's spread by mosquitoes. It had been highly effective at controlling lymphatic filariasis, and I surmised that it was probably also effective at controlling scabies as well. So you've probably heard of ivermectin. It's a terrible drug for COVID, but it's a fantastic anti-parasitic. And in fact, the discoverers of ivermectin, Satoshi Omura and William Campbell, won the Nobel Prize for Medicine in 2015 because of ivermectin's contribution to global public health. So there's been over 3 billion people who've been delivered ivermectin for the control of lymphatic filariasis, which I showed you before, and river blindness or onchocerciasis, which I also showed you before. But we wondered whether ivermectin could have an impact on scabies. So we started doing some research with colleagues in the Pacific, particularly in Solomon Islands and Fiji. And we worked with the Ministry of Health in our first trial called SHIFT, or the Skin Health Intervention Trial. And the picture here depicts cleaning up children's hands with ivermectin. This was published in 2015. And I'll just take you through the study very quickly. So we had 3 island groups. The first island group of Motariki, we went into the community, we did an examination of all 803 people in the community, and anybody with scabies, we referred to the local clinic. The second island group, we went in and we examined all 532 people to see if they had scabies, and if they, and for everybody in that community, we gave them a topical treatment, a cream called permethrin. And then a third group, we went into the community, examined all 716 people, and we provided mass drug administration of ivermectin, which is an oral tablet. And we came back 12 months later to see what the results were. So the standard group is in green, the permethrin MDA group is in red, and the ivermectin MDA group is in blue. And what you can see is that the prevalence of scabies was very high, over 30% in all of these 3 island groups. So that means that 1 in 3 of you in the audience would have scabies at the moment if you lived in these communities. When we came back 12 months later, we saw a reduction in our standard of care arm just by providing treatment. We also saw a quite large reduction in our permethrin group, but we saw a huge reduction in our ivermectin group, over 90% reduction. We didn't provide treatment for skin bacterial infection, but in the ivermectin group, just by treating scabies, controlling scabies, we were able to see a reduction in impetigo as well. So this led to more studies and more study logos, as you can see, in the Solomon Islands and Fiji, which in summary showed that ivermectin delivered to whole communities can reduce the prevalence of scabies by some 90%, quite consistently, in 3 big trials in Fiji and in 4 big trials in the Solomon Islands, involving over 140,000 participants in Fiji and over 50,000 participants in the Solomon Islands. So with this in mind, we started working towards policy outcomes. We developed an international alliance in 2013, and we supported Ethiopia, Fiji and the Solomon Islands in their application to WHO for scabies to be an official neglected tropical disease, which it was. And then in 2019, we worked with the WHO to develop international guidelines for the control of scabies. And then we formed the World Scabies Program. And we went through a process looking for funding and applied for the Macquarie Foundation grant in 2019. And this is where Sir Gus comes in. So Sir Gus supported our application and played a significant role in us being able to achieve this funding. So the World Scabies Program supports countries to implement control strategies, raises global awareness, contributes to global efforts in mapping, engages with the pharmaceutical industry, and develops implementation-ready guidance. So these are some of our team members in Fiji and Solomon Islands. So we are currently doing national programs delivering MDA in both of these countries. We've completed a first round in both countries, and we're doing a second round now in the Solomon Islands. Our starting point for the program is a prevalence of 23% in the Solomon Islands, so 1 in 4 people have scabies, and 16% of children in Fiji. We're targeting 1.6 million people with MDA, and so far we've delivered 8.6 million ivermectin tablets. And there's a whole other conversation about the complexities of these sorts of logistics. These are our teams in action. We use very high-tech dosing poles, as you can see here, painted by some of the local school children in Honiara. So a very simple approach to how we do our public health work. Our next steps are to measure impact in Fiji and Solomon Islands, and we have some initial results which are quite exciting but are not quite ready for prime time just yet. And we are expanding to other settings because the WHO has provided 2030 targets, which is to undertake MDA in endemic settings in 25 countries and provide scabies management in universal health coverage packages of care in all countries around the world. I did want to just highlight one thing, which is many of you will be aware of Cox's Bazar, the largest refugee camp in the world in Bangladesh, and there's been a huge outbreak of scabies in that camp, with 40% of people having scabies. We've worked with the WHO and other partners to provide technical support, and WHO has just finished delivering ivermectin-based MDA to 1 million people in that camp, and we hope to see positive outcomes from that work. So I'd like to thank our funders, and I'd also like to thank the many people that have been involved in this work over a long period of time, and also my family, including my wife, who's in the audience. Thank you.
Thank you very much, and congratulations again. I'm sure there's lots of questions. Yep, down here, 2 down here.
Congratulations on marvellous work and congratulations on the well-deserved award. Could you tell me how often do you think you'll have to return to communities to reapply the ivermectin treatment, and are there any side effects?
Yes, so in regards to thank you for your question. In regards to the first question, we do have some modelling which suggests it's probably between 2 and 3 rounds. There is a little bit of devil in the detail though, which is that ivermectin is fantastic at killing the mite, but doesn't kill the eggs. So it's the female mite that burrows under the skin. The male mite's just involved in reproduction and sort of not really that involved and lays eggs. So we actually have to give 2 doses of ivermectin. So we've got to give a dose today and then give a dose in another 7 days. So that actually is a complicating factor. We're involved in some research in a group from Melbourne with a new drug called moxidectin, which has a longer half-life. So we're sort of hoping that might solve one of those problems in the future. Yeah, so the general concept is that we will do 2 rounds a year apart. In fact, in Fiji, like I say, I can't talk too much about the results, but we don't need to do a second round, which is terrific. In Solomons, we're doing a second round now. But the idea is after that, we just use clinical care. So we do health system strengthening so that the clinics can manage it as a clinical problem, not a public health problem.
And the side effect?
And the side effects, it's a very well-tolerated drug. It's literally been given to billions of people. There are some restrictions about its use in younger children, which we don't think are actually based on good evidence, and we're working to bring the dosing down to young children where we think it's going to be quite safe like it is for adults.
So look, my question was related. Congratulations on a lovely talk. Do you get resistance to ivermectin? I guess you only give it once, so you probably don't.
Yeah, so ivermectin is used widely in the veterinary world, and so there are concerns around resistance for non-ectoparasites. So scabies is an ectoparasite, particularly some of the roundworms. But as you say, giving it just that one time, it's not a prolonged exposure. It's in the community the one time. So the evidence suggests at the moment that it's not going to be an issue for scabies.
Last question.
Thank you. Thank you for excellent work, Andrew. It must be very gratifying to you to see this difference. What is the reservoir of scabies? Where do people get the scabies? And is that treatable also with ivermectin?
Yes, that's a great question. So there's many varieties of Sarcoptes scabii. There's a dog form which causes mange. There's a wombat form. There's a koala form. But the human form, variety hominis, is a human-only pathogen. So the reservoir are humans. And in my mind, actually, the reservoir are kids. So a lot of the sort of, after the mass drug administration efforts, is around targeting our clinical treatment on children to reduce that reservoir. The reality is that scabies is everywhere. It's in Australia. It's just a clinical problem here, not a public health problem, like it is in these sorts of settings.
Okay. Please congratulate Andrew once again for the fantastic work. Thank you. We'll now move to the medal ceremony for the 2024 awards. Session will be led by our former Vice President and Secretary of Physical Sciences, Malcolm Sandbridge.
Thank you, Glenda. So we begin with the Mawson Medal. This award recognises research in the earth sciences and honours the contribution to science in Australia by Sir Douglas Mawson, a geologist and Antarctic explorer. The 2024 recipient of the Mawson Medal and Lecture is Dr. Adriana Dukic. Adriana, please come and join us to accept your award.
Dr. Adriana Dutkiewicz may be a geologist, but her job resembles that of a cold case investigator.
So what is this? These are deep sea nodules from the Coral Sea.
She tracks down data stored in everything from filing cabinets to diverse online portals to piece together a detailed picture of the global seafloor and its history.
Probably about 150 years ago, people still used to think that the seafloor was quite homogeneous, just a muddy pit. But we now know that the seafloor is actually quite complex, and the seafloor provides the largest substrate for a biodiversity in the world.
Remarkably, before Dr. Dutkiewicz came along, this was the best map we had of global sediment distribution in the ocean depths.
It was a hand-drawn map, and maybe only 5 different sediment types were shown on that map.
But the data uncovered by Dr. Dutkiewicz and her colleagues revealed a much more detailed picture. So, in 2015, they unveiled this, the first comprehensive map of seafloor sediment.
We use a very large data set. This is based on legacy data that have been collected over the last 50 years. This is an enormous treasure trove of data.
A lot of that data comes from scientific ocean drilling. Since the 1960s, researchers have drilled more than 4,000 holes in the seafloor, each providing hundreds of metres of ancient sediment.
And of course, there are large areas of the ocean that have never been sampled, have never been mapped. And so for these regions, we had to use machine learning to fill in those gaps in knowledge.
The digital map uses different colours to represent 13 classes of seafloor sediment. For example, in this area, it's possible to see how the Murray-Darling has impacted the seafloor composition where the river system drains into the ocean, with yellow blotches indicating where sand has settled and brown representing mud. The map can also be combined with other data sets, such as ocean circulation models, to shed light on seafloor processes, including how oceans have responded to past climate change. Dr. Dudkiewicz's seafloor map drew media coverage from around the world, but she is equally proud of another project that received much less attention. She led research looking into the movement of seafloor sediments.
We actually discovered a huge pile of sediments along the Southeast Indian Ridge in the Southern Ocean, which had not been known before.
Strong deep-sea currents had been pushing the sediment against the ridge for millions of years, creating a sediment pile 8,000 kilometres long.
So that area potentially contains a very high-resolution record of Earth's past climate, but it still remains to be drilled and properly mapped using ships.
Dr. Dudkiewicz's work in combining traditional sedimentology with cutting-edge technology has now seen her awarded the Mawson Medal from the Australian Academy of Science.
The next medal is the Anton Howes Medal. I acknowledge again Denise Howes, who's here in the audience today. The Anton Howes Medal recognises distinguished research in the earth sciences and honours the contributions to the earth sciences by the late Professor Anton L. Howes. Professor Howes was the founding director of the Research School of Earth Sciences at the Australian National University. Please welcome the 2024 recipient of the Anton Howes Medal, Dr. Andrew King.
As a climate scientist, Dr. Andrew King spends a lot of time thinking about the future of our planet. He's concerned that we might be locking in damaging climate change impacts for future generations.
I find it alarming that we still have record high greenhouse gas emissions, even decades after we've really understood the problem of climate change. We're seeing really alarming extreme weather events around the planet, and the action we're taking on tackling climate change is far too slow.
That makes the climate projection work of scientists like Dr. King all the more important. He uses observational records with climate and Earth systems models to understand the long-term impacts of continued greenhouse gas emissions and a warming planet on extreme weather events. But he's also looking at scenarios where we successfully reduce greenhouse gas emissions under the Paris Agreement.
Traditionally, climate scientists have been more focused on the risks of continued greenhouse gas emissions and global warming, and less on the consequences of actually achieving net zero emissions.
Dr. King says the path we take to net zero can make a big difference.
If we're quite quick at decarbonising and achieve net zero emissions sooner in the next few decades, then we would start seeing benefits, we think, in the latter part of the century, including reduced heat waves across many land areas, including Australia. But because the ocean is much slower to evolve, we would expect to see continued sea level rise and continued warming of the oceans.
But the outlook would be much grimmer if we don't slow our current pace of global warming.
If we are much slower to achieve net zero emissions, then we're going to see much bigger changes before we actually reach net zero.
Dr. King has been involved in many high-impact publications, including the Australian Academy of Science's 2021 report, which looked at the consequences for Australia in a 3 degrees Celsius warmer world.
We would see really big changes in the cryosphere, with major ice loss really increasing the chance of extreme sea level rises, and also potential for other tipping points in other parts of the Earth system. Every increment of global warming is raising the risk of having really severe damage.
He says communicating the impacts and science behind climate is critical.
It's a problem where the solution is necessarily collective. We need all governments to really push towards net zero emissions. That really comes about from people understanding the impacts of climate change and pressuring their elected officials to act on reducing greenhouse gas emissions.
For his contributions to climate science, Dr. King has now been awarded the Anton Hales Medal from the Australian Academy of Science.
Next, we have the Christopher Heide Medal. The award honours contributions to mathematics by the late Professor Christopher Heide. In recognition of Professor Heide's broad interest in mathematical sciences, the award is offered in one of 3 fields of mathematics on a rotating basis. Congratulations to the 2024 recipients of the Christopher Heide Medal, Professor Serena Di Piero and Dr. Christopher Lustri. May I first invite Serena to join us on the stage to accept your award.
One mathematical equation at a time, Professor Serena Di Piero is helping us better understand the world around us.
I really enjoy looking at concrete phenomena from which you can cook up a mathematical model.
That's included phenomena ranging from how bushfires move, to how animals, such as the albatross, forage for food, through to how we reshape metal, say from aluminium foil into a can. These questions can present a challenge.
The life of a mathematician is mostly going through a lot of frustration because most of the time things don't work. But that moment where you're pulling it through and something works is really a moment of intense joy.
Professor Di Piero uses partial differential equations to solve real-world problems. Partial differential equations are capable of representing a variety of complex phenomena by combining variables and their partial derivatives to describe the physical attributes and dynamic changes in a system. Take a bushfire model as an example. It might combine variables describing things such as initial temperature and the effect of the wind. In a recent project with researchers from the University of Wollongong, Professor Di Piero used equations like these to model how fires behave and spread under different conditions.
The solution of the equation, this will tell you something about the spreading of the bushfire, when it will stop, which shapes it will take, and things like that.
It's work that could help monitor and predict real-world bushfires. But Professor Di Piero is an advocate for maths even when it has no immediate applications.
Mathematics should be somehow left free to go and explore uncharted territories because you never know what's going to happen in 20 years, for instance.
Growing up in Italy, Professor Di Piero initially planned to follow her father's footsteps and become an engineer. But a high school teacher encouraged her to pursue maths after seeing she had a knack for the subject.
Many people have this impression that mathematicians are usually antisocial and that mathematics is a very lonely discipline, and so sometimes students are scared of it. But I want to say, especially to young people, mathematics is a very social activity. I've met many collaborators that are also really good friends for me.
In 2016, Professor Di Piero moved to Australia, and she is now a professor at the Mathematics and Statistics Department at the University of Western Australia. She says the mathematics community here has been nothing but warm and welcoming.
Moving to a different country is always tricky, it's always complicated. You don't feel you're home in the beginning, so that made me feel really home in a sense.
For her contributions to mathematics, Professor Di Piero has now been awarded the Christopher Heide Medal from the Australian Academy of Science.
Now please welcome Christopher on stage to accept his award.
Watch the way this ship moves through the water. When designing vessels like this, engineers often turn to applied mathematicians for advice.
What they do is they say, I have a ship, I have the ocean, what's going to happen?
The challenge is to minimise the kind of wave disturbance that can slow the ship down.
But modelling full ocean systems is incredibly complicated, and the mathematical methods that I use can unpack some of these ideas without having to do the massive simulations that are typically involved in fluid dynamics modelling.
To do that, Dr. Christopher Lustri relies on partial differential equations.
Which basically means they're equations where things can happen in space and things can happen in time, and it's all mixed together.
His methods help capture the key dynamics of various scenarios, even if he can't provide a precise answer.
The vast majority of mathematical problems can't be solved, but just because you can't solve a problem doesn't mean you can't say something really important about the physical situation it's modelling.
Collaboration is at the heart of Dr. Lustri's work. His diverse range of projects have included studying what happens when you shoot optical pulses down cables and predicting the behaviour of insect populations.
And that's actually the real joy of being an applied mathematician, is you get to dabble in all of these different areas and work with real experts and translate their problems into mathematical language to help them understand what's going on.
He first experienced that joy at university while studying to become an electrical engineer.
I was enjoying my engineering just fine, but the math subjects really captured my imagination in a way that I didn't expect to happen.
That passion has earned him a reputation for tackling even the most complex of problems. The fact he also gets results has now seen him recognised with the Christopher Heide Medal from the Australian Academy of Science. Dr. Lustri says the award shows people value his work.
I always believed it had value. I think you have to believe your own research has value. But it means a tremendous amount to me to be acknowledged by the Australian scientific community, because it means that other people have seen that there's a reason I do what I do and that they want me to keep doing it.
Next is the Dorothy Hill Medal, which honours the contributions of the late Professor Dorothy Hill to Australian earth science and her work in opening tertiary science education to women. Please welcome the 2024 recipient of the Dorothy Hill Medal, Associate Professor Ali Gallant.
Australia is no stranger to wild weather, from droughts to flooding rains. Associate Professor Ali Gallant has spent much of her career studying those extremes.
I've always been a bit of a weather nerd. Being outside in the pouring rain or the baking sun, chasing a tornado, that's where I'm at home.
Associate Professor Gallant is a climate scientist here at Monash University in Melbourne. She's investigating the complex systems behind variations in precipitation and drought and how they're changing from year to year and over decades. Her team found that the reduced rainfall that causes and sustains droughts actually comes from the absence of a relatively small number of heavy rain events. When these heavy rainfall events return, a drought breaks.
Those types of rainfall days actually only happen on a handful of days in the year. This is maybe kind of 2 weeks worth of rain in the southeast of the country. If we can understand what's going on in those very few rainfall events, our research has really shown that we can understand drought.
Australia has rich climate and weather records beginning in the early 20th century. This wealth of data allows Associate Professor Gallant and other scientists to understand how the climate has changed over time and what might happen in the future.
There is also a lot of evidence and increasing evidence that droughts are becoming thirstier. Thirstier droughts basically mean that, all else being equal, if we had the same amount of precipitation coming into the surface, we would have more coming out, which fundamentally makes droughts more intense.
Some of this data has Associate Professor Gallant deeply concerned about the future.
When we have global temperature records or even localised temperature records being absolutely smashed, heat waves hotter than we have ever experienced before. When you see those types of events, even as someone who is used to seeing these changes, it's cause for concern, and it really does make even us hardened climate scientists think, what are we getting into in the next few decades?
But she hopes her research can help policymakers and communities make sense of those changes and take action.
Science is not finished until it's communicated. A very wise colleague of mine once said that. And people don't know what they can use the science for unless we tell them about it, and unless we can tell them in a way that is accessible. And so accessible science is impactful science.
Useful research that has now seen Associate Professor Gallant awarded the Dorothy Hill Medal from the Australian Academy of Science.
Next is the Fenner Medal, which is awarded in honour of outstanding contributions to science by the late Professor Frank Fenner. It recognises distinguished research in biology. The recipient of the 2024 Fenner Medal is Associate Professor Anna Martins Siqueira. Please welcome Anna to the stage.
Associate Professor Anna Siqueira studies marine megafauna. Part of that involves going into the field to collect data on these ocean giants.
My first field trip with megafauna, marine megafauna, was with whale sharks. Yeah, so that was the reason why I came to Australia in the first place. I wanted to work with whale sharks, and that's what I did my PhD on.
As a PhD student, she dived off Ningaloo Reef in Western Australia to find whale sharks and track their movements to better understand their behaviour.
The whale sharks travel entire ocean basins if they want to, so to me that was fantastic, how an animal could just go wherever they wanted underwater. I wanted to understand how they did it, where did they go?
Questions she's been able to answer, thanks to analytical work she does back in the office. Associate Professor Siqueira developed the first global distribution models for whale sharks, capturing the full range of their movements across the global ocean. It's work that has provided important context for species assessments and been used as evidence to change their conservation status from vulnerable to endangered.
We are entering a new era where there's a focus on protecting 30% of the oceans. And so where should those 30% be? My research is helping answering that question.
Today, Associate Professor Siqueira leads a group focused on marine megafauna conservation here at the Australian National University. She's concerned that we still don't have a full picture of the threats they face.
Shipping traffic is all over the global ocean now. Fishing has a massive footprint as well. Plastic pollution, noise pollution is everywhere in the ocean. And what really worries me is that we don't know what these changes are creating, what are the impacts that they are providing to these species.
So tracking these animals remains a crucial part of Associate Professor Siqueira's research. She goes on regular trips to sites such as Shark Bay in Western Australia to tag megafauna, including sharks, dugongs and turtles. In 2020, she also founded a global initiative called MEGAMOVE, which brings together tracking data from scientists around the world.
The dataset that we've put together with MEGAMOVE involves almost 15,000 different tracks across more than 100 species.
She hopes her analyses will give us deeper insights into how we can use our oceans more sustainably and protect our marine creatures. For her outstanding contributions to marine biology, Associate Professor Siqueira has now been awarded the Fenner Medal from the Australian Academy of Science.
Next is the Frederick White Medal. This award honours the contributions to Australian science of the late Sir Frederick White. It recognises the achievements of scientists in Australia who engaged in research of intrinsic scientific merit that has made an actual or potential contribution to community interests, to rural or industrial progress, or to the understanding of natural phenomena that have an impact on the lives of people. The recipient of the Frederick White Medal in 2024 is Dr. Hamish Clark. May I invite Hamish to join us to accept his award.
How will bushfire risk change as our planet heats up? That question is at the heart of Dr. Hamish Clark's research here at the University of Melbourne. The answer is far from straightforward.
In terms of some of the things that drive fire, we're talking fuel, vegetation. We're talking, dryness. We're talking weather conditions, those kind of hot, dry, windy days. And we're talking that all-important ignition that sets things off. And climate change can actually affect all of those things.
Those effects can vary depending on the location and the fire regime in the area.
So in our forested regions where we have plenty of fuel but it doesn't always dry out enough, then if climate change is bringing hotter and drier conditions, that's really a sign that we could get more fires and bigger fires that are harder to control. In some of the more arid areas, it's already hot and dry, and making it hotter and drier isn't necessarily going to change risk unless there's also changes in fuel too.
Dr. Clark studies all things fire, from its drivers to its impacts on people, property and the environment, and ways we can mitigate risk. Part of his research looks at prescribed burning, which is widely used in Australia to help with fire hazard reduction.
We found that there's good evidence that these windows of opportunity for conducting prescribed burning are going to change in the future. In particular, as extreme conditions increase, we're probably going to be having to move closer to winter, further away from the peak fire season.
Dr. Clark's research often involves working with decades worth of observational records.
It's my colleagues who get to go out bashing around the bush or setting fire to stuff in the lab, and I'm trying to stitch together things like weather and climate change data, evidence about how fires have spread, where they are in the landscape, to really understand what makes fire tick and how we can try to coexist with it.
You can see some references up here. Here we have some important colours.
One of his projects involves building computer models that allow fire managers to simulate bushfire scenarios and weigh up different interventions. Dr. Clark's passion for his work is driven by its real-world applications.
Yes, these are fascinating scientific questions and problems, but at the end of the day, the measure of the success or failure is, are we doing things differently? Are we making a difference?
That includes making a difference at the community level. In 2014, Dr. Clark co-founded an initiative in the Blue Mountains called Science at the Local to support scientific literacy in the community.
I played basketball with a guy there who turned out to be a science teacher at the local high school. He's from New York City. He said, it's a great place, the mountains, a little bit quiet sometimes. Why don't we try starting a science in a pub? And then, 10 years later, it's still going.
For his contributions to science with real-world impact, Dr. Clark has been awarded the Frederick White Medal from the Australian Academy of Science.
Up next is the Gottschalk Medal, recognising outstanding research in medical science and honouring the contributions to science of the late Professor Alfred Gottschalk. The recipients of the 2024 Gottschalk Medal are Professor Eric Chow and Associate Professor Kirsty Short. May I first invite Eric to join us on stage to accept his award.
Sexually transmitted infections, or STIs, are on the rise globally.
gonorrhea, syphilis actually have a significant increase over the last decade, particularly affecting gay and bisexual men, but also we are seeing rises in heterosexuals.
Professor Eric Chow is on a mission to bring down those rates. Here at the Melbourne Sexual Health Centre, he's working to advance our understanding of how STIs spread and how we can better cut their transmission. He and his colleague, Professor Christopher Fairley, were the first worldwide to identify kissing as a major risk factor for contracting gonorrhea, an STI that can affect the genital area, anus, throat and eyes.
Gonorrhea is a very old disease, and it has been primarily thought to be transmitted through sex, but we're actually thinking why there has been such an increase in infection in the throat.
Their studies revealed gonorrhea can be transmitted between partners via kissing, even in the absence of sex, which has significant implications for public health messaging. Prevention is crucial, especially as treating gonorrhea becomes more difficult.
I think the main big challenges this day is the rise in antimicrobial resistance in gonorrhea. And now we are down to one last recommended first-line treatment for gonorrhea.
So Professor Chow has been exploring possible novel preventative measures to help curb transmission and reduce antibiotic use. He's also involved in work on the human papillomavirus, or HPV, which can cause genital warts and serious diseases such as cancer. Australia was one of the first countries to introduce an HPV vaccine program to schools, initially for girls and later expanded to be gender neutral.
Over time, we're doing lots of surveillance and find that there is significantly dramatic decrease in genital warts and also HPV prevalence among young schoolboys and schoolgirls. And that actually lead to that we can eliminate cervical cancer and also potentially other cancers such as anal cancer.
Australia has a relatively high vaccination coverage rate, with about 80% of schoolgirls and boys vaccinated against HPV. But Professor Chow hopes to increase those rates, particularly among groups with low coverage, such as Indigenous Australians and non-English speaking communities.
We're working very closely with the community and also with social scientists, try to understand the gap of this knowledge and try to see whether we can actually design any intervention or program that increase the knowledge.
Originally from Hong Kong, Professor Chow lived through the 2003 SARS epidemic.
That actually made me thought that public health is really, really important, and that actually drove me into public health research. And I really love this area that we can actually do research to help people, to help the community, to protect the community.
It's research that has now seen Professor Chow awarded the Gottschalk Medal from the Australian Academy of Science.
Now, before I invite Kirsty to the stage, I'd like to note that Kirsty is herself the offspring of two Academy Fellows, Roger Short and Marilyn Renfrey, who is in the audience today. And also note that Marilyn herself won the Gottschalk in 1980, so we're keeping it in the family. Kirsty, would you like to join us on the stage?
Associate Professor Kirsty Short never thought she would become a scientist.
My parents are scientists, so I always said that that's not something I'm going to do.
But whilst applying to study international relations at university, she was persuaded by her mother, renowned biologist Professor Marilyn Renfrey, to do a science degree on the side.
I selected microbiology and immunology, and as of the first lecture, I was absolutely hooked. I knew that that's what I wanted to do with my life. And it was a really pivotal light bulb sort of moment. But it was also quite sad because it meant that I had to go back to my mother and say, turns out you were right, I do want to be a scientist.
Since then, Associate Professor Short has gone on to become one of Australia's leading experts on respiratory viral infections, such as COVID-19 and the flu. Her work has helped us better understand pandemics, including how we can prepare for future outbreaks.
One of the challenges about pandemic preparedness is we don't know what the next pandemic will be. Could be a coronavirus, could be an influenza virus, could be a completely new virus that we've never seen before. We don't know. So we really need to have things in our arsenal that work across multiple viruses.
That includes antiviral drugs that target features of a virus shared across multiple virus families. Associate Professor Short is also looking at mechanisms that target the host and help reduce the hyperinflammation associated with severe viral infections.
And specifically, they try and get that inflammation back in balance so that you've got enough inflammation to kill the virus, but not too much that you're getting tissue damage.
Her research also demonstrated a link between metabolic conditions such as obesity and diabetes and severe infection. Her team found that being overweight, obese, or diabetic impairs your immune response to the flu and COVID-19, meaning you're not only at higher risk of severe infection, but also at higher risk of reinfection.
But interestingly, that doesn't apply to the immune response to vaccination. The immune response to vaccination seems to be pretty consistent between overweight and healthy weight individuals.
It's work that she hopes will influence public health policy considerations here in Australia.
We now have a population where people are frequently living with one or more chronic medical conditions. We need to integrate that into our policy.
Collaboration is a key part of Associate Professor Short's work, and not just with policymakers.
It's really important to have clinicians involved because they've got the first-hand experience of treating patients. It's really important to have the patients themselves involved, in particular for conditions like long COVID, where it's really been patients who've been defining the condition and saying to the medical community, this is something important to look at.
Associate Professor Short says she's now thankful her mother pushed her to pursue a career in science.
Our experiences in science have been very different, in particular as women in science, because some of the discrimination my mother went through, I can't even imagine. And so I think she's really happy to see that sort of come full circle and that her daughter is able to succeed in this field now.
And in another full circle moment, Associate Professor Short has received the same honour that her mother did back in 1980, the Gottschalk Medal from the Australian Academy of Science.
Next is the John Booker Medal in Engineering Science, which recognises outstanding research in engineering mechanics that addresses problems in the static and dynamic response of physical systems within engineering and applied mathematics disciplines. It honours the memory of Professor John Robert Booker, who worked in the area of theoretical geomechanics and was Professor in the Department of Civil and Mining Engineering at the University of Sydney and held a personal chair in engineering mechanics at the University of Sydney. I'm pleased to invite the 2024 recipient of the John Booker Medal to the stage, Associate Professor Linning Arnold Ju.
Associate Professor Arnold Ju had just begun an undergraduate degree in theoretical mechanics and mechanical engineering when his entire world was turned upside down. His dad suffered a heart attack at the age of 54. He still remembers the conversation he had with doctors about the link between his father's heart attack and fluid mechanics in the bloodstream.
And then they said, well, but we don't know why, this is associated with the clotting issues, causing your dad a heart attack. Maybe you can research this.
It was a challenge he couldn't turn down. So he switched his academic focus to what was then the still emerging field of biomedical engineering.
All this whole career transition decision was actually purely based on, I think this thing, I need to be motivated, to devote that to my family.
15 years later, Associate Professor Ju now understands a great deal about the biomechanics and mechanobiology of the bloodstream.
Hello guys.
He's used this expertise to develop nanotools like this single-cell biomembrane force probe, which can physically manipulate platelets, tiny cell fragments that help in the blood clotting process.
And then we can use them to understand how these little platelets get activated in response to shear force changes and mechanical environment changes. Our circulatory system, the cardiovascular system, have lots of forces. The cardiac contractility, blood pressure, shear, stress. There are a lot of forces in our body. But these things, to keep your life ongoing, keep the cardiovascular system ongoing, you need these forces, to push the flow, to move it forward.
In the case of our heart, if blood isn't flowing properly, platelets are more likely to form clots. Professor Zhu and his team have studied that process by applying different forces to blood samples.
And this was imaged in real time through the microscope, and we can see that this stenosis device, we can see very dynamic responses in the way blood clots are formed.
He's now assembling an interdisciplinary team here at the University of Sydney, modelled on his own diverse skill set.
I did my first training in the pure mechanics, and my second trainings in the biomechanics, and my third training in pure medical science. And now I'm leading my teams with everything combined.
The group's mission is to develop a point-of-care blood clot test that can detect dangerous changes in blood flow.
That's very useful because this heart attack or stroke oftentimes happens, people got caught by surprise.
Associate Professor Zhu wants the final product to be as accessible as the blood glucose test kits commonly used by those with diabetes, meaning it would be simple enough for people to self-monitor, reducing the need for specialist appointments or hospital visits. He believes that a test like this might have provided his father with enough warning about his risk of heart attack to allow him to seek preventative treatment. Associate Professor Zhu's personal crusade with societal real-world impacts has seen him awarded the John Booker Medal from the Australian Academy of Science.
Next is the Lefebvre Medal for Research in Chemistry, which commemorates the work of the late Professor Raymond James Wood Lefebvre. Its purpose is to recognise outstanding basic research in chemistry. Please welcome the 2024 recipient of the Lefebvre Medal, Professor Yao Jun.
For Professor Yao Jun, this beach is much more than a place for a casual walk. It's the source of a vital ingredient for one of his most groundbreaking projects, splitting seawater into green hydrogen.
We need the hydrogen to power the vehicle. When we're talking about the target of 2050 carbon neutral, we need a lot of hydrogen. So that means we need a lot of the water.
Scientists have been able to free hydrogen from water for the past 200 years using a technique known as electrolysis. But producing hydrogen at scale requires large volumes of purified fresh water. This usually means drawing from precious freshwater reserves or relying on costly and energy-intensive processes to desalinate seawater. So Professor Jung wanted to find a way to use seawater without the need for pre-treatment.
At the beginning, we ran the seawater from the Hunley Beach, but unsuccessful.
Pre-treating the seawater is usually necessary to remove the salt. Without desalination, the same process that separates hydrogen from water molecules also converts the salt in the water to chlorine gas. The corrosive gas degrades the electrodes and catalysts, causing the electrolyzers to fail. Professor Jung's team here at the University of Adelaide set out on a mission to develop a new catalyst to avoid such reactions. They became the first in the world to succeed.
The catalyst is robust to the seawater. And we introduced a new concept. It's called the local environment. Using this concept, we can use the engineer the catalyst and using the conventional electrolyzer to run the seawater electrolysis to produce the green hydrogen.
As you can see here,
The coating that they developed to synthesise their catalyst can also be used with existing commercial catalysts.
The commercial catalyst can't run the seawater, only the pure water. But with our advanced coating technology, people can run the commercial catalyst in a commercial electrolyzer with the seawater electrolysis. So that way can significantly speed up the commercialization of this technology.
The team is working closely with local industry to scale up this exciting technology by using South Australia's coastal seawater and renewable energy resources. Professor Jung says his achievements wouldn't be possible without the people around him, including his wife.
My wife is also a scientist. She did the computation work. I did the experiment work. We start to collaborate during our PhD study, and we still do the collaboration until now.
Collaborative work that has now seen him awarded the Lefebvre Medal from the Australian Academy of Science.
Next is the Palsy Medal, and again I'd like to recognise members of the Palsy family in the audience. The Palsy Medal recognises distinguished research in physics and commemorates the work of the late Joseph Palsy. Please welcome the recipient of the 2024 Palsy Medal, which is Professor Jacques.
When COVID-19 first swept across Australia in 2020, Associate Professor Jacques was racing against the clock.
We saw an opportunity using our tiny and light-emitting materials for detecting the virus.
She was part of a team at UTS that knew they had the technology. The trick was ensuring it could be modified to rapidly detect COVID-19.
This is the first generation device we make for detecting the SARS-CoV-2 antigen.
The technology was more sensitive than other tests available at the time, able to identify viral proteins at very low levels. It was also able to return a result in under 15 minutes, much faster than the standard PCR test that took several hours to process. It's the type of translational science Associate Professor Zhou is renowned for.
I naturally love solving problems, so science provides me the tools to uncover the wonder of nature.
But she started out in fundamental science, observing the characteristics of nanoparticles. Her challenge was to make nanoparticles even smaller while retaining their useful optical properties.
I found a very cool way to make the tiny and light-emitting material even more amazing.
That often means working in the dark. Her techniques turn what would normally be invisible light into something we can see.
The cell type is different.
That's enabled Associate Professor Zhou's team to create thermometers so small they can measure the temperature within living cells. The technology not only helped realize the COVID-19 rapid antigen test, but also caught the attention of a major milk producer.
They want a very sensitive and rapid method to detect the A1 milk protein.
Milk typically contains a mixture of A1 and A2 proteins, but there is a growing market for milk with just the A2 variant. Associate Professor Zhou's technology has been shown to detect the protein reliably and quickly and could be used to test for A1 contaminants throughout the production chain.
I feel the responsibility to keep pushing forward the boundary of science and technology.
Her ever-expanding list of achievements has now seen her recognised with the Palsy Medal from the Australian Academy of Science.
And finally, this afternoon, the Ruth Stevens Ghani Medal. The award honours the contribution to human cytogenetics of the late Ruth Stevens Ghani. It recognises distinguished research in human genetics, including clinical, molecular, population and epidemiological genetics and cytogenetics. The recipients of the 2024 Ruth Stevens Ghani Medal are Dr. Sonia Shah and Dr. Stefan Vevold. First, may I invite Sonia to the stage to accept her award.
Heart disease is one of the biggest killers in the world, claiming about 18 million lives each year. But a growing number of people who get heart attacks don't have any of the traditional risk factors.
So they have healthy lifestyle, they don't have high cholesterol, high blood pressure, but they're still getting a heart attack. And it could be that, that part of it comes down to their genetic risk.
Here at the University of Queensland, Dr. Sonia Shah is working to answer some of the big questions around that genetic risk. She uses cutting-edge technologies to analyse the genomic data of thousands of individuals, all with the goal of finding which genes ultimately contribute to heart disease. Her previous research has led to major findings, including on a genetic disorder known as familial hypercholesterolemia.
This is a condition where individuals have very high cholesterol levels, and they tend to, if it's not treated, they can get heart attack or heart disease much earlier on in life.
Familial hypercholesterolemia was once thought to be a monogenic disease, one caused by inheriting a single large alteration in the DNA of a single gene. But we haven't always been able to identify these mutations when looking for them in patients with familial hypercholesterolemia. Dr. Shah's research revealed that in some of these patients, the disease may be a result of the combined effect of many small alterations to the DNA across many different genes, what we call polygenic. This shift in understanding prompted a major UK healthcare body to update its guidelines on managing the condition.
Traditionally, we've used very much a one-size-fits approach to treating individuals, but we know that our genetic makeup makes us unique, and so our risk of getting disease, our response to treatment is partly determined by our genetic makeup. And what we're trying to do is incorporate that information to better tailor the treatment and management of patients.
Dr. Shah was also the lead analyst for the HERMES Consortium, an international research collaboration investigating the genetic basis for heart failure. She co-led the largest genetic study of heart failure at the time and worked with researchers from around the world to discover new risk factors for the condition.
We now have 150,000 heart failure cases, and we're starting to uncover more genes that have not previously been linked to heart failure.
This research could be used to improve heart disease risk assessment tools.
Having genetic data being incorporated into these predictions will help improve our ability to identify high-risk individuals and our ability to prevent disease in the population.
That's not the only avenue Dr. Shah is taking to improve risk assessments. She's also passionate about addressing biases in genomic research.
We know that cardiovascular risk, the presentation of symptoms, also response to treatment can vary based on whether you're male or female, but also depending on your ancestral background. So if we don't address that lack of diversity within research participants, this actually leads to health inequity.
In one project, she engaged the Australian South Asian community to better understand barriers to their research participation. It's work that sits close to Dr. Shah's heart.
I have family history of cardiovascular disease, but also as somebody of South Asian ancestry, that's something that's become really important for me to be involved in and to make sure that we're taking those steps to address that health inequity.
For her contributions to human genetics research, Dr. Shah has been awarded the Ruth Stevens Ghani Medal from the Australian Academy of Science.
Now please welcome Stefan to the stage to accept his award.
Cancers such as leukemia are notoriously hard to treat. But a better understanding of what causes and drives their growth could open up new avenues for cancer therapy. That prospect has underpinned much of Dr. Stefan Vervoort's research over the past decade. Here at WeHi in Melbourne, he heads a lab that studies gene regulation, the systems that control when and how genes are expressed.
This process is critical for our normal development and how we respond to our environment,
And when that process gets disrupted, that can cause human diseases.
Dr. Vavort focuses on a key component of gene regulation known as RNA polymerase II. Much like a photocopier, this enzyme reads our genetic code and sends faithful copies of it as messages to our cells in a process known as transcription. These messages act as instructions, directing processes within the cell.
When this photocopier malfunctions, that's when things start to go wrong. And really importantly, that fuels oncogenic or cancer growth.
Dr. Vavort's team discovered that the transcription process has several distinct checkpoints.
RNA polymerase II can do about 2,000 to 3,000 base pairs per minute, so it goes really, really quick. But it also needs to ensure that there is no mistakes in the copying of that message. Those checkpoints are there to ensure that at each point everything is still okay.
When a checkpoint fails or is compromised, that can lead to errors in the messages sent. One example that Dr. Vavort has studied is CDK12, a signalling molecule that regulates one of these checkpoints.
And when this one is lost in solid cancers, pretty much RNA polymerase loses the possibility to make long genes in our cells. And the long genes happen to be heavily involved in fixing DNA damage. And so when that DNA damage response doesn't work, you accumulate mutations, and that then results in cancer.
It's fundamental research that could help pave the way for novel anti-cancer drugs that block malfunctioning parts of the transcription process.
We've now made discoveries around this CDK12-related checkpoint, for example, where we've identified critical genes that are causing resistance and sensitization. And we believe that those might be prime candidates for therapeutic targeting down the line, and we're currently exploring opportunities to actually go down the route of drug discovery.
Originally from the Netherlands, Dr. Vavort moved to Australia after completing his PhD.
When I finished my PhD, I felt like I can go anywhere in the world, but where do I really want to work? That's what drew me back into Australia, just the possibility to do groundbreaking science, but maybe even more importantly, do it in the Australian way, I would call it. Australians have a really good way of having fun, doing what they love, but doing it in an exceptional and thorough way anyway.
For his impactful research in human genetics, Dr. Vavort has received awards including the Snow Medical Fellowship and the CSL Centenary Fellowship. He now adds the prestigious Ruth Stevens Garney Medal from the Australian Academy of Science to his growing list of accolades.
Thank you, Melbourne. Oh, it's that time of day. Thank you, Malcolm. Can we please have a final round of applause for all of our awardees? Before we draw to a close, I have the pleasure of announcing the winner of the Grant Ed Group's competition that's been running across Science at the Shine Dome this week for early and mid-career researchers. The prize is a place on Grant Ed's Sprint Research Grant Development Program that's worth up to $8,500. And the winner of the program is Dr. Nicola Strayton from the Australian Catholic University. Congratulations, Nicola, and we'll be in touch with you after Science at the Shine Dome to ensure you're able to access that opportunity. You will also be received... We would also like to ask for your feedback on the event in order to make improvements going forward. There is a QR code on the screen. You will also receive an email. It's a short 5-minute survey. I know everybody gets surveyed every time they do something these days, but we would really appreciate if you could spend a couple of moments doing that perhaps on your way back home while it's all still fresh in your minds. May I please extend heartfelt thanks to our fellows, all of our awardees, their guests, family members, everybody who has joined us this week, our event partners and others who have made this such a memorable occasion. Thank you. For our guests who have travelled from interstate, your luggage is now available at the front of the Shine Dome, and coaches have been arranged to transport you to the airport at 5 p.m. We hope that you have enjoyed Science at the Shine Dome 2024 as much as we have.
I'd like to personally thank our President, Jagadish, our Executive Committee and Council members for all of the time and preparation they have put into delivering this event, as well as the many staff members across the Academy, led by the events team, Lisa Crocker and Jamie Evans. I think you'll all agree they've done an absolutely splendid job, and I'm certainly very, very proud of them. I personally wish you safe travels, and I look forward to seeing you here in the iconic Shine Dome again in the not too distant future. But Jagadish, for you to close.
Thank you, Anna Maria. Before closing, I would like to acknowledge 3 past presidents, those are in the room, and then Professor Brian Anderson, Professor Suzanne Cory, and Professor Andrew Holmes for their advice and guidance and mentorship to me, being the experienced people. When you're gaining the experience, you don't have the experience, but they have the experience. I'm benefiting from their experience in terms of whenever I have a question, I go to them and they're always generously giving time to me. And I want to thank all 3 of them. Those are here. Please join me in thanking on my behalf as well. Of course, the Academy's work, we've had last 4 days, excellent excitement of science and learning from early career researchers, mid-career researchers to career researchers, and of course fellows, and it's been wonderful 4 days. And so I also would like to take this opportunity to thank the council members and ExCom members, not only present ones, also past ones. We invite them to come back and help us at the Science at Shine Dome or so. They put a lot of passion to the Academy. They dedicate their time. They're willing to give any time they need, Academy needs. And please join me to thanking all ExCom and council members, current ones and the past ones as well. Because if you want the Academy to really work outstandingly, in addition to fellows, there's a group which really makes these things happening, and then they are our staff members. In fact, they're doing lots of background work, and then you don't see them. Some of them may be behind the cameras or somewhere else, but they're here with us. And could you please join me thanking all of them? I would like to invite all the staff members, all of them to please come forward so that we can show our appreciation to all of them. Please, all the staff members, please. Anna Maria, Melissa, Shona, all of you, please. Thank you, and keep up the good work, and have a good trip back home. Thank you all. Thank you all of you. See you next time. Thanks a lot.
Hello, take a seat. Welcome to your studio.
I'm ready. Fellows, interview, take 1.
I do research in accelerating the energy transition.
We use venomous animals to develop eco-friendly insecticides.
My research is focused on corals.
I use computers to try and understand how things in space work.
And I study plants that kill.
My father realised that I had an interest in science. He was a politician and he brought me to Canberra quite often, and he took me across to the Shine Dome. It was the centre of academic life for scientists. It stayed with me, that memory. But of course, back in 1968, it was really out of the ordinary.
Science is a systematic way of thinking about the world.
That camera that found everything is because of science.
Science matters because it's fundamental to truth.
I want to become a scientist to do something that can change the world.
When you look at the Earth from the space, we have to remember there's no borders.
It's about what you can contribute to the community, not just your local community, but the global community.
I'm passionate about sharing science with the world. To do that as a fellow of the Academy of Science means an awful lot.
The opportunity to really present Australia on the international science platform. It's a very, very special feeling.
We really need to advocate science at every opportunity in order to make sure that evidence is brought to the table in decision making, and the Academy's really good at that.
It just feels exciting to be in a position, being a part of the Academy, that I'm able to have greater impact.
Science is this fantastic way of finding out about stuff that's independent from your own prejudices. And it's just such a powerful tool, probably the most powerful way of finding things out that humans have ever invented.
Research fulfills our role as pioneers in the grand journey of discovery.
Whatever problem we're faced with that really matters to humanity and to our planet, the answer is going to be in science.